Io c'ho già il mal di pancia.
Ah, meno male, ok.
No, no, è che classico, no, no.
Da un lato forse la sala, quella con le costate, era più contenuta, l'hai vista?
Non la voglio vedere, no, se mi
È
parli
bella, però qua è top, ragazzi, è molto bello.
Buongiorno. Posso, scusate, chiedervi se durante le domande ci sarà qualcuno con i microfoni? Perfetto, grazie.
L'ho trovata qua.
E se vuoi ho una bottiglietta chiusa. Ok, però se...
Guarda, lì per terra c'è. Senti, se si mette lì, dove no, non vedrei.
Vai. Così?
Sì,
Ok.
non so se gli speaker devono andare giù.
Forse vanno giù perché sono solo i due,
Sì, sai,
però...
devi andare giù. Vado giù. Lui fa il concerto con me perché non c'è specchio. Ah, ok, ok, ok. Vado giù.
Se ti dà fastidio, ho messo lo zaino, credo là sotto, l'ho sposta
Cosa
però.
c'è?
Il mio zaino era se ti dà fastidio vedere le gambe, lo sposto.
Aspetta, devo prenderlo io così.
Grazie.
Che ore sono? 8:20. Oh, a voglia. Allo? Sono first line. Sì, già. Mi so che sì. Grazie. I moderatori siete voi?
Sì, però non c'è lui, ci sono io, ovviamente non c'è il mio nome, ma io sono uno speaker.
Ah, ok.
Ma non
Uno speaker.
e
Allora
faccio...
forse ce l'abbiamo.
Ok?
Ok,
Sì, sì, sì.
allora cambiamo nome.
Non è cruciale, però grazie. Però
Quindi...
ho tolto quello, l'avevo messo così in modo per non dare
infatti,
adito che fossi
no.
un impostore.
Ah, facciamo cosa?
Grazie.
Michele, allora tu presenti i primi due,
Sì.
perché se io torno almeno tu già presenti Mavi e io presento te e Laura.
Quindi sei tu, Mavi, io e Laura.
Io Mavi, poi ti presento io. Ok. Wow. Ah, ci dobbiamo presentare.
Piacere.
Si devono presentare, ok?
Chi si deve presentare?
Allora io faccio il benvenuto
Ok, ok.
e poi
Sono
sì,
Di qua.
alla fine potresti, visto che fai il benvenuto, puoi dire sono...
esatto.
Poi l'introduzione è rapida, ti dico prima...
Sì, io ti dico
Sì.
Silvia Marinelli e
Mamma mia, aspetta.
Tutto in inglese, giusto? Ok, ok.
non ci vedo niente senza te. Anche leader, veramente sì, the investigator, Bradley, and my colleague Michele Giuliano e tu dici che sei quello che sei. Esatto,
Chi sono?
mi tolgo subito. Poi figurati, questo bicchiere è vostro?
Mi fa
Cosa
senti Silvia?
No, ma alla fine è solo una forma
Fanne un po' così.
per
Tutti.
riempire gli spazi, non pensare che sia così cruciale. In teoria
Ti
dovrebbe
ringrazio.
essere che il chair è quello che anima se non ci sono domande, questo ci proviamo, oppure dovrebbe interagire storicamente, ma anche adesso è next. Quindi non è così formale. Un tempo era più, forse nei congressi o comunque come is. 8:30 is, but we have the proper lighting, so we have to be awake and performing even. We have no excuse. Okay.
Non so se dobbiamo iniziare a chiedere anche se la sala è vuota, iniziamo.
Iniziamo sharp,
Sì, penso sì. Ok.
perché non so se c'è un simposio dopo che inizia esattamente quando finisce il nostro, o magari 10 minuti
C'è
dopo.
poco tempo, sì, con quello dopo, quindi dobbiamo iniziare qualche tempo sharp. Ok. Magari c'è un paio di minuti, potete aspettare. Posso spostare il nome? Certo, scusami. Grazie. Signore benedetto.
No stress, no, è divertente, è una cosa carina. Siamo fra amici, sono gente bella, brava, amabile, riuscita, quindi sarebbe stato
Sì,
un casino
esatto.
se non ci avesse fatto vedere. Quindi
Abbiamo dei risultati sulla drag
molto bene. E quindi in qualche modo avete una qualche autorizzazione o una strada per poter dire, visto che funziona in vitro, si può provare sul
ex vivo
paziente.
funziona,
Sì,
perché
ex
sennò...
vivo si può provare sul paziente o
Sì,
potenzialmente
perché comunque puoi usare i drag
già
ripresi
dovrebbe.
per la gerontologia. Vabbè, comunque, sono un po' provocativa.
Nelle prossime settimane o mesi quell'incontro di lavoro
Sì, ti
a
ricordi, avevo
Modena,
mandato il
sì.
video, però poi da lì
Jonathan si è urtato perché non era i giorni che forse non sarebbe stato. Ho detto ok, lo spostiamo, non c'è da essere da offendersi.
dobbiamo fare i prove in modo.
Sì, assolutamente sì. Lui sosteneva no, assolutamente bisogna che invitiamo assolutamente i clinici. Sì, ma non è una cosa così formale, più secondo come me l'avevi detto e come ce l'avevamo immaginato, qualcosa di informale per magari coordinarci, visto che...
Prova. Aspetta, abbassa un po'. Perché rimbomba. Prova. Vabbè, ti provo i microfoni del tavolo.
Buongiorno.
Prova.
sa che ti stanno salutando. Comunque,
Ciao!
confermo che potete aspettare anche 5 minuti in più. Ok, grazie. Possiamo aspettare 5 minuti in più, ho detto.
Fai magari 2 minuti, in modo tale che nel tempo che parliamo si fanno quegli altri, si fanno i 5 minuti e tu inizi i 5 minuti ai 5 minuti, ai 35.
No, non ho problemi.
Questo bla bla di presentazione lo possiamo iniziare fra 2 minuti.
Loro hanno detto...
Quello che intendo è che l'introduzione non è contata nei 20 più 20 più 20 più 20. Sei già abbronzato, pronto per ventotene, nel caso ci sia sole.
Nel caso.
Quindi ti dicevo che potrebbe essere che i 20 più 20 più 20 più 20 minuti escludano questi 2-3 minuti di introduzione.
Sì, ma allora ha detto che possiamo iniziare
fare,
tra
ok.
5 minuti, quindi va bene.
Adesso capisco perché hanno dato il ventaglio, perché effettivamente fa caldino qui dentro. Pensavo fossi io che ho corso per venire, ho camminato rapidamente per venire qui, ma no, fa caldo proprio.
Good morning. Thank you to be here. Welcome to our symposium. Let me introduce myself. I'm Silvia Marinelli, a principal investigator and scientific coordinator of EBRI, European Brain Research Institute in Rome, and I will chair this symposium together with my colleague, Michele Giuliano.
Hello to everyone. I'm Professor of Bioengineering at the University of Modena and Reggio Emilia as well. I retain a small appointment, temporary appointment at Pisa in Trieste. And today we have 4 talks, all focused on the fascinating field of studying directly acute human tissue in terms of structural and functional activities. And the first talk is by my colleague Silvia Marinelli and is entitled New Therapeutic Strategies for Drug-Resistant Epilepsy, Human Brain Slices Recordings.
Okay, so before I start to present this study, I would like to introduce all people that have contributed to obtaining this interesting data. And first of all, I would like to thank Professor Enrico Cherubini. He's the head of the lab, of the joint lab, and he's had this visionary mind idea to set up this lab in the Bambin Gesù Pediatric Hospital. And then I would like to thank Michela Giustizieri, the postdoc of the lab, and all the electrophysiological data that I'm going to show has been carried out by her. And also Sabrina Petralla, he's a PhD, a postdoc of the lab, and he carried out all post-hoc immunoidentification and performing all acute slice, brain slice and organotypic brain slice. And lastly, Antonino Cattaneo, he's strongly believer in this project, and especially he strong set up the human brain platform at EBRI, that is a very strategic platform. On the side of Bambin Gesù Hospital, these are the people involved, and there are all experimental neurologists, and the co-head of the lab is Nicola Specchio. And they all belong to the neurological and neurosurgical disease unit. So our studies focus on focal cortical dysplasia, and this cortical dysplasia is, has shown in these 2 tables, is the mostly common cortical dysplasia around the form of seizure, epilepsy drug resistance form. And among the focal cortical dysplasia classification, the most common is the type 2, that is characterized, comparing the other 2 forms, by dysmorphic neuron and balloon cell. In this slide is show how these patients that are affected by this dysplasia have an impairment in layer stratification of the cortex, and and the appearance of these neurons that are not contained in normal cortex, such as balloon cell, dysmorphic pyramidal neuron, and cytomegalic neuron. And here on the side, there is immunohistochemistry assay in which is shown all these cellular characteristics. So the aim of this study is to use human tissue to gain insight on the mechanism underlying drug-resistant form of epilepsy, and to use this mechanistic knowledge to screen drug and develop new treatment strategies. So how this focal cortical dysplasia is detected? So the clinician follow specific steps. First of all, they assess by multimodal neuroimaging evaluation the localization of the dysplasia, in this case in the frontotemporal lobe, by using MRI and PET. Then they perform EEG and in vivo and also acute recording directly on the tissue during the surgery. And this type of electrophysiologists help to detect the exact localization of the seizure loci, surgical guidance, and ensure complete resection of the dysplasia. The second assessment is genetic testing. Since the focal cortical dysplasia can also due to a genetic mutation. Indeed, 60% of this form rely on a mutation in the mTOR pathway. And our results show that around 24% are due to a mutation and to the TSG2 gene that is a loss of function mutation, and mTOR mutation that is a gain function mutation. And of course, we have still ongoing result regarding this genetic testing. And finally, we have a confirmation on histological assessment in which the neuropathologist do specific marker, such as vimentin, that is a marker of balloon cell. And in this table is summarize all patients that has been enrolled in this study and from which we obtain tissue for our electrophysiological recording. And as you can see, the patient has been treated with sodium and calcium channel blocker drug, and also drug that potentiate GABAergic transmission. The resection of, so the surgery is induce a free seizure, so kids that undergo surgery are free of seizure, most of them. So here in this slide is represent a workflow of the preparation of human acute brain slice. And Laura will explain better in her talk. So after surgery, we bring the tissue in our joint lab at Bambino Gesù Hospital. And the transport is a very critical step for the health of the tissue. Then we resection the tissue and we perform electrophysiological recording, both in voltage and current clamp. Here is an example of resect tissue. This is an example of dysplasic tissue, and this is an example of tissue that has been removed to have access to the focus loci. And here there are some slides in which it has shown how we characterize this tissue. So we record the normal appearing pyramidal neuron, and that appear with this typical action potential and have this inhibitory spontaneous GABAergic current. And this recording has been performed from a pseudonormal tissue. Then we are able to record the putative balloon cell these cells are not epileptogenic, and they lack of both sodium and calcium channel. And then we have this low threshold cytomegalic neuron, and this neuron are those that are believed to induce epilepsy. And then we can also record putative immature neuron and also dysmorphic cytomegalic neuron that, as the previous one, can be induced epilepsy. And here is a summary of the passive electric properties of this recording neuron, and both the resting membrane potential and the membrane capacitance. And here is a summary of GABAergic spontaneous current that are different between cells that we recorded. After recording, we perform post-hoc immune identification, and in this tissue, we found that there is an altered GABAergic signaling compared to the pseudo-control tissue, and we assess this GABAergic component by using two types of antibody, antiparvalbumin and GAD. And then we also perform some experiment by using a GABA transporter antibody, and as you can observe, this antibody is also expressed in the nuclei of the cell, and not only around the membrane, has been described in the literature. Also, this post-hoc immunoidentification confirm our electrophysiological experiment in which these tissues are rich of balloon cells. These balloon cells are cells that express genes from both glial and neurophenotype. And then we do characterization by using an antibody that is a marker of anti-tau phosphorylation, hyperactivation. And as you can see by the white arrow, this neuron express this mTOR hyperactivation signaling. In this slice, in this type of slice that belong to the focal cortical dysplasia, we have a very low incidence to detect ictal-like activity, and so this is the reason why we use convulsant agent to induce interictal burst. And in this case, we use 4-aminopyridine, that is a blocker of potassium channel. And in this paper, recently published, it has been shown that epilepsy activity in malformation or cortical development express an alteration, a perturbation in chloride homeostasis, as shown by the two chloride cotransporter, KCC2 and KCC1. These chloride transporter during development have the KCC1 is highly expressed, and this is the transporter that guide the influx of the chloride intracellular. So in the immature neuron, there is a high chloride, and so the GABAergic response is excitatory instead of inhibitory. And we confirm this altered balance between these two chloride cotransporter in our section, as shown by this yellow arrow, in which we show this white arrow, in which we show that the transporter and KCC1 is more expressed than the KCC2. And so our idea was that if we block the NKCC1, that increase the chloride intracellular, and so induce GABA depolarization, we can induce epileptiform discharge. So if we block this transporter, we can block and we can counteract epilepsy. And so we test bumetanide, that is an FDA-approved drug. This drug has been approved for the treatment of edema and is known as a diuretic. However, there is in literature has been performed this clinical trial on neonatal seizure, and the result from this clinical trial was positive, and meaning that bumetanide could be effective also in focal cortical dysplasia. And here our experiment. So this is an epileptic activity endogenously that we were able to record in our sample. And when we applied bumetanide, we were able to observe a reduction in the frequency of this endogenous epileptic burst. And interestingly, when we applied picrotoxin, that is a GABAergic antagonist, in particular is a chloride channel blocker, we still decrease the frequency of burst. And here is another example in which is shown how by blocking GABAergic transmission, we have an amelioration of the epileptic activity, contrary to what it has been shown in literature. And these bumetanide do not affect cell intrinsic excitability, since it's not acting on voltage gate ion channel function. And is blocking also affect the GABAergic spontaneous that we record from this cell by affecting both frequency and amplitude of this current. And the protective bumetanide effect, we were able to detect also in epileptiform burst induced pharmacologically by 4-aminopyridine, and as shown by this spectrogram, you can observe that both bumetanide and picrotoxin were able to decrease the frequency of epileptic discharge. And also in the pharmacologically induced epileptiform burst, bumetanide do not affect intrinsic excitability of neuron. And also in this experimental condition, bumetanide and picrotoxin were able to decrease both frequency and amplitude of this GABAergic current. So summarizing our result, we can point to that focal cortical dysplasia tissue show aberrant GABAergic signaling due to an increase in intracellular chloride and putative excitatory GABA, that blocking NKCC1 control interictal activity, suggesting NKCC1 upregulation in functionally implicated. Both spontaneous and pharmacologically induced bursting discharge are triggered by GABAergic transmission rather than block. And so we can say that bumetamide as well as GABA antagonists may be effective antiepileptic medicine. So all this data has been partially curated, and they are available for the building of the computational model, in particular for model brain with epilepsy. And this is the take-home message. So is cortical dysplasia truly a drug-resistant form of epilepsy? In these are ongoing experiments. We are working on human organotypic slice culture. And this type of preparation allows you to do chronic drug treatment and also to induce expression of protein through viral infection. And this is our next study in which we want to assess if epileptic engram may contribute to the recurrence of seizure by using a synaptive toolbox. That is a toolbox that allows you to manipulate potential spine. Thank you for your attention.
Thank you very much, Silvia, for the excellent talk. While I invite the next speaker slowly to approach the podium, I think we have time for one question. Then I will ask, please. Middle of the room.
Thank you very much. I may have lost it, but what are the neurons from where you made the recordings? Were they the giant neurons or apparently normal neurons?
Okay.
And did you, in case you use one of the two, check the other?
Okay, you mean the recording with endogenous epileptic fibrosing? This neuron, putative cytomegalic neuron or dysmorphic neuron that are the epileptogenic one, and they have the intrinsic electric passive property that relate to this type of neuron.
Okay. And did you check also apparently normal neurons? Do they have the same?
No, we haven't checked on, because apparently normal tissue are very rare, and once we obtain this tissue, we will do it. Thank you.
Let me thank again Silvia. So the next speaker is Professor Mavi Sanchez-Vivez, who is principal investigator at the Institute of Biomedical Research, August P. Sunyer, in Barcelona, Spain. And her talk is entitled Spatiotemporal Network Dynamics and Structural Correlates in Human Cerebral Cortex. The floor is yours.
Okay, thank you very much for the invitation. And I want to thank Silvia Marinelli for organizing this symposium that reflects an increasing interest in the human cortical tissue, not only because now we can get these very nice intracranial recordings from humans, but it's important also to have this type of preparation that we just saw, where one can go to the local circuits, to the even molecular and cellular basis and have experimental manipulation. So it's a very important issue, and I'll be talking about spatiotemporal network dynamics. So I'll talk about the cerebral cortex. The cerebral cortex has this unique property of having a recurrent connectivity, and therefore it can generate per se activity even in the absence of inputs. And I will be talking about slow waves, which is a type of activity that is spontaneously generated in the cortex, and that most of you probably know because of the slow wave sleep. They consist of these periods of activity and periods of silence, and they happen in situations of functional disconnection. When it's global, there is usually unconsciousness, like in deep sleep or under anesthesia. there is these slow waves that are generated and that dominate cortical activity. But very interestingly, it also appears in completely isolated pieces of tissue. This was demonstrated by the Steriat's group, where they saw that if one cuts out a complete gyrus or even a slab, there is still enough circuitry to generate these slow waves. Therefore, they can occur in the absence of any inputs. And in a more extreme condition, we demonstrated some years ago that cortical slices, in the absence of any chemical or electrical stimulation, they could also generate this sort of network activity that you can see there in intracellular recording at the top and multi-unit activity at the bottom, where the activity of most of the neurons in the cortex gets structured in these slow oscillations. So because they can appear in this disconnected cortex, they have been also identified from the early 70s in situations where there are lesions, like, for example, a subcortical stroke. The stroke area could be identified because in awake preparations in cat, or also it's been described in humans, there can be in the awake state this local emergence of slow waves that, as you can see in the number 6, it's useful to localize where the stroke was. So we proposed some years ago, along with Mauricio Mattia, that cortical slow waves, therefore, they can be considered the default activity pattern of the cortical network because they appear in all these different conditions that I presented. And interestingly, because there is this tendency, there is this attractor, the cortical circuit tends to generate this activity. There are more and more evidence that they can appear in the awake locally, for example, in physiological conditions such as sleep deprivation, is being described in association to lapses of attention, but also in the context of pathology. For example, slow waves locally appear in patients with epilepsy, or as in the group of Marcello Massimini, and we actually collaborate in a project on this. There are these local slow waves generated in the periphery of lesions in awake patients interfering with cognition. So therefore, there is an increasing interest in these slow waves, not only associated to sleep, but to all these other physiological and pathological conditions. So how is this? The ones I showed you so far, they are from animal preparations. So how is it in the human cortex? And one characteristic, one of the differences in the human cells is that there is this increased synaptic connectivity. They are more complex neurons. They can receive up to 40,000 inputs. So we were interested in studying in humans, but differently from the previous talk, we got human tissue, but we were interested in physiological activity, if that is possible. And this is a problem we are going to encounter when we study human tissue, which is that obviously for ethical reasons it's never going to be from healthy people. So therefore, we have to assume that this is a limitation. So in our case, we obtained tissue from refractory epilepsy from the neurosurgery department in our hospital. Most of them were anterior medial temporal resection. They were all adults. I will present you data from 22 slices, and we selected only slices from refractory epilepsy. At the beginning, we had also a sample with peritumoral tissue, but we wanted to have a more homogeneous sample, so it's only from refractory epilepsy, but from areas that are considered structurally healthy. So we collaborated with Javier de Felipe, and he did the anatomy, the structural studies to select areas that, different from the previous talk, precisely they would not have any dysplasia, and they were structurally normal, but always understanding that this is from patients that they have had an average of 22 years of epileptic discharges. So of course there may be changes at molecular levels, and probably there are. So we did a similar process to the one described in the previous talk. We obtained, I think we are a bit closer because we don't need a car. So, but still we had to carry it from the surgery room all the way to our lab. We also did acute slices, and then we recorded. We placed them in an interface chamber, and then we recorded with arrays of 16 or 32 electrodes. You can see here, well, these are patients also that they have been, as you can see at the top, they have been recorded, and all the studies have been done with intracranial pre-surgical studies and so on. And then we recorded with these arrays that cover the surface of the cortex with the idea of investigating whether there was also this spontaneous activity in the form of slow waves that we were interested in. So we saw that in a majority of the slices, we could observe this spontaneous rhythmic activity, which is very similar to the slow oscillations that we have recorded for many years in animal slices. And as you can see here, there is this rhythm which was of 0.75 hertz in average, with an upstate duration of 80 milliseconds, downstate duration of 2.62 milliseconds, and this rhythmic activity that you can see in the autocorrelogram. So there is a difference here, even when the frequency is similar to the one we observed, maybe slightly higher, but we see an up duration that it's a bit shorter than the usual one. So this is one of the differences that we encounter, and that, it may reflect. It's difficult to separate when we find a difference, whether it reflects a difference because it's human tissue or because it's a tissue from a person that has been, a patient that has been suffering from epilepsy. One aspect that we wanted to study is where in the different layers of the cortex slow waves originate. There had been some discussion in the field. Some other papers had described that they were generated in upper layers, while always very consistently in animal preparations, they are generated very consistently in layer 5. And what we found is that indeed we find exactly the same results in the human, where the layer 5, which are more excitable neurons, they lead the emergence of these upstates, and they have very similar profile to the one that we have described previously ourselves and several other groups, where the layer 5 leads the upstates, and then there is a recruitment of the upper layers and layer 6, and there is a whole columnar activation that reaches the peak almost simultaneously, and this wave is going to travel along the whole network. And this is the Ranger causality study showing that actually the activity in deep layers is leading the activity in superficial layers. So we think it's very much like in other species, in spite of the fact that these neurons in layers 2, 3 are more complex in humans. These are some data from animals, from slices, and from in vivo, from different groups, where, as I was mentioning, is very common that is consistent that it appears in the deeper layers. So once there is this activation of the column, then there is the slow wave propagation. This has been described in humans. This is an article from Massimini et al. 2005, where they describe the slow waves propagation during a slow wave sleep. These are the waves from sleep. And we have also studied in different studies how is the slow wave propagation in the cortical slices or in vivo, where we see that it follows many of the same properties of propagation that have been described in vivo. And wave propagation is a very interesting magnitude because it's very informative about the state of the network. So we studied the slow wave propagation in the human cortex. There is, you can see here on the right-hand side on A, the vertical propagation, which means the columnar activation, and then the horizontal propagation, how this wave propagates along the cortex, as you can see here in a spatial map in C. And we found a difference with the propagation in other species, which is a faster propagation in slices. So usually we have an average, for example, in ferret cortical slices, an average of 10 millimeters per second, and in humans we have an average of 65 millimeters per second. So there is this faster propagation. So this faster propagation, along with those shorter upstates, they suggest that this is a more excitable tissue. And this tissue with higher excitability, again, can be something typical from human cortex with higher connectivity, but it can be also a consequence of this pathological state, even if it's, as I said, healthy tissue. So what happens during upstates? So during upstates, we know, and this is not human, we know that there are excitatory potentials and inhibitory potentials. As you can see here with the bringing the membrane potential to different values. And also this is from a study where we characterize the occurrence of excitatory potentials and inhibitory potentials during upstates, and you can see in red and in blue that basically they are always co-occurring, and we know that excitatory and inhibitory neurons, they fire during upstates. So what we wanted to see is how if we reduce inhibition progressively, we know, of course, if we block inhibition completely, the activity transforms into epilepsy. But what we did was to do a very progressive block of inhibition to see what was the transformation of the activity. So as you can see here on the left, if we remove progressively inhibition, there is an increase in the firing rate, as you can expect if you reduce inhibition. And then each upstate is a recruitment of the local network through these recurrent connections that I showed that are typical from the cortex. So as you can see, the slope, as we block inhibition, the slope becomes progressively faster. And what this means is that when there is less inhibition, the recruitment of the local neurons becomes increasingly faster. They are recruited faster. The transition slope is faster. And we know that when it's epileptic discharge, the recruitment is very synchronous, very, very fast. It's almost vertical. So therefore, we know that even if these slices would have a bit less of inhibition because of the condition, we know that there is enough room to be transformed progressively into these different discharges. And very interestingly, when there is this faster recruitment because of the block of inhibition, this correlates with a faster propagation of the activity, as you can see here. So the less inhibition, the faster the local network is recruited, and the faster is going to travel across the network. And this is becoming progressively, is a transition towards the epileptic form discharges. So as you can see here, at the top, there is in the spatial representation, there is the propagation of the, let's say, physiological waves. And then when we block inhibition, progressively they become more massive and they become faster. And as you can see here, they become very massive discharges approaching the epileptic form discharges. And then eventually, when there is this epileptic form discharges, they can get organized into this rhythmic activity that you can see here. Maybe it reminds a bit some of the discharges that we saw also in the previous talk. And I find very interesting that there is this infra-slow rhythm, which is surprisingly regular, but still it can have intervals of like 100 seconds. So this suggests that these very slow or infra-slow dynamics can be preserved, and they can be generated and maintained in this very local piece of tissue. There is enough machinery to have this very slow, regular, highly regular dynamics that can have 100 or more seconds of interval, and they occur, as you can see, almost like a clock. So therefore, without long-range connectivity and so on, it's possible to have this infra-slow rhythm in the cortical tissue. And then if we expand these discharges, you can see them here with the local field potential, multi-unit activity, and so on. And you can see how there is this more typical epileptiform discharges after blocking inhibition. So my conclusions are human cortical slices express spontaneous slow rhythmic activity in vitro. Deep layers lead the initiation of a spontaneous slow rhythmic activity in human cortical slices. Wave propagation speed and excitability in human tissue are higher than in other species. Progressive GABAergic blockade alters the excitatory and inhibitory balance, resulting in higher firing rates, faster network recruitment and propagation, and infra-slow rhythmicity at the order of 0.01 hertz or lower. And the emergent activity at different levels of excitability in human cortical slices provide a model of the cortex to investigate both physiological, we think it's to some extent physiological, and pathological dynamics, the mechanisms, and potential neuromodulation. So we think it's important to work on this development of preparations of a human cortex where we can do these sort of studies that we heard before, and in this talk where we can analyze in depth the mechanisms and the circuits and see after so many years of running on mostly animal preparations to be able to compare and to see what are the common and different aspects and how we can get to know better the human brain. So I want to thank my group. This work was, the first author is Joanna Covello in my lab, and there has been many collaborators, and I thank also the funders, and I thank you for your attention.
Thank you very much for the very, very impressive talk. So the session is open for questions. Please, Antonino.
Thank you for this very nice talk. I have two questions on your preparation. The first one is, you said you record from structurally normal tissue. Now, presumably, the structure of the tissue you analyze it after your electrophysiology. So do you also have data from non-structurally normal physiology? This is question number one. And second, do you have some genetic data on your patients?
So so the first question, the structure was healthy. When there was some dysplasia detected, they were eliminated from the sample. We don't have data from them because we wanted in this first study to concentrate in what we thought it was the most physiological. For the structural, we trusted the person that ran the anatomy, who is Javier de Felipe, who has a very long experience, as you know, in epilepsy and also in the cerebral cortex. So they told us retrospectively which ones were completely free of any structural difference. And we gave them both. We gave them both tissue, not recorded tissue, and recorded tissue, so they had a bigger block that they could process. So this was a bit far away, I mean, in the borders of the extracted, the removed tissue. And regarding the genetics, probably they have some studies, but this is not something that we have looked into. So I don't know exactly, but I could find out what exact genetic studies these patients have.
Thank you. I think time is up.
Okay.
So maybe thank the speaker again.
Thank you, Mavi. So The next speaker is Michele Giuliano. He is a professor at University of Modena and Reggio Emilia, and he will talk about broadband cortical neuron ensemble.
Thank
Please.
you. It's a bit intimidating to giving a talk in this very nice venue. And the main character of this talk, it will be clear what I mean with broadband, is excitability. That is, in my opinion, far from being completely understood. It has been the topic of interest and research in my group for many years, and it's more than an all or none phenomenon. It has been shown also by experiments in our lab that it's not only a very trivial firing of action potential like a pacemaker, textbook physiology, but it includes some feature of complexity when you start probing the system to reveal its time scales. In this case, by some optogenetic stimulation that is not relevant for very long period, up to several hours, the excitability of single cells is probed and the latency is measured. And what you can see is that over time scales of several tens of seconds, hardly compatible with spike frequency adaptation, the latency of neuronal responses that otherwise should be stable shows drift, shows intermittency and chaotic behavior and even failures. So its richness of temporal scales is important in our opinion to be studied, and it is normally neglected by conventional electrophysiological protocols. There are a couple of examples that I would like to give by which the complexity of excitability is revealed perhaps functionally in vivo. In this case is a very old data set from the end of the 90s from Christoph Koch and collaborators, in which single unit recording from the anesthetized temporal cortex of a monkey has been recorded while the animal was subjected to natural scenes videos. And what you actually see here is the raster plot, the same unit recorded over and over, repeating over and over, so assuming ergodicity of the process of initiation of action potential, so the encoding phenomenon in the cortex. And in the lower panel, you actually see the PSTH, the peristimulus time histogram, that surprisingly, when you analyze it in terms of spectral content, reveals energy content at very high Fourier frequencies, above 100 or 150 cycles per second, while individual neurons, this guy here, was only firing, say, maybe 10, 20 spikes per second. Another example is reported very common in the cerebellum, where Purkinje neurons are firing maybe a little bit faster, but collectively they give rise to very fast rhythms of the order of 200, 300 cycles per second, as revealed by this spectrogram here. And so it seems that you do have some properties associated with excitability that are determining this very fast information encoding, so representing temporal information downstream that is not compatible with the, say, passive properties of the membrane, as well as with the low firing rate of individual neurons, as well as ultra-fast oscillations where individual neurons are participating in this rhythm sparsely. So they are not firing at every cycle of these, in this case, 200, 300 cycles per second oscillations. So when this complex feature of excitability has to be studied, the classical paradigm of understanding input-output relationship, here depicted, it should have been an animation, by the so-called FI curve, output firing rate versus inputs, cannot be used anymore because the input is now changing rapidly, and this is a static description, cannot any longer tell something about how the system is performing when, again, fast temporal scales and non-stationary phenomenon are in play. And these deeper understanding that I'm somehow advocating, that I'm interested on the time scales of neuronal excitability, are very significant, not only in the domain of, say, modeling. I'm mentioning very briefly that it might have dramatic consequence for the expressive power of our architecture in machine learning and artificial intelligence,
but also for basic neurophysiology, to study biological networks, to interpret these fast encoding properties that I showed you before in the anesthetized primates, for interpreting these high-frequency oscillations with sparse spiking activity, and also, but I will not have time to tell you about, also to inquire and interpret the correlated, the omnipresent correlated activity that is recorded at any spatial scales and temporal scales in the cortex. So what we did, inspired by computational and theoretical approaches, we were the first few years ago to propose a new kind of characterization of these non-stationary properties of cellular excitability. So looking at neurons as somehow a black box, being myself by training an engineer, so I see black boxes and Fourier analysis everywhere, we've been basically developing a protocol where not stationary, but so non-stationary inputs have been delivered to neurons, and the output firing frequency have been characterized in terms of the so-called instantaneous firing frequency. It's very similar to what I showed you before in the PSTH. In that case, it was a sensory stimulation. In this case, is an intracellular stimulation by current clamp or by conductance clamp that is represented here. I'm just neglecting the fact that we have been injecting some sort of fluctuating synaptic recreated inputs. This is for another time. And we could, by this kind of protocol, control in a synthetic way the contribution from the excitatory presynaptic population as well as inhibitory presynaptic population in a synthetic way. So mimicking the synaptic inputs, of course, at the soma. That is a weakness of this approach, neglecting clearly the input at dendrites. And when you do this, the characterization that you end up with is very similar to what any physical system has, that is of a filter. I don't know how many of you recognize a graphical equalizer. What neurons are devices that are not particularly interested, like in this case, from the rodent, layer 5 pyramidal cells from the somatosensory cortex, not particularly interested at slow input oscillations. Neurons are firing collectively, but they do not show in their output spike trains any modulation that is reflecting the modulation that we're injecting in the input. If this modulation is too slow, 10 cycles per second, or it's too fast, 1,000 cycles per second. But if we go fast, unexpectedly fast, given the firing rate of these, mean firing rate of these neurons that was in this case around 20 to 30 spikes per second, one order of magnitude higher oscillation frequencies like 100 cycles per second or 300, up to 300 cycles per second, neurons are showing this modulation. They are able to relay, to encode this temporal information in their spike train. So ultimately, there are many interesting features. Here I'm only showing the so-called magnitude of the response. I'm not telling you about the phase. Here you actually clearly see that there is a phase lag. There are very interesting consequences of these phase dependency on the frequency. The bottom line is that the bandwidth, what I call the bandwidth, so when this response starts to cut, the cut goes like 1 over f, but this is not crucial. This becomes a very important predictor of network activity. And indeed, this allows us to explain this fast encoding. In this particular case is a PSTH of neurons recorded in our lab, in which we subjected all the neurons to a very sudden step change. And here, by comparison, I'm indicating the typical mean interspike interval. So in the typical sort of old-fashioned view, you need to wait for another action potential of the same neuron to see whether the inputs change. In this case, the population response changes much, much faster, only after 5 milliseconds. And in this case, we could, just by taking this filter picture and filter box, black box, and giving an arbitrarily fast, it was a frozen realization of an Ornstein-Uhlenbeck process, we could predict in red the PSTH in blue of these cells on a time scales of a few hundreds or few tenths of milliseconds. So even the very fast transients were predicted, suggesting that this sort of characterization that is inspired and avoidably related to linear system theory, linear dynamical system theory, still does work if the neuron is not at all firing small, so the modulations are not small and the neuron is not firing at small firing frequencies, but in this case it goes between 0 and 90 spikes per second. Similarly, we can explain and interpret this fast rhythm. This is an example from unpublished data from rat Purkinje cells in the rat, and we could see that the magnitude shows a dramatic even resonance, not only a large bandwidth, resonance up to 100 or 200 cycles per second that is reminiscent or directly predictive of what the spectrogram was indicating. And we could, and I don't have time to give you more details, we could show that it's not per se some active properties at the dendrites, because if you inject the same sort of stimuli in the dendrites you have at the dendrites, you don't have this sort of resonance, but at the point where action potentials are initiated, it's there that this amplification and resonance is taking place. The most crucial and interesting property that we could find, and again, this is a direct translation from what mathematical models of a variety of integrate and fire neurons are telling, where you could do the math and you can predict why you have this short or wide bandwidth of spike frequency, initiation, is the slope, the initial slope, the so-called velocity at the onset of the action potential. And in our data set, in this case from rodents, we could actually see that, we could test that it was indeed, there is a correlation between this rapidity at the onset in the phase space, membrane potential, versus the velocity of change in the membrane potential. At the onset, the slope of these tangent lines correlates with the bandwidth, with the cutoff, where these dynamical Fourier domain characterization starts to cut the signal. And you actually see that depending on the cell, the steeper the action potentials, the broader the bandwidth, up to 800 cycles per second. So this was very intriguing, because in the context of, and I'm ignoring the reasons why this has been considered, so the interpretation of this, in the context of exchange and access to human tissue before, in collaboration with the Amsterdam University, the group of Hyde Mansvelder, and today in Modena, the interaction with and the access to human tissue from, in the case of the data I'm reporting, from non-epileptic, not epileptic patients, but patients with cortex, healthy cortex removed in surgical operations, interventions to remove tumor tissue that was deeper. We could assess that, we could test that the action potential shape was very different comparing rodents and humans. In humans, it was middle temporal cortex and layer 2-3 neurons, and in rodents was the same, was temporal cortex of mice in layer 2-3. And what was surprising is that if you compare isolated action potential, evoked or spontaneous, they don't particularly differ in terms of action potential threshold. Define whatever you wanted to define in terms of, say, second or third derivative, it doesn't really matter, as well as in terms of the onset rapidity. But when you study trains of action potentials at different frequencies, is there where presumably different subunits of voltage-gated action potential reveal a difference. And in case of humans, the onset still is revealing to be much higher than in rodents. So single action potential are indistinguishable. Human cell action potential, as soon as the neuron is engaged in, say, 1 to 5 hertz or 5 to 10 hertz firing, is much larger than in rodents. So we tested the hypothesis that having a different action potential initiation dynamics, then the bandwidth would have been different, perhaps accounting for physiological superior performance in terms of information encoding. And indeed, what we found is that while rodent neurons from temporal cortex, layer 2-3, are basically unable to transfer modulation that is as fast as 1,000 cycles per second, they are filters, they are cutting off at some point. For human cells, they are up and kicking up to 1 kilohertz. We could not go faster because of technical problems. At some point, they do cut. In this case, it's the continuous lines that you see is a fit from a theoretical formalized way of the dynamical transfer in the Fourier domain. It's a complex rational functions, but the bandwidth of human cells is surprisingly larger, up to 1 kilohertz. So one order of magnitude compared to rodents. So not only unexpectedly larger compared to what the theory would account for, given the passive membrane properties. I'm thinking of the membrane time constant as well as of the typical mean firing rate, which is few spikes per second, but also even 10 times faster than in rodents. And we search for possible correlates of these functions. I don't enter into the details. Mathematical modeling of conductance space neurons from reconstructed morphology is one crucial tool, and comparing very large, this is the one key point of human neurons, cellular anatomy that is clearly striking comparing to rodent tissue. It seems that the dendrites, in this case for pyramidal cells, seems that the very large dendritic tree leads to electrotonical properties that are relevant, still relevant for the point of initiation, the axon initial segment where the action potential are generated, to account for the broad, even broader bandwidth of the response. And talking about the axon initial segment in modeling, in silico, we found that surprisingly the location of the axon initial segment is also responsible for broadening of the bandwidth. Not quantitatively accounting for this tenfold, going from rodents to humans, but combined with the large dendritic tree, possibly contributing to the explanation why human cells have these broader encoding possibilities. And of course, the next step for us is to measure quantitatively what is the distance in human cortical neurons of the axon initial segment. So in summary, I somehow emphasized and suggested that this dynamical characterization of excitability is crucial, and it accounts for fast responses and sparsely synchronized rhythms, like they are known in the literature. It is bandpass with very high cutoff frequency, surprisingly high, despite the low firing frequencies, and it can explain these ultra-fast encoding properties, ultra-fast reaction times that I demonstrated in a simple experiment, and the bandwidth cutoff is correlated and affected as predicted by computational models by the upstroke velocity of action potentials. And any morphological, subcellular, and molecular heterogeneity, like what has been described already in the literature for different species in primates, and particularly humans, of sodium voltage-gated ion channels, determining or contributing to action potential shape, is expected to give, as we verified, give an impact on this bandwidth characterization. And so human cells do fire sharper action potentials and thus have a broader bandwidth. I conclude thanking current collaborators and colleagues, particularly the people from the Baggiovara Hospital in Modena that are enlightened and allow us to access a human tissue, as well as previous collaborators that pioneered the field of single-cell human cortical cell dynamical characterization. Thanks for your attention.
Thank you, Michele, for your nice talk. There is time for one question. Please, Antonino.
I'm very intrigued by this ultra-fast collective mode of firing. Now, I have a few questions on this. So, if I understand correctly, one explanation that you give to this, despite it being a collective behavior, is a property of individual neurons, that is a sharp action potential. Now, I understand that this may be one explanation, but as a collective phenomenon, don't you think that connectivity properties underlie this? And if so, how could you measure them? And second question, how does this property, I'm not clear, depends on statistic properties of the stimulus that you're giving?
So I answer the second part. It's a property of the cell. It's not the property of the protocol, of the statistics of the protocol. So we need this noise, but the color of the noise, for instance, is not having an impact. So how fast is this noise fluctuation does not have an impact, as opposed to what you see in integrated fire neurons, where the slower the noise, the faster the response. And going to the first part, all of these characterization of ensembles where we were only measuring one neurons over and over and over, repeating in time, so swapping spatial ensembles with temporal averaging, basically, is assuming disconnected cells, and trivializing, it is as if individual neurons are not following the rhythms, but being many, there will be always one or few neurons relatively close to threshold, and the steeper their action potential, the faster there will be their reaction to go to threshold and lead to synaptic release to convey downstream this information. This is the way to trivialize it. Connectivity would further change this picture in a way that is difficult to measure, if not with microelectrode arrays that we are currently investigating. Giving rhythms to all the neurons might be exploiting optogenetics, in which just wide field optogenetics having transduced cells, and we would love to do it with human neurons by organotypic slice culture, would be key to give some sort of common rhythm and possibly probing the network like I did for individual neurons, frequency by frequency, low frequency, faster frequency, etc., etc. So this would be my answer to your questions.
Michele, could be due to also this different, this sharper action potential due to also to a different ionic channel composition, like there are some channels that are expressed in human neuron and not in rodents.
Yeah, absolutely. The easiest explanation is that it's a subunit of voltage-gated sodium channels. Definitely the contribution from slow cumulative inactivating sodium channels is playing a role. And it's, so computational models, conductance-based models are an ideal settings for falsifying or verifying hypotheses before going with pharmacology in a slice. But then definitely that would be the ultimate goal.
Okay, thank you. Thank you. And the last speaker is our younger Laura Monni, and she from University of Modena in Reggio Emilia, and she will talk about how to prepare acute human hippocampal slice for electrophysiological recording. Thank you.
Thank you. Thank you, Silvia. Good morning, everyone. After these inspiring talks on human neocortex, I would like to shift our attention to the hippocampus. So the hippocampus is, of course, a central region for learning and memory, but also for epilepsy. And I'll show you how we prepare acute human hippocampal slices from tissue obtained from patients, but also how this tissue can be used to model other human pathologies. So the hippocampus is central to uniquely human functions, so lifelong memory storage, so our ability to remember lifelong experiences, but also imagination, mental time travel, conscious experience, and so on. And these uniquely human functions cannot be, of course, fully captured in animals. Another thing is that human hippocampus, or the hippocampus in general, is a key region for epilepsy. And so all this make this region both fascinating and clinically important to study directly using human tissue. Indeed, the ex vivo human brain tissue, so both cortex and hippocampus, can help us bridging the consequent translational gap that exists between the results that we obtain with animal models and the actual clinical application of these results for the treatment of human pathologies. To illustrate these human specific functions, I would like to talk about a couple of studies that help us understanding why the animal models cannot really capture many human brain mechanisms. First of all, the spike timing dependent plasticity has been demonstrated to be characterized by a much wider temporal window than a rodent hippocampus. Basically, this means that the human hippocampal synapses can listen for more time and can be and are more flexible to encode and store the temporal relationships, the information about the temporal relationships of the neuronal spikes. And this flexibility is probably one of the possible causes contributing to the complexity of the learning, memory, and cognition mechanisms in the human brain. But also, more recently, the human hippocampal CA3 microcircuit has been shown to be characterized by a much sparser connectivity compared to the rodent hippocampus and also to the human cortex. But the synaptic transmission is also highly reliable and temporally precise. Moreover, the dendrites are more than twice longer than the rodent dendrites in the hippocampus. And all these features together may suggest that the hippocampus is not simply a scale-up version of the rodent hippocampus, but it exhibits species-specific microcircuit specializations that underlie probably the complex computations that our brain is able to do. So how do we obtain this precious tissue? Most of it comes from, of course, surgical resections of patients diagnosed with drug-resistant epilepsy. And in particular, this could be temporal lobe epilepsy, but also cortical dysplasia, as we have seen in the previous talks. But also patients with epileptic seizures caused by a lesion or a brain tumor. But occasionally, we can get the human hippocampus also from patients showing not epileptic seizures, but with other conditions, like low or high-grade gliomas and brain metastases. So this means that we can get tissue, both healthy-like tissue and possibly disease-damaged one, so we can have a window into both near physiological and pathological tissue. So once we get the piece, of course, careful preparation is the key to preserve its viability and high quality. And in particular, the preparation starts, of course, with the transport. It could be also 3 hours or even less. And this transport is done in a sterile and modified artificial cerebrospinal fluid that is basically aimed at protecting the tissue from different types of damage and hyperexcitability in general. The block is then sliced, of course, with a vibratome, and here I put an example of a schematic of a slice, hippocampus slice, ideal, containing all the principal subregions of the hippocampus, so starting from the dentate gyrus to all the cornus ammonis and also the subbicum sometimes. The slices then undergo a 2 or 3 steps recovery process, depending on the electrophysiological recording that you want to do. And this recovery process simply is aimed at re-establishing a sort of near physiological condition to store the tissue. So with these slices, we can, first of all, record the network activity via, for example, local field potential recordings. And here, I put 2 examples of evoked activity or network activity that can be evoked in the CA1 region, in this case. So on the left, you see interictal-like spikes and bursting activity that can be evoked by perfusing the slices with a combination of high potassium and foraminopyridine. But also we can evoke seizure-like events, which are network events lasting more than 10 seconds with a particular pattern. And this can be evoked with low magnesium or 0 magnesium and combined with a GABA receptor blocker that could be bicuculin or gabazine. But one of the most important aspects of this evoked activity is that it's sensitive to the effect of anti-seizure medications. Here I put 2 examples. The first, the upper traces show you the effect of a classical anti-seizure medication, which is lacosamide, while the lower traces show you the effect of DMEA, or dimethylethanolamine, on the evoked pathological activity, in this case induced by high potassium and 4-AP. The plots show you that both these compounds significantly reduce the pathological activity, but in this case, DMEA is the interesting aspect of this slide. DMEA indeed is a novel compound that is being tested for its anti-seizure potential. And with this, I want just to tell you that this tissue can be used for both drug screening or for classical anti-seizure medications, but also to test new compounds, new therapeutic strategies, both before and alongside the clinical trials. So this will give us a translational bridge, of course. Then, zooming in into single cells, of course, the activity of single cells in acute human hippocampal lysis is totally possible. But here, I want to show you the activity recorded after 24 hours from the preparation in a sort of short-term culture condition. So the trace on the left are example traces of firing of a CA3 pyramidal neuron after 24 hours, taken from the anterior hippocampus. And with the two plots, I want just to simply show you that the frequency of the CA3 pyramidal neurons after 24 hours from human hippocampus is very similar to the frequency of the same class of neurons, but recorded from the ventral hippocampus of rodents. But one of the most exciting, of course, aspects of or applications of the human brain slice cultures, even if short, like in this case, is modeling human pathology. And today I brought you the example of limbic encephalitis. Limbic encephalitis is a disease that is caused by the production in the central nervous system of patients of autoantibodies that can target different proteins or receptors. In this case, they target LGA1 in the hippocampus. And these autoantibodies cause epileptic seizures, but also psychotic symptoms and memory and cognitive impairments. In this case, the autoantibodies are targeting LGA1, as I said, and LGA1 is a protein, transsynaptic protein, that forms very important complexes at the excitatory synapses in particular, but also in inhibitory synapses and in the axonal ischial segment. And these complexes are very important for the regulation of, in particular, potassium channels Kv1.1 at the presynaptic terminal, and also AMPA receptors at the postsynaptic terminal. And so these autoantibodies disrupt these complexes and cause the internalization of the potassium channels, Kv1.1, and sometimes also of the AMPA receptors, leading to, of course, hyperexcitability that is one of the main causes of epileptic seizures. So what we thought was to incubate the human hippocampal slices with patient-derived LGA1 autoantibodies to check, mainly to check if this tissue can be useful as a species-specific model for human pathologies other than drug-resistant epilepsy. And in this case, we also use a control, DTXK, which is a dendrotoxin selective for the blocking of Kv1.1 channels, simply as a control for the effect of the autoantibodies. And the main result that we got was that the LGA1 autoantibodies indeed significantly increase the excitability of the human CA3 pyramidal neurons after 24 hours, mimicking one of the main disease features, so the hyperexcitability indeed. And also DTXK exerted a similar effect. It increased the excitability of these neurons, but then it had some different mechanisms of some different effects with autoantibodies, but now I don't have the time to talk about this. With this, I just want to say that the human hippocampal slice cultures can be a very helpful platform, very useful platform and model to model human pathologies, and so not only drug testing, but also to have a sort of homologous system to study disease-relevant substances. Then, to take it a step further, we also are currently establishing the use of human hippocampal slices and cortical slices with advanced electrophysiological systems like high-density multi-electrode array, or HDMEA, These are biosensing chips based on complementary metal oxide semiconductor, or CMOS technology, which basically combines a dense array of microelectrodes with an advanced signal processing, allowing to have a chip with thousands of microelectrodes packed in a very tiny area. And this allows us to record the extracellular activity from hundreds of or thousands of neurons simultaneously with an incredible spatial and temporal resolution. And so these systems open the window, open the ways to the possibility of investigating human-specific neuronal dynamics at a very high spatial temporal resolution. Here I put an example of an event recorded in one of these more than 4,000 electrodes, just to show you that from the raw trace, from one electrode, we can obtain, let's say, two different views of the network activity. So by low-pass filtering, we can get the local field potential, so we can analyze the dynamics of the neural tissue surrounding each electrode. But also, band passing the raw signal, we can get the so-called multi or single unit activity, which reflects basically large spikes that represent the activity of more or less single neurons very close to the tip of the electrode. And then with different sorting algorithms, we can assign all these spikes to different neuronal classes or clusters. But if you are interested in our results with this advanced system, I invite you to see Manuela's and Alice's posters. They will be very happy to show you our results on both human hippocampal slices and human cortex slices. So, to summarize, the human hippocampal slices offer a unique platform to study human neurophysiology. An optimized preparation allows to have a viable and high-quality tissue that can be used for advanced electrophysiological recordings, both at single-cell and network level. And this tissue enables also translational research because we can, as I said, test different decision medications effect directly on the human networks, but also other disease-relevant substances like patient-derived autoantibodies. And then also the human hippocampal slices are compatible with advanced technologies like HDMEAs, and this opens new ways into the study of human-specific network dynamics. So I want to thank you all, and I thank my current lab in Modena, but also my former lab at the Charité in Berlin, Germany. I thank also all the neurosurgery teams, both in Germany and in Modena. And of course, I thank also all the patients who kindly donated a sample of their brains. Thank you.
Thank you, Laura, for your very useful talk. And now there is time for question. Okay, I have a question. You have shown some experiment in which you have applied bicuculin and you have inhibited your interictal discharge. Which type of human tissue? Was a tissue from children, from adult?
It was from adult drug-resistant temporal lobe epilepsy. We got, in Berlin, we got mainly that tissue, TLE, basically.
Okay. Yes. No, because I was translating your result with
our
results,
yes,
so
yes,
I was worried.
Yeah, yeah.
Okay.
No, it's not. It's not cortical dysplasia. It's another type of diagnosis. Yes.
Okay, thank you. And another question is regarding your 24-hour recording after the slicing. So how do you keep your slice in this 24-hour recording once you slice that?
I just put them in culture, in a culture medium. So I'm applying actually a protocol that was developed in the Ellen Brain Institute in Seattle. And this protocol, so it's very important, I noticed that it's very important, this recovery process to really keep alive and the tissue, which is basically the storage of slices in first the modified CSF, so they could be NMDG, but also choline or sucrose. And then you slowly put back the slices in in a sort of near physiological condition. So first a solution in which the sodium ions are slowly reintroduced. That is this low sodium ACSF. And then I put them in a medium normally, in a 6-well plate, in the incubator, as a normal organotypic slice culture.
Okay.
Yeah. And they are preserved, let's say, until 6-7 days, I would say. Then after they start to modify their firing activity, there is no network activity anymore, so I would say 6 days maximum.
Grazie. So the symposium, thank you to have attended this symposium, and we thank all the speakers to join this event.
Ho fatto il conto un giorno che abbiamo iniziato alle 25, quindi è stato... No, no. Sì, sì. Perché? No, no. Sì. Grazie. Grazie.
You can probably just be there, so you're gonna be the first one, right? So we can
Why are we so separated?
Don't be so far from me.
Io non voglio stare qua giù.
Time.
Questa va già? Sì, questa va già.
Okay.
Ok, va bene, quindi fanno loro le domande.
Dobbiamo aspettare qualche minuto?
Che ore sono?
Non mi dici due minuti.
Secondo me io aspetterei forse un secondo.
Sì?
La gente sta ancora...
Perché non ci proiettano? Ci proiettano...
Tutti quelli che sono qui di sicuro non stanno seguendo.
C'è scritto hairs, no chairs, questa cosa è interessante.
The Horoid. Mi piace. Sta andando tutto.
Questa slide. Me la facciamo correggere? So it works very well, I think, pointing there, but in way too much.
Okay, yeah. It's too sensitive.
Otherwise, I guess that you can also just use, I mean, if you're not
Yeah.
moving, you can, I think that it's less sensitive as well.
Yeah, but we need to point it.
Do we?
Abbiamo qualche errore nella slide. C'è scritto, qua ci manca la C, coral
anche qui.
plexus, e anche lì, chairs, c'è scritto hair.
Ah, ok.
Ne abbiamo tanto di capelli, però magari non è quello che volevamo dirgli. Lo rubate a mio marito però, se la consola.
Questa cosa a me non la devi.
E per il ponte usiamo questo, ma lo possiamo puntare sia qua
Sì,
che là.
sì.
So we can point here.
Okay.
Ah, ok.
Okay. Okay. And next slide is this. No. Okay. And do I click on here? For
No, no.
You click. Okay. Is there like presentation notes?
No.
Okay.
There will be no presentation view.
Okay.
They told us it's on, non c'è la presentazione con le note a fianco, giusto? C'è solo il...
No. Okay. That's it.
You don't remember what to say? Nobody knows what
You
we're
move to the next
supposed
slide.
to say.
Yeah.
I move to the next. That's messed up.
Allora, c'è una C,
Code
colored, dopo la A, la prima parola comincia con una C maiuscola, e poi Simona Lodato c'è il resto. Oppure lo può anche togliere, possono mettere c'è, come se lo già plurale, può proprio togliere questa cosa. Siamo due, c'è quindi plurale.
So the first one is for pointing, this one is for moving slide.
Yeah,
He said,
these two.
okay,
Yeah.
perfect. Lovely. I don't know if I'm tall enough for Yes, I am. I am. You definitely are. If you want, I guess that you can also just like move them upwards.
Okay.
I'm really
Quindi
wondering
magari
why
questa
we
la
have
sappiamo
this big
proprio,
room.
lo dico solo io. Ok, allora noi
Perché un po' diamo...
dobbiamo aspettare ancora qualche minuto.
At least until the presentation is over. Sì. So I don't know if they told you, I think somebody told you that there is the bar with the time that is moving while you're talking, and then there is a dong, like very strong whenever
Yeah.
you're out of limit, but I don't think that you're going to arrive to that point, but don't get
Hopefully.
scared.
No. Okay.
It's just very loud.
Yeah.
Ti sei preparata.
Ma secondo te io non avevo preso il ventaglietto, non me lo avevo messo.
Allora sarà contenta.
Sì, sono un sacco di gente. No, non di tassare
Io
me
non
lo
ce l'ho
presto.
neanche. Oddio, forse io ce l'ho dietro, devo vado a prendere il mio. Non l'ho ancora aperto, quindi non so neanche se ce l'ho.
Sono Massara. Io so cosa c'è di così. Io devo
King's College.
Ok, perfetto.
Going to show the slide, so whenever you want, it's going.
Do I have to press it or do I have to click on it? Okay. Okay.
So they don't show that slide, it was a bit messy.
Okay.
You see?
Hello, everyone. Okay. Cool.
So good morning, everyone, and welcome to this symposium. I'm Simona Lodato, and with me there is Valentina Scarpetta. We are the chair of this symposium, and we would like to sincerely thank all the organizers
for selecting our topic. We're truly excited for this opportunity to discuss today about the core reflexes in development, in disease, in repair, and also presenting new data about different models. It's my great pleasure to introduce Elia Tang from from London, from the lab of Laura Pellegrini, who has actually agreed to take her to present since Laura could not be joining us today. So he's a postdoc in a lab, and we're very excited about hearing about his work. Thank you so much.
Thank you for the kind introduction. Hello, everyone. I'm Elijah. I'm a postdoc in the Pellegrini lab. It's a great pleasure to be speaking on behalf of Laura and the Pellegrini lab. Just a short introduction about the coral plexus in case anyone needs it. The coral plexus is a specialized epithelial tissue, and it functions both as a secretory organ where it produces cerebrospinal fluid which fills up the brain ventricles, and it functions also as a physical barrier which tightly regulates the exchange between the peripheral bloodstream and the ventricle system. Apart from the location and the function of coral plexus, the coral plexus itself is quite a complex tissue comprising of various different cell types. Other than the main epithelial cells, we have a stroma layer which supports the main epithelial barrier. This stroma layer consists of fibroblasts, parasites, and endothelial cells. And in addition of that, there is also various different immune cells, both resident and circulating from peripheral regulations. Cool. From mouse study, we know that the coral plexus epithelium originates from the cortical hem or rhombic lip. Specifically in the lateral region, cortical hem is exposed to BMP4 and Wnt signaling during E11.5 to E14.5, and this drives the coral plexus epithelium differentiation. The differentiation begins with a pseudostratified layer, which subsequently morph into a cuboidal monolayer with a apical-basal polarity established. Although this differentiation trajectory is well studied and mapped out in mouse experiments, it is quite hard to map it out in human fetal samples due to the limited access we have to human samples, which is why the coral plexus organoid protocol developed by Laura and the Lancaster lab in 2020 has been valuable to our studies. For the coral plexus organoid model that we are using, developed by Laura, it involved a pulse BMP4 CHR and CHR treatment at day 10 of the standard cortical differentiation. This is to model the lateral coral plexus development trajectory. With this model, we can then study the tissue development across embryonic to fetal stages. We can also understand how the blood CSF barrier works, and we can also study the changes in CSF secretome across developmental time points. Cool. Here you can see example images of our coral plexus organoids, both in culture and after staining. As you can see, an important feature of our organoid is that it's built inside out, where the basal layer is medial facing. So CSF that is secreted through the apical surface is going to fill out the CSF fuchs gap, and that allows us to extract CSF whenever we want, if we do want to test the CSF. In our lab, we use this system to test two different directions. Firstly, we would like to map the human coral plexus development, looking at the multiple different cell types and how we can stage map it to adult tissues. In addition, we are also looking at how different environmental stressor and insult can affect and influence the development of coral plexus and the barrier function. For the rest of today's talk, I will only be focusing on these two topics. First, how we are mapping out the human coral plexus development, and if time permits, I will go through how we are damaging and studying the repair response of the coral plexus epithelium. When we look at the adult mapping of the choroplexus organoid model, we first look at the proliferative status of the organoid epithelium. From our epithelium, we can see that there's a decrease in Ki67, which is a proliferative marker across the different time points. And this is consistent with our human fetal tissues. In addition, we also saw a reduction in proportion of G2M and S phase cells in the cell cycle. This results tells us that our choroplexus epithelium reached post-mitotic stage in the later stages of the model. To understand how we can study the temporal development of the human choroplexus using our model, we collected samples from a broad temporal time point ranging from day 14 of differentiation to day 130. Samples collected are analyzed through single-cell RNA-seq, which allow us to resolve the heterogeneity of the tissue, as well as allow us to track the changes in individual cell state and transcriptomic progression. So as you can see, it's a broad spectrum of different cell types that we have in our model. But to summarize, we have obviously the epithelial cells, and from that cluster, we can subcluster it to the multiciliated epithelial cells. We also have some stroma cells present in our model, as well as cortical, progenitals, and neuron cell types. From this diversity emerging out from our model, we then look at whether we can recapitulate the developmental trajectory. By overlaying the sample age that are over the plot, we can see that there's a clear pattern of the younger organoids being covering the progenital clusters and the mature organoids covering the mature cell types. We then do a pseudo-time analysis, specify the choroplexus lineages, and we saw that there's a straight progression from cortical hand progenitals to mature epithelial cell types. From there, we then look at some of the mature features in terms of the transcriptomic changes, transcriptomic program involved. From our single-cell RNA-seq data, we can look at the top and reach genes. And from there, we look at 5 different genetic modules, namely the proliferation, neurogenesis, lineage specification, celiogenesis, and transmembrane transporter. We overlay it to our pseudo time point, and as you can see, there's a clear progression of the time across the different modules. From there, we then look at the specific epithelium maturation features, both celiogenesis and transmembrane transport in terms of the cellular behavior changes. Firstly, looking at multiciliogenesis, there are 2 phenomena that has been reported. Firstly, the shortening of the cilia length, and then the clustering to form multiciliation. This too has been reported to be present during multiciliogenesis of choroplexus maturation. And from our confocal images of our choroplexus organoid model, as well as human fetal tissue, we can already visually see that 2 phenomena happening. But when we look at our single-cell RNA-seq data, we wanted to find the specific time points where this is happening, and it is happening during day 40 to day 70 of our differentiation protocol. From there, we collected samples to analyze and quantify whether we do really see those 2 phenomena. And we can conclude that we did see shortening of the cilia length as well as the clustering of the cilia to form multiciliator as our organoid epithelium matures. We then look at the upregulation of transmembrane transporter. From our data sets again, we can see a whole range of different transmembrane transporter genes being upregulated at the point of day 70, and these transmembrane transporters are important in the maturation of choroplexus as it functions as a secretory organ. More specifically, SLC5A5, which is a sodium iodide syn porter. From our confocal images, we can see that it is not showing up in earlier organoids, and it started showing in later organoids. Also in this set of confocal images is EMPP2, which is a secretor enzyme involved in lipid signaling. This enzyme is also upregulated at later time points. Moving forward, we also tested if we can actually see any functions upon maturation of the adult human. To test that, we extracted CSF from our organoids. From our CSF collection, we can then run through mass spec to see if we can compare the secretome differences. We recently did a comparison, and as you can see, one of our analysis, we tested the CSF collected for day 140 organoids to a human CSF sample of a 55-year-old donor, and there is a striking 76% overlap of secretome in our samples. We can also test whether, we can also look at whether there's a change in secretome across the differentiation and maturation. And from here, younger CSF collected has a different secretome profile, as you can see from the heat map over there. And again, mentioning EMPP2, it's clearly upregulated and secreted in later stages, and is also present in human adult CSF samples. From here, it's a natural transition to the repair, because when we collect CSF, we always have to puncture and sort of injure the coral plexus. And after a few days, we often see that the cyst will come back and the coral plexus would repair. So we begin to ask how it is happening and what kind of mechanism it is going through. So in terms of in vivo injury to the coral plexus, it can happen by various ways, namely pathogenic infection, procedures such as tumor removal, cauterization, even adult aging would affect the barrier. Although not much is known about the repair response, we can postulate from references of other epithelial tissues. Firstly, the skin and intestinal epithelium uses reactivation of proliferation or a stem cell population to regenerate the epithelial tissue. And for lung airway epithelium, they uses dedifferentiation mechanism where the mature cell type will redifferentiate into naive and then repair the tissue that way before going back to mature state. And lastly, we have transdifferentiation, also commonly known as epithelial mesenchymal transition, whereby epithelial cells undergo transdifferentiation into a different cell type totally.
Pronto?
To answer that question, we went to the extreme by physically disrupt the whole cyst by cutting out into pieces. And we followed those pieces across different time points and collected them for bulk RNA-seq and IF histology. Although our model doesn't have any immune cells, we first noted that in the absence of immune cells, the epithelium
Io adesso non tocco più niente, lascio il computer così.
In various chemokines and cytokines after injury. Specifically, CCL2 is also detected
11:40.
and CCL2 is a chemokine that has been reported in previous coral plexus study to recruit residential as
Ok, benone,
macrophages
grazie
that
mille,
help
perfetto.
in repair and wound healing. We then look at some of the possible way of repair. And firstly, we didn't see any reactivation in cell proliferation. There's no change in the Ki67 marked cells along the epithelium. Looking at the selected cell cycle and cell proliferation genes, we also saw no changes in those, re-illustrating that it is not possible because coral plexus is terminally differentiated. We then look into our bulk RNA-seq data for other signaling associated to any repair response. We first saw that there's a decrease in normal epithelial function like cilia aerobic and anaerobic respiration, but also there's an increase in cell migration, wound healing, cytokines, and ECM remodeling associated genes. Interestingly, there's also an upregulation of stroma markers during injury response. An example is transgelin, which is a myofibroblast marker. We saw that it increases significantly 24 hours post-injury, and that increase is sustained for weeks. On the other hand, there's no change in mature epithelia or early cortical hand markers, indicating that dedifferentiation as a repair mechanism might not be possible for choroplexus to undergo for repair after physical injury. For EMT, it is induced by a set of transcription factor, namely Z family, SNIL, SLUG, and TWIST1. And to do that, those transcription factors have to be active before the epithelial cells can start transdifferentiating into mesenchymal. From our bulk RNA-seq data, we saw that there's an increase in certain of those transcription factors, namely SNIL2, TWIST1, and ZAP2, thus indicating that EMT might be a possible route of repair for choroplexus after injury. So in summary, we have talked about how we are mapping the choroplexus development in human using our human choroplexus organoid model, namely by focusing on the maturation features like multi-celiogenesis as well as upregulation of transmembrane transporter. I've also touched on a little bit on the injury response to physical injury. We saw no sign of reactivating cell proliferation. There's a whole bunch of matrix remodeling and immune response genes being upregulated. And then we also saw induce of EMT signature. Works are on the way to look at what happens after EMT, whether that EMT is permanent or partial. So I think hopefully by next year, Laura can come here and talk to you about more about this work. Before I end, I would like to acknowledge the following people. So the members of Pellegrini Lab, most of today's work are done by Lizzie Aspley, which is another postdoc in our lab. Myself and Marianne took part in certain parts of the paper. And also for our collaborators, Ivan and Madeline contributed a lot for the first part when we are mapping the organoid model, and for Katie Long for providing us with the human fetal tissue samples. Thank you.
Thank you very much, Elijah. It was really fascinating. So we have time for a couple of questions, if there is any from the audience. We have one.
Thank you, Elijah. This was huge.
Thank you.
Do you think also regarding the epithelial mesenchymal transition that is possible, since you are doing bulk sequencing, right? Since if it's possible the opposite, that the stroma is differentiated to epithelial to replenish and, I don't know, repair the injury?
It might be possible, but from our bulk RNA-seq experiment, we only collected the epithelium, not the stroma. So it is clearly an epithelial signature. But whether the stroma will contribute to that, because we are studying the epithelium in isolation, I can't say for that.
So it's a bulk of purified epithelial cells?
Yes, because we cut the cyst out and then we collected only the cyst part.
I see, thank you.
Maybe I can ask. Oh, there's another question, please.
Thank you very much. I'm not very familiar with the system, but I was wondering whether after injury there is a change in celiogenesis, and if there is, what can be the consequences for, development?
Yeah, we haven't really looked at the celiogenesis just because we saw that there's a decrease in those celial marker, and I think we are running an experiment whereby we keep going through the samples for longer to see if it recovers any of those maturation features. So hopefully we will have those sets of data soon. But for now, I can't tell you.
Okay, thank you.
Thank you.
Maybe I can jump in with a very quick one. It was fascinating to see how many neurons you got in your organoids. Did you guys profile them in order to understand which type of identity they acquire, if or if they mature over time?
Yeah, so we didn't profile the neurons. We focused specifically on the epithelium, but I know in our lab we have plans to do that, just to see where it leads to and how long we can push the system, whether the cortical also matures along with the choroplexus. So, yeah.
Thank you so much, Elia. So please
Thank
join
you.
me in thanking him again, and now we move to the next speaker.
So the next speaker is Violeta Silva Vargas from the University of Basel, Switzerland. And I guess that now is going to appear also her presentation so that I can introduce her better. Can you see it? Okay. And she's going to talk about the quadriplexus and physiological states. That is a dynamic duo. So please, the floor is yours.
Try to click on the slide, on your name.
Yeah.
Here we go. Thank you.
Thank you. Well, special thanks to the organizers and also to Anna Rita that cannot be here today for putting us together and in this amazing venue, and thank you for coming. And so today I've had a fantastic introduction, so I think I can flick through some of the introduction. So I'll talk about a bit different. So it'll be choroplexus and physiological state. And this is mainly in the adult mouse brain. So, as you heard, the cerebrospinal fluid, this molecularly complex fluid that fills the cavities of our brains. And it not only provides brain cushioning and a very important delivery system for signals and nutrients, and also it has very important waste removal function. So CSF is produced by the choroplexus, and okay. So this is the laser is with the mouse.
Pointing? No, you can point.
Yeah.
Just keep pressing.
It's the top one.
Yeah.
Okay. So produced by choroplexus present in each ventricle. Lateral, third, and fourth ventricle, and CSF, it's constantly flowing in a unidirectional manner. So from the lateral ventricle to the third, and then into the fourth, where it's reabsorbed into circulation. And this happens several times a day. And so factors from the CSF are in direct contact with the ventricular, subventricular zone stem cell niche, and that's why we started our interest in seeing how CSF factors regulate stem cells. And so importantly, the composition of CSF is not static, and as you've heard already from the previous talk, it changes throughout development. And importantly, from very early on, it encompasses changes driving some of the key processes during development that first make up for the formation of the brain, but later in adulthood can regulate stem cells and also in aging. So this is also across short timescales, so CSF composition dynamically changes also during the day and the night. So choroplexus, already you heard, but in addition to making CSF, it's a very important barrier and entry route into the brain. And so here you see a whole mount of a choroplexus stained by CD31. You can appreciate the vast vascular network that then, if you look in this cross-section, you see that this central vascular core, it's surrounded, covered by this epithelial layer that you heard, that So these epithelial cells are joined by tight junctions, which then constitute the blood's CSF barrier. And then also, these cells are very important providing a detoxification system by providing a lot of enzymes into the CSF. And importantly, it's a transport hub. And usually we are hearing of blood into the CSF, but I would say that I think both directions are important to keep in mind. And then ultimately this important also surveillance hub for immune system. So today I will focus more on a function that is as a reservoir of signals that then are secreted into CSF or the other way around. Okay, so then already in the 10 years, let's say 10 years, there's been kind of an explosion in really bringing back choreoplexus in a position that, in addition to making CSF, really is shaping the brain environment. And so we and many others have shown that then has an important role in stem cell regulation, but also in sensory function and cognitive function, as well as, for example, very important for providing cortical period plasticity, as well as a clock regulator. And only this morning I saw in bioRxiv that the choreoplexus is the first clock ever to be active in the brain. So this is kind of very exciting. And so then it's not surprising that its role in disease is kind of being really reevaluated. So I think it's clear the involvement in Alzheimer's or Parkinson's, but I think in general, I think everybody is kind of stepping back to the early events and seeing how choreoplexus can contribute or contributes to disease. So an important function, and it's kind of our now working model, is that the idea that the choreoplexus acts as a sensor of body state. So cells in the choreoplexus express a large array of receptors for different classes of the main growth factors, hormones, and neurotransmitters, as well as also physical and chemical signals. So the idea is that with this receptor repertoire, they can sense and receive inputs either from the CSF, by other brain areas, from the blood, but also via innervation or by the immune arm of this special structure. And so then these different inputs are integrated, and then the choreoplexus can respond by either changing the transcription or the secretome, and so therefore changing the composition of CSF. And this, as I said before, it's actually a very dynamic and surprising, extremely fast, or can be extremely fast process. So about 6-7 years ago, we started a very exciting project in which we wanted to look at lateral ventricle choreoplexus across different physiological states. And so today, I'll just give you an overview on sex differences, so look at male and female, as well as in circadian, so throughout the day. So, so we took lateral ventricular plexus from male and female mice. So we did, so we looked at the transcriptome, the proteome, and also at the secretome. At this, we recently uploaded in bioRxiv. If you are more interested in more details, don't doubt to reach out for more discussion. And so if we just look at very general, so I'll just give you some snapshots, but if we look at the GCA, so global analysis of the transcriptome, we see majority of enrichment in the female transcriptome of processes that are involved in immune related. And then, and also secretory signal. And so in the male, it's more related, so you have less enriched genes, and then it's more related to cilia and different aspects of metabolism. And so we took advantage of Alan Brain ABC Atlas just to have a look to see these changes occurred in a particular cell type. And we see that some of the, so here I just show you some of the DEGs, differentially expressed genes. These are expressed throughout different multiple cell types. So for example, here is a male enriched gene, this BST2. This is a very important entry route of HIV virus, for example. and this kind of I was talking to you about the immune hub function of choroid plexus. And then here another alpha-2 microglobulin, a female-enriched gene. It's also a very curious protein with multifunctionals that can change, for example, CSF composition. And it's involved in AD, for example. So you heard from the first talk, choroid plexus has many resident cells, very different cell types. So in addition to the epithelial layer and the vascular and the perivascular components, you also have these immune, different types of immune cells. And so we wanted to know, so from the DEGs, is there a particular cell identity that is more represented in this, in the female or in the male enriched genes? And so we ran this cell identity inference analysis, and you can see here highlighted in yellow that macrophage and microglia signature is quite predominant in the female transcriptome. So we then went and see if this would, correlate with a change in immune cells, and so we looked for macrophages. So we look for IVA1, and we see this is in a choroid plexus whole mouse. You see an enrichment of macrophages in female choroid plexus. And so macrophages in the choroid plexus are called BAMs, and as known as BAM-associated macrophages. And so these are specialized type of macrophages that reside in the different borders or entries into the CNS. And so in the choroid plexus, there are two types. Those that are residing in the stroma, stromal macrophages, and those very special cells that live on top of the apical layer of the epithelial cells facing the CSF, known as epiplexus. And here you can see in the arrowhead is showing epiplexus, and here you can see this then stromal, more elongated. So when we look in cross sections, we see that an enrichment here in these red arrows, enrichment of IVA1 positive macrophages in the female. However, when we look into the epiplexus, we see that actually there is an enrichment in the male choroid plexus. This is quite exciting because not only this shows kind of a subtype sex preference, which could be interesting functional consequences. So one aspect we saw in the global analysis was potential differences in secretory signaling. So we the next wanted to see if there was sex difference in the choroid plexus secretome. So before we had shown that choroid plexus secreted factors can regulate stem cells and their progeny, which are kind of across the street within the ventricle. So we took advantage of a system we developed in which we can excise the entire structure from the mouse brain, and then we can condition for a few hours and then extract choroid plexus derived factors. And so with these, we could test their functional effect on, in this case, we FACS sorted from the in vivo brain male or female neural stem cells, and then expose them to either sex match or sex swap choroid plexus factors. And so then what we saw is, So the graphs, the bars have vanished, but you can see the individual experiments. So what we saw is in both cases, the female choroid plexus factors can elicit much higher clone recruitment on both male and female cells. And interestingly, female factors can recruit more clones, more cells to stem cells to activate in the case for the female cells. So when we look, for example, then at canonical ligands that are known to regulate neural stem cells, then you see that there is differential expression across male and female could potentially explain some of these effects. And so, so one thing we did then so one part I didn't tell you, so either for choroid plexus secreted factors or from mouse CSF, so we looked at those that were male or female enriched, and then we saw that when you run in this disease platform logarithm, then you can see that, from these factors, you see the emergence of different types of disease enriched either on the male or on the female, including neurodevelopmental or neuropsychiatric disease. So it'll be interesting to see how this then correlates. And so this also prompted us to look in the human. So our main model is the mouse. So we wanted to see, are there also sex difference in the human lateral ventricle choroid plexus? So we looked at both a transcriptomic level and a proteomic level on human CSF. And so as a global view, we see that, for example, if we look at enriched biological processes, in red, the female, you see also most of the terms are related to immune-related processes, as well as a response to external stimuli for the female. And then for the male, you see this metabolic and cilia-related. And so when we look, for example, then at what cell type is represented or more represented in the female transcriptome, we see again this microglia macrophage signature for the female, and then this epithelial ciliated phenotype for the male. So when we, for example, so we've done transcriptional factor mapping, and we see that, so if we look at those transcription factors that were active in the mouse female transcriptome, for example, we see a high correlation with the human female transcription factor activity. And more interestingly, I think this is my favorite part of the slide, is that if you then look at some of these transcription factors, we're not really transcription factor lab, but what was really interesting is that if you look at these transcription factors, first of all, you see the similarity in the activity on both mouse and human transcriptome, but especially the fact that some of these transcription factors are directly responding to different cues, external cues. For example, hormones in terms of sex hormone, but not only, oxygen levels, immune status, and global metabolic inputs. Just to show you that, before I referred to receptors as being, but also at this transcriptional level, there is a greater response by this organ. So, and then just finally, so if we look at sex-enriched factors in the mouse proteome, we see also that we see them coming up in the human CSF proteome. And here, just some examples, including this A2M that I referred to earlier. So we see similar sex differences between the mouse and the human choroid plexus at the immune compartment and secretory signaling, and this is kind of opening an exciting avenue for just looking into more effects on the human choroid plexus biology. And so another short story I wanted to share with you today was done in collaboration with Selina Hartman, a PhD student in the lab, so looking at choroid plexus overall during the day. So as it's already well known that choroid plexus displays circadian rhythmicity, both at transcription level and also at proteome level. And importantly, the clock machinery is cycling throughout the day. And as I mentioned in the very beginning of the talk, it seems that choroid plexus oscillations are preceding those of the central clock, so giving a role that will be exciting to see of choroid plexus driving kind of central circadian rhythmicities throughout the body. And as I said, CSF composition and epithelial cells display diurnal fluctuations. So just as a reminder, mice are nocturnal creatures, so that means that during the day they sleep, and during the night they are in their active phase. So we did a time course experiment in which we took choroid plexus at different time points throughout the day and light and dark cycle. And and just globally you can see, so if you compare transcriptome from day and night, they look quite different, so showing there are transcriptional changes. Just to zoom in very briefly, but if you, for example, look at the transition times, then you see that majority of processes are related to either membrane dynamics, more, again, the cilia, for example, but also other structures, as well as lipid-related signaling, as well as, for example, macrophages and other immune-related. So we are kind of looking in the details of this. And just going back to this secretory signaling, so when we, for example, look at canonical ligands driving neural stem cell regulation, we see that some of them are oscillating at different time points throughout the day, but not all. So this is kind of a specific for a group of ligands. And this was for us very exciting because it had been shown that neural stem cells can proliferate at different rates depending on the day and on the night. So we wanted to test, for example, factors derived from day or night choroplexus if they had different effect on NSCs and could be driving some of these. And so what we did again, we used this fact-sorted assay. We fact-sort the stem cells, and then we exposed to day or night factors. And then we see that indeed stem cells, in this case, were like sleeping stem cells. So during the day, cells respond differently to the day or night factors. So these open very exciting questions in relation whether there is, set of signals that will drive proliferation versus quiescent, whether, there are particular pools of stem cells that would be responding diurnally or nocturnally. And so we are kind of following up some of these exciting questions. And of course, the final slide, and very kind of opening a very important question, is how then, going back to the beginning, how then this choroplexus can sense these changes in physiological state. And so the epithelial layer, as I mentioned, is kind of a central on that function. And so we've been looking now for some years and trying to understand how epithelials are already in vivo and seeing that in addition to what was described, these are very heterogeneous cells. So we are now trying to understand how this heterogeneity impacts these different functions that we described throughout this symposium. And so today I showed you choroplexus is a central hub. So here you can see this structure, this tiny, but seems to be quite important. And sex differences and time of the day seem to be an important physiological determinant for the function of choroplexus affected by the physiological state. And then we've shown you that secretory signaling seems to be dynamically attuned to then change CSF composition and then affect brain environment, as well as then changes in this immune compartment. Of course, the challenge now is to see, not only these two players, there are many others, how they are, cooperating, how they are regulating, and so how in turn then regulate stem cells, but then other areas of the brain in health and disease. And would like to thank you very much for the attention, and would like to especially acknowledge the yellow names. So Carol Kaiser, a postdoc in the lab, which together with Luca, a PhD student, and myself, we worked together in this sex difference project. And then Selina Hartman, a PhD student, we are doing this circadian. And thank you very much.
So thank you so much, Violeta, for a very interesting presentation. So I guess that we have time for a couple of short questions, if somebody has some. Yeah, sure. There.
Thank you. I'm wondering about physiological factors that can influence choroplexus expression of molecules. You see this very big difference in the circadian rhythm. But what about food intake? Do you have any evidence of this? Is there anything in literature?
Yeah, I mean, that's a fantastic question. And yes, definitely there will be an impact. So I think now there's a beautiful study showing that, for example, in a situation of gut inflammation, you have direct impact on the choroplexus, like barrier, for example, and other functions. And so, yes, I think the dietary input per se. There are some different studies suggesting, but whether then you have different diet effect, etc. This is kind of, yeah. I think several groups are looking more in detail into that. But definitely there is a communication, and yeah, that's a great question.
Thank you. Any other question? So I guess that I can just ask you a very brief one, and then we move on. I was wondering, your studies about the sex differences happen like during adulthood. So I was wondering if you have any insights about when these changes are like happening between the two sexes, or if they are like embryonically derived, or like they change dynamically during lifetime.
That's a great question. So we've mainly focused on the adult. However, of course, yeah, something I haven't mentioned, but we've looked into is the estrous cycle, and so of course that comes later in puberty. So yeah, after postnatal P21 in the mice, and so yeah, we haven't really looked at then what was earlier different. Or there are, for example, on the microglia macrophage, there is kind of a lot of literature showing that this is actually even sex differences are dynamic, and so I would not be surprised that then, yeah, this is a dynamic, yeah, and I would say that this also in relation to the epithelial maturation, this is kind of also very important. So I would say this is not static, perfect. Literally.
Thank you so much.
Thank you very much.
So we move on, and while we're actually very sad that Annarita Patrizzi had a family emergency and could not join us today, I'm pleased to introduce, and I wish to thank you, my co-chair, Valentina Scarpetta, for presenting the work that she did, and part of additional work that was done in Annarita Patrizzi when she was a PhD student in the lab. So she will be presenting the chorioplexus and the non-cell autonomous brain regulation. Thank you.
Thank you so much, Simona, for the kind introduction, and thank you all for attending. I'm sorry that Annarita couldn't be here, but I'm going to give the presentation on RB alpha, and I'm going to talk about this non-cell autonomous regulation. First of all, I think that I would like to highlight that the research goal of Annarita's lab has always been to try to understand which is the non-cell autonomous control of the brain-body communication. And recent evidence are discovering that the choroid plexus could be one of those. And this is due to the central position of the choroid plexus at the level of the ventricular system, and the many different cell populations that are occupying the tissue, and different compartments, such as, for example, fenestrated vessels that are regulating like the arrival of information from the periphery, and the semi-permissive barrier that is created by the tight junction that is instead regulating like the exchange of molecules with the central nervous system and the rest of the parenchyma. And so I guess that we can actually skip the presentation about the choroid plexus, but I would like to highlight that it has been historically described that the choroid plexus epithelial cells are the major population, and they play the major role in the secreting and producing like the cerebrospinal fluid in the ventricles, and that these cells are like specialized for the secretion of the cerebrospinal fluid. But there are many other different cell populations at the level of the choroid plexus that we didn't want to do this, that are populating both the stroma and the apical side, as Violetta already mentioned. And I would like to highlight the differences, especially between macrophages and Colmer cells, that are these epiplexus cells, because macrophages and Colmer cells are transcriptionally different, and also their location is changing. And we are going to touch more this topic at the end of this presentation, but this is going to be important for the regulation of the responses of the choroid plexus in many physiological and pathological states. So, in the last decade, a lot of papers have been published about the role of the choroid plexus in general, and I think that it's now recognized that the choroid plexus is playing a very important role in regulating brain homeostasis, and also thanks to the release of different nourishing factors and growth factors at the level of the cerebrospinal fluid throughout lifetime, it's able to regulate the adult neuronal stem cell homeostasis and development. But the real important thing thing is that also the choroid plexus is an important immune getaway, and it actively regulates the autoimmunity of the central nervous system with the many different cell types that we described previously. So the central position, all these different immune cells that are populating the choroid plexus, makes it important also the study of the choroid plexus in many different physiological but especially pathological stages and states. And these are spanning from infectious diseases, but also like neurodevelopmental and neurodegenerative disorders that we are going to touch more at the end. So I would like to wrap up like the main goal of Annarita's laboratory, that is how does the choroid plexus is able to sense or respond to the stimuli, and which are the stimuli that are actually affecting both the morphology and the function of the choroid plexus. And so when I first started my PhD in Annarita's laboratory, I started investigating one of the major processes that is determining some changes at the level of the choroid plexus. And so we started our analysis on the morphological changes happening at the level of the epithelial cells. And what we discovered is that there are many different compartments of these polarized cells that are actually changing during aging. And these changes are happening very early during development, starting already in 8-month-old animals, and then they are becoming extremely evident in 20-month-old animals, so that is almost 2 years. And these changes are affecting, for example, the capability of communicating with the cerebrospinal fluid, with a reduction of the microvilli, that are this brush border that is increasing the permeability and like the acceptance of molecules that are in the cerebrospinal fluid, but also changes at the level of the basolateral invagination that are here displayed like in magenta, that are becoming larger and like increasing their area, kind of suggesting that there is an increased permeability to molecules that are coming from the periphery. But given the lack of time, I'm not going to like describe all these changes, but what I would like to focus are the changes that are affecting the mitochondria. And of course, when we started like our aging study, we decided to focus on the mitochondria because it was the one in which we were expecting like the most changes, because it's an hallmark of aging, the fact that mitochondria are changing. But we were expecting like morphological changes more towards a decrease in their dimension and an increase, for example, in the fission of mitochondria. And we were very surprised to see that actually the mitochondria of young adult animals are already like spheric, as you can see here in the first image in 2-month-old animals, while they are becoming more elongated here in 20-month-old animals. And we kind of started wondering if this could be correlated to some functional alterations at the level of these mitochondria, and so we decided to perform more functional experiments. And to keep the integrity of the choroid plexus tissue, we performed this analysis by combining 2-photon analysis, live analysis, on choroid plexus explants that were kept in oxygenated CSF for keeping the tissue alive and functional. And what we have done is tracking the movement of the choroid. It's not working, I'm sorry, it was supposed to be a video, in which you can actually see, you should have seen the movement of these mitochondria. Trust me, it's not a movement in space, but it's kind of a wiggling of these mitochondria like in position. And this wiggling movement is already peculiar, and it can be like determined by the fact that there are a lot of mitochondria at the level of the cytoplasm of the cells. So we tracked what is changing during aging in this movement of the mitochondria that is correlated to the capability of the cells of producing ATP and so energy. And what we saw is that actually if you compare the mitochondria movement of young adult animals with old animals, you can clearly see that there is a reduction in the total displacement of these mitochondria. And so we started wondering if this could be correlated to something like more functional. And we decided to perform another experiments with 2-photon and choroid plexus explants, but this time using another gold standard probe for tracking the mitochondrial membrane potential. And the TMRM is a probe that is perfect for this, and its fluorescence intensity is kind of directly proportional to the capability of the electron transport chain of working, and so determining the maintenance of the membrane potential. And what we saw is that during healthy aging, there is a significant decrease in the mitochondrial membrane potential in aged animals compared to the young ones. But when you actually apply some drugs that are blocking the functionality of the electron transport chain in 2 different moments, so at the beginning of the electron transport chain, so only decreasing the functionality and the capability of producing ATP, or like completely shutting down the membrane potential, you actually see that old animals and young animals are reacting with the same kinetic. That is kind of suggested that even if there are basal differences between the young and aged animals, the choroid plexus of old animals is still able to buffer detrimental stimuli that can come from the external environment. Another organelle that is actually changing a lot during aging, but in a different time frame, is the cilia. So my colleague, PhD Kim, started studying like the cilia structure at the level of the choroid plexus epithelial cells. And here is just a schematic overview of how the cilia is easily formed. So it's composed by a basal body that is anchoring the cilia to the apical side of the epithelial cells, and by an axonym that is like a microtubule-rich structure that is ejecting inside the cerebrospinal fluid, and it's the longest part of the cilium. But in literature, when we started digging into it, we didn't find a lot of information about which could be leading the formation of the cilia, their structure, or even their maintenance. So how did we arrive to the cilia? The goal of Anna-Rita was trying to track how the choroid plexus epithelial cells are forming the tissue. And what she did at the beginning was like performing a canonical birth dating experiment in which animals have been injected with a single shot of EDU that gets incorporated inside the cells that are actively replicating, and during different time points during embryogenesis and during postnatal days 2, 10, and 17. Then all the animals have been sacrificed at P25, and we started tracking when these cells are proliferating and how they are disposing in the tissue. And you can clearly see that there are actually 3 different locations. And so the animals that have been injected early during embryogenesis see the majority of the proliferating cells that are in the most distal part from the cortical hem. While the animals that have been injected later during embryogenesis, so at embryonic day 16, see the majority of the proliferating cells in the most proximal part. And this is suggesting that this choroid plexus is formed by a treadmill of these multiciliated cells that are arriving into the tissue and then disposing in time and space. So what we wanted to know was, okay, these changes are happening very early during embryogenesis, and then they are not changing, but what is happening in a perinatal or postnatal period? And so what we did was like performing some immunofluorescence reaction to cilia markers to highlight the basal bodies and the axonym of the cells in both murine and human choroid plexus. And what you can clearly see is that already in 1-month-old animals, you can see a reduction in the number of multiciliated cells that is already happening in P35, and then it's staying stable again, once again, during aging, and also the cilia length of these cilia was decreasing with the same kinetic. And it was interesting to note that when you stain also human tissue, you can actually see that this happens more or less with the same kinetic, and so this is something that is preserved through species. So this was, I'm not sure that this video is actually going to work, but this is a 3D reconstruction of how the cilia look like on the apical surface of the epithelial cells. And their structure is pretty peculiar, and the structure of the cilia can be determining also which is the function of the cilia. That is still something that is unknown. So if you look at this, you actually can appreciate that in embryonic stages, the axonym is very long, and when you cut the cilia, the axonym of the cilia, you can actually see that these microdoublets of microtubulin that are forming the cilia are disposed in a very organized way in the space, like forming this circle. And they are anchored to the basal body here, as displayed in H, by these basal feet that are like displayed in purple. But when you look at postnatal days, so what happens during postnatal development, you can clearly see that there is a rearrangement in these microtubule doublets, and when you come closer to the basal body, the structure is still preserved, while when you move to the axonym that is getting like reabsorbed, like we said before, you can actually see that it's very, very disorganized. And what is leading to this? How are these cilia actually dynamically changing? This is something that we still don't know. What we can infer and what we can say is that these tubuli dimers are actually continuously adjusting in space and time with a continuous subtraction and apposition of different microtubule structures. And we started investigating many different ways in which this reabsorption of the axonym can happen during aging in the choroid plexus. And what we actually describe is that there are some enzymes that are leading to acetylation and deacetylation, and like to the construction and the subtraction of the doublets. And we demonstrated that there is actually during development, during aging, a decrease in the mRNA expression of some of these enzymes that are leading to cilia assembly, and a decrease instead in the enzymes that are leading to cilia disassembly. So this could be one of the reasons, one of the many different pathways in which the cilia gets reorganized, but the function is still something that is unknown at the moment. So I hope that with this I demonstrated that there are a lot of different dynamic changes that happen in different time frames at the level of the choroid plexus tissue. And for example, here you can see that what happens in 1 month old was like changes at the level of the cilia that then were staying stable during aging. Maybe instead the changes at the level of the epithelial cells are happening later during development and during aging, so around 6 to 8 months old, and then again they are staying stable during aging. And so we started wondering if some of these changes could also be present in other conditions that are related to aging, but not in healthy conditions, so in pathological. And the natural evolution of this study was trying to find a model in which we can study like a pathology that is related to aging, and of course we selected like the Alzheimer's disease. And we screened many different models that I'm not going to go into detail at the moment. So what I'm going to present you today is like some sneak peeks of a study about APPPS1 animals that are like a standard mouse model for the study of Alzheimer's disease, in which the major modifications are happening around 6 months old, in which the disease comes symptomatic. And the first thing that we did was try to understand if the hallmark of the Alzheimer's disease, so the deposition of amyloid and plaques, is happening also at the level of the choroid plexus, given the central position and the involvement in the metabolism of the amyloid. And to our surprise, we didn't find a significant increase in the deposition of these amyloid deposits. So we also demonstrated it by electron microscopy, in which we like analyzed animals until 12 months old, so in which a stadium in which this disease should be fully symptomatic, and even here we didn't find any difference in the deposition of visible plaques at least. So I decided to screen like many different compartments of the cells to try to replicate also the study that we did like during aging, kind of expecting some major reorganization of the epithelial cells. And instead we were surprised to see that when we analyzed both the mRNA expression, the protein expression of different transporters at basal and apical side, we didn't find any significant difference in these cells. So it seems like, at least until 6 months old of age, the morphology of the epithelium is kind of preserved. And so I started wondering if, given the many different populations that are populating the choroid plexus, there could be one population that is affected during the disease. And so, of course, we focused on the immune cells, and I'm just going to give you like a brief idea of our findings, but if you want to know more, you can also come to my poster later in the afternoon. And as Violetta mentioned before, there are different populations of immune cells, and this is the major distinction between stromal and epiplexus. Stromal are like in the stroma, they are macrophage, so they also have like a more phagocyting role, they are like cleaning the debris, and they have a different trajectory like origin compared to the epiplexus. The epiplexus are a unique specialized microglia-like population that is only present at the level of the choroid, and their function is still unknown. There are multiple studies that are trying like to discover it now, but it seems like thanks to their position, they are more able to like sense the signaling molecules that are present at the level of the cerebrospinal fluid and communicate with the rest of the brain. And so our focus was like to try to understand what is happening at the level of stromal and epiplexus macrophages, and we were surprised to see that actually in disease, and especially in the symptomatic phase, it seems like there is a significant increase in the number of macrophages that are present in male choroid plexus compared to females, kind of sensing that there is a sex-specific response of the immune cells in a condition of disease. For the epiplexus cells, it happened the same thing, and again in 7 months old, so these changes are happening when the disease is fully symptomatic and not before, and this is there is kind of a tendency of it happening also in the females, but also with a different time frame. That is something that we can discuss later. So with this, I think that I will conclude my talk, and I would like to thank you all for the attention personally, and I guess that Anna-Rita would have loved also to thank like the entire laboratory that collaborated to these studies, and all the past members and the fundings that like are supporting her in all these many different like studies that she's pursuing, and all the collaborators that participated. And I will be happy to take any question, and I hope that I can answer correctly.
Thank you so much, Valentina. I think we have time for a very quick question. Maybe I'll break the ice. Maybe we do two very quick. All these different changes that you guys are observing over time, it seems from the neurodegenerative model, they don't seem to suggest it to be an alteration, but do you have a feeling if they can be positively selecting the cells in a way or negatively impacting in aging? Is there a way to push the time window for the cells to retain the length of the cilia?
That's a very good question. For the cilia part, I honestly don't know. I guess that I think generally that these changes are like something that is positive, and this like there are multiple proofs that can demonstrate it. For example, also the fact that the choroid plexus doesn't proliferate, so whatever it's part of the tissue, it has to be there, and it has to continue, like keeps the energy demand, sustaining the energy demand, and like the function of the choroid plexus throughout lifetime, because they still need to produce and secrete the cerebrospinal fluid. So my theory is that all the changes that are happening are more like adjusting the tissue and their functionality like to the rest of the environment, because it needs to keep sensing and adjusting to the other molecules that are coming from the external. Regarding the cilia, I have the feeling there are some mouse and zebrafish studies in which actually, the cilia are beating when in the first like perinatal phases, and then they are in the mouse, for example, they are not moving that much, so they are not moving the CSF, but they could be signaling and sensing some information. So I guess that the structural modifications are related to this capability, but it's still not known how it happens. Sure.
Beautiful talk, thanks. I had a kind of follow-up question. So you showed, well, we didn't see, but the mitochondria moving, and I was wondering if there is any relation with the ciliary patch, and whether it's moving or any correlation or relation?
That's a very interesting question, thank you. The easy answer is that I don't know, but it could be, because, usually mitochondria needs to move like a lot in the cytoplasm, like they follow like the cytoskeleton remodeling of the cell. And what I think that is happening is that, for example, the decrease in the movement during aging is because like the cytoplasm is becoming more fibrotic, so maybe also the cytoskeleton is more difficult to follow like in the movement. But the changes that we see at the level of the cilia are happening very, very early when the mitochondria are still like sustaining their activity. So in 1 month old, I don't think that that's like there is a kind of correlation. I think that it could be because there is a metabolic switch in the time frame that has been proven also by the Latin laboratory. That it could be that the change of metabolism of the mitochondria, not really their movement per se, is maybe affecting the reabsorption. But actually, we also demonstrated that there is an active mechanism due to a spastin enzyme. So there could be many different ways in which it's altered.
Okay, thank you so much.
And now, I'm going to introduce Simona Lodato, that is coming from Humanitas in Milan. And she's going to give us a talk about
Choroplexus organoids.
Choroplexus organoids. Unlocking the guardian of the brain with choroplexus organoids. Please, the stage is yours.
Thank you, Valentina, and thank you very much, all of you, for staying. We started a little bit later. We're trying to keep you not too long. I was very fortunate to be the last one today of this session, so I basically can probably skip a lot of the intro, because I'm sure I just want to give you a sense of why we are in the choroplexus business. My lab has always been interested in myself and my lab, always interested in how the genetic programs, the activity-dependent processes and environmental inputs can actually converge and interwave in shaping brain development, with a particular focus on the cerebral cortex. And today I'd like to focus on environmental inputs by defining, as hopefully you have grasped from our session today, that the CSF is actually the first environment that the developing brain is exposed to. You have heard beautiful talks before me of how this CSF can impact on many different aspects of development of stem cells or different kind of states of the body, and how much this it's the medium through which basically our body can perform the interactions with the brain and the rest of the body. You have heard that it's produced by this very peculiar and underlooked structure that is called choreoplexus. You have already seen where it's located, very centrally within the ventricles, and actually it formed extremely early during the neural tube definition, around gestational week 6 in humans. We already have the stemming of the structure, and as soon as it's formed, it starts producing this fluid that actually occupies a very, very large volume during the early stages of our brain development. You have also heard how much complex this structure is, how many different cell types. Despite the chief cells have always been considered epithelial cells, and we've heard how important and diverse they can be. There are many other different cellular contributors, and a wealth of literature defining how much the CSF can indeed impact being a hub of signaling, not only the development, but in general brain function. You have already heard about how the secretome can support the recruitment of different types of stem cells, how the stepwise coordination of immune-mediated barrier repair is relevant, and we were lucky to contribute to a recent study a few years back already, where also not only the immune cells or the epithelial cells, but also the endothelial cells, which are represented by a fenestrated, rather permissive kind of endothelium within the choreoplexus, can actually have an unexpected role in regulating what actually passes to the brain and what does not pass to the brain. Indeed, the endothelium seems to be an inflammatory condition, so in other challenging conditions can be selectively closing, shut it down even before the epithelium, and disconnect the brain from the rest of the body. So this is just to tell you, at the end of our symposium, how much the entire tissue, the entire complex tissue of the choreoplexus, which is in the brain but is not like the brain tissue, can actually be relevant and impacting and how the brain function, and that's how we got interested in this structure. You have heard beautifully from Elia this morning how species, and from Violetta, how species-specific difference may actually justify the idea to look into the comparison between human and a mouse system. And given the early stages of these aspects that we are interested in, we also were troubled by the idea of looking into human brain development at very early stages, given the limited access. So we turned into the 3D organoid system, and we were lucky to walk on the shoulder of giants when they, by following the work that Laura Pellegrini and Madeleine Lancaster did already, and established the first 3D structure human-derived organoids for the choreoplexus, where you have seen from Elijah this morning, mainly the neuroepithelium is generating, and the epithelial cells are largely represented, diversified, and as well as neurons in that. But with this idea that we developed over time, that many different cell types might be contributing to the complexity of this tissue, we really wanted to take up the challenge of generating a choreoplexus organoids that contain a rather vaster, larger complexity, including both the ectodermal-derived components. As you might have memories of your neurodevelopmental embryology class, epithelial and neuron are actually derived from ectodermal and neuroectodermal, which is very important from stem cell biologists because it tells us which kind of cocktails provide our iPS cells when we differentiate, but also endothelial immune cells has mesodermal derived, so totally different choice that the embryo actually has to make very early on, and we want to be happening within the same 3D structure. this challenge of capturing chorioplexus heterogeneity to mirror what was this multicellular specialization of the chorioplexus was taken upon by a brilliant PhD student in the lab, Vanessa Aragona, who recently defended her PhD. And the idea was indeed to generate a novel protocol for 3D human chorioplexus model that could recapitulate all these different cell types. And eventually, to be able to respond to many questions, no answer will be provided for that today, I promise, but the idea is that we could add a more faithful system where at least the crosstalk within the different cell types of the chorioplexus could be investigated in a human system. So a lot of attempts, and of course I'm showing you the successful results, but Vanessa was really stubborn, and eventually she developed a protocol where she could simultaneously, sort of in a staggered way, induce both the ectodermal differentiation, then sort of hold on that, just nudge the cells into an ectodermal patterning, and then eventually adding a mesodermal differentiation to it, to form what we name our vicus, vascular, and immune chorioplexus organoids, where both the ectodermal lineage as well as the mesodermal lineage will be generated simultaneously in the same patterning protocol and in the same three-dimensional structure. Unfortunately, I can't share too much about the details, not only for sake of time, but also because it's protected by patent at the moment. But what can I tell you is that we were able to follow our early neuroepithelial induction here. As you can see, genes that are highly expressed in the early stages of neuroectodermal induction, like OTX2, PAX2, SOX2, and also later at different time window of the patterning, the mesodermal priming and specification, and eventually the post-induction and maturation. So we could see how this would develop within the time window of the induction. Then Vanessa started looking into these organoids. Oh, sorry, I didn't mention these organoids can be growth for a long time. This is an example of a 9-month-old, and as Elijah show us beautiful, they develop large cysts with fluid field structure that resemble, at least to a certain extent, the CSF production. Vanessa started looking into the composition of these cells, both into these organoids, both at the FACS sorting, so isolating the different population, as well as by immunofluorescence compared to chorioplexus derived from fetal tissue, gestational week 16. And hopefully you can appreciate here that we can isolate both endothelial cells, macrophages, epithelial cells, and fibroblast-like mesenchymal cells in different proportions. This was also very helpful to be able to quantify how reproducible the protocol was across different batches and different organoids. And on sections, we could appreciate the presence of the epithelial cells staying with this beautiful aquaporine on this cuboidal epithelium, but also the presence of endothelial cells labeled by CD31, as well as the presence of ciliated ependymal cells and eventually macrophages. So it seems that in these organoids we had the contribution of cell types that are actually coming from different lineages. But to really be convinced and unbiased, we perform single-cell sequencing. So Vanessa paired up with two fantastic people in the lab, Ilya Simutin, which is a PhD student that is a bioinformatician, and Sara Mancinelli, who is an outstanding postdoc that is basically involved in every project in my lab. So she has a talk this afternoon. Please go listen to her because it will be a blast. And Sara was involved in the previous studies on in vivo chorioplexus, so she was really instrumental to characterize this model. So what Vanessa and the team basically did was profiling organoids derived from both ES and IPS. So we had both embryonic stem cells and induced pluripotent stem cells at different time points, day 30, day 50, day 70, and now we have later stages from different organoids and different batches. And we could confirm in an unbiased manner that we have both ectodermally derived epithelial cells, both ciliated and non-ciliated, as well as mesenchymal cells, immune cells, endothelial cells, as well as neurons. So this really convinced us that across this differentiation, we could generate a rather heterogeneous plethora of cell types that had dual origin. So we start looking more into details of the arrangement of the structure and the epithelial and endothelial cells as they are organized in the human chorioplexus are very close to one another, just juxtaposed, as you saw in the graphic that we have been showing today. And this is the case. The organization was very similar, and it was not random, highly patterned, where, sustaining the crosstalk between the epithelium and endothelium in the structure. We went looking at the morphological level at the composition of this epithelium, and also ultrastructurally the presence of, we could confirm the presence of different kind of junction, both tight junction, desmosomes, which were also confirmed at the molecular level in the profiling of the selective clusters of endothelium, which eventually support, as Elijah also saw in Laura's model, a role of barrier of this epithelium. Indeed, we could see that larger molecules that of 10 kilodalton could not pass this epithelial barrier, but anything that was around 3 kilodalton or lower would actually be more permissive allowed in our system, supporting this fenestrated endothelium and a rather limited permeability. We also check for cilia. You have heard a lot from Valentina about cilia. These epithelial cells are ciliated, so they show cilia. We are actually investigating if these cilia are motile, if there is any contribution, but we could observe also the molecularly they express a set of genes that are indeed present in the cilia structure, and as well as the molecular profile, we could see that there was a strong enrichment of cilia in the epithelial ciliated cluster. Now you have heard of the capability of these epithelial cells to sense the environment, even to respond and change their secretome. So it was recently published a beautiful work from Maria Lehtinen from the Harvard Medical School, where they show that epithelial ciliated cells in mouse could actually sense serotonin and respond secreting insulin, a type of insulin that has nothing to do with the pancreatic insulin, but is actually only brain-specific and can be released upon modulation of the serotonin signal. So we wonder if our organoids, human organoids, could actually be a good setting to test if this is happening also in humans. So Vanessa checked first if the receptor for serotonin was present in our epithelial cells, and she could confirm the presence of the receptor and of the secretory path, and she performed an experiment where she administered serotonin externally in the medium and then collect the CSF from the cyst of our organoid. As you can see here, we saw a drastic increase in insulin level within our cyst upon serotonin administration, suggesting that even in human and an in vitro system like the one we provided, this type of apocrine-like secretion modulated by signaling like serotonin that are normally provided by the RAFE in our brain can actually be recapitulated. We also found that this type of secretion, let's see if I manage to show you the video, is calcium dependent. Oops. Is calcium dependent. Yeah, so those are calcium dynamics that are now showing. Those are calcium dynamics upon serotonin administration. We can easily segment this 3D image and follow the traces and the changes in calcium that are sustaining potentially the release of different type of secretome, including the insulin. So there are corresponding calcium traces, and we can follow these in the untreated organoid as well as in the treated organoid with serotonin, how this is triggering the transcription of activity-dependent genes like CFOS, so sustaining that calcium transient are induced upon serotonin stimulation. But I told you we don't only have, these epithelial cells, but we also have immune cells that are, interestingly enough, not appearing as soon as we have the identity of these choreoplexes, but rather appearing later in time, where we have around day 50. We don't appreciate their presence at day 30, following the developmental trajectory that is observed also in fetal development. And also, you have heard about this beautiful heterogeneity also of the immune cells in vivo, and we found that at the molecular level, these immune cells are really specific for the choreoplexus. They are macrophages of the choreoplexus and not immune cells of the parenchyma, like brain cells, microglial cells. We tested at this point if these immune cells are able to respond to any immune stimuli. They can actually actively respond, for example, to IL-6 as an pro-inflammatory cytokine. Vanessa treated the organoids with IL-6, and she could see not only an upregulation in numbers in terms of proliferation of these cells, but also a change in their morphology that are normally amoeboid in a sort of non-activated phase, and they will turn in a more circular shape once activated. They will follow these also at FAC sorting, again, to understand how robust was the response that we were observing. But again, going back to the study that we performed in vivo, we know that any inflammatory stimuli not only would drive the response of the immune cells, but also the epithelium and endothelium were actually dynamically changing upon the inflammation insult. So we wanted to check what happened. And to our surprise, we actually saw, because that study was mainly performed in adult, we saw that both endothelial cells and epithelial cells follow exactly the same dynamics. So meaning closing and opening, so deranging the barrier that is warrant these choreoplexes. You can see here a drastic downregulation of the expression of the presence of PV1, which basically labels the fenestration of these of these vessels of the endothelial cells. So when we no longer can see PV1, it means that the fenestration are no longer present and the endothelia are actually closed. And on the other hand, we see an increase in the presence of ZO1, which is a marker for the zone occludens for the tight junction that instead seems to be increased every time that there is an alteration of the permeability, a loss in the selection of the permeability. So it seems that our barriers also remodels under the effect of an inflammatory insult. But the last thing I would like to tell you in a few seconds is that, again, we're interested in the loop back to the brain, how much the CSF can actually change, and the choreoplexus biology can impact on the brain. So this is relatively preliminary, but I wanted to show you what Vanessa did here. We basically wonder if the CSF that derives from this more complex choreoplexus model could actually impact the activity pattern of cortical organoids that we can develop in a lab and we can physiologically follow in terms of their spontaneous activity. So basically, Vanessa collected the CSF from our vicus organoids and incubate our cortical organoids now, both in brain fees, which is our physiological media, where we incubate for 15 days before recording the calcium dynamics, and in another conditions with 10% induced CSF, the one that is derived from our vicus. So hopefully here you can appreciate, no, it doesn't work. Okay, too bad, it was working all over there. You should be seeing that our spontaneous wave of activity, which are a sign of synchronicity and immature state of the brain, are actually completely lost in the one that have been incubated with our ICSF. You can see the graph here and the quantification suggesting that the CSF can actually impact the maturation of our network in the cortical organoids, and again, supporting that CSF can actively modulate neuronal network excitability. So I hope that with this, I'll convince you that a model that, like ficus, can replicate many important features of the naive choreoplexus complexity and function, including CSF secretion and immune response, the dual tight endothelial barrier layer, but also the dual origin of this structure is something that can teach us a lot of how the cells can influence each other during development and define their final fate. And hopefully this will become a physiologically relevant platform for study both brain development, as well as diseases that can be associated with choroid plexus dysfunction. With this, the most important slides, I already give you the names of the people that have been involved. I'm very thankful for their commitment. Also, all the people in the lab, present and past, and of course the collaborators and the funding, without which nothing will be possible. And thank you all for the attention.
So thank you so much, Simona. Very beautiful presentation. Is there any curiosity or question, like very briefly, and then maybe we can discuss later? Yeah, sure. She's coming with the microphone.
For your wonderful talk. So do your organoids produce BMP, and if this is the case, is this production and secretion influenced by interleukin or inflammation?
That's a wonderful question. Clearly, BMP is one of the pathways that we drive to generate this type of organoids. There is sustained expression, but we haven't checked if the secretome, the BMP changes upon IL-6. It will be very interesting to see. You're right. Thank you.
Any other question? Okay, so I guess that we can move on the conversation like maybe in the coffee break. And thank you all for attending this symposium. It was a great pleasure to present you our data, and enjoy the rest of the conference. Bellissimo, io ho un sacco di domande per te. Very nice, very nice.
I never finished 3 minutes before anyway in my life, huh? So that has to be, reported somewhere. 3 minutes before, okay. I have so many questions. Sorry. Scusate, abbiamo cominciato 10 minuti dopo e quindi ci siamo tirati gli alunni. Però io ho finito 3 minuti prima, quindi vi guardavo, ho detto adesso mi uccideranno.
Buon lavoro.
Vi lasciamo il stage.
Grazie.
Ciao,
Grazie,
ci vediamo
ciao.
dopo.
Buongiorno.
Io sono Tognini. Grazie. Il professore? Ok, quindi lo proiettate voi subito, immediatamente?
Grazie.
Perfetto. Sì, ce l'abbiamo, grazie. Senti, ma devo cominciare?
Scusa.
Un po'? Comincio subito?
Sì, perché siamo in ritardo.
Ok.
Grazie. Andiamo in blocco.
Grazie.
Ce l'ho qui dietro.
We shall start. Sorry, they are telling me that we are late. Good morning. Thanks to the audience for being at this symposium. I'm Paola Tognini from Scuola Superiore Sant'Anna, and my co-chair is Pervitolo Priore from University of Pisa. The first talk will be online. I'm sorry, Professor Magistretti couldn't come, but he is here now with us. Hello, Pierre, can you hear me? Pierre? I think there is a technical problem. So Professor Magistretti from KAUST, Saudi Arabia, will give the first talk. Is neuron-glia metabolic coupling, relevance for neuroenergetics, plasticity, and neuroprotection. I think Pierre. Pierre, can you hear me? Okay.
Hello?
Hi, okay,
Hi.
you are here. Thank you. Okay, we couldn't hear you, and I think you couldn't hear us. Okay, thank you, Pierre. I just introduced you, so if you want, you can share your slides and start your presentation. The stage is yours.
Very well. Thank you very much. Thank for the introduction, and most of all, apologies for not being able to be present today in person, but I was operated last week for bilateral hernias, inguinal hernias, and I was heavily discouraged to travel. So my apologies. I will tell you briefly work that has driven my lab for the last essentially 40 years, which is really to try to understand the cellular and molecular mechanisms that are at the basis of the coupling between synaptic activity and energy delivery to the brain in relation to synaptic function. And of course, this is relevant for functional brain imaging, but of course, also for many other aspects of brain function. And my lab and then many others have shown that actually astrocytes play a key role in this coupling. The work goes back to really many years ago when I was a graduate student at the Salk Institute. As you can see for the picture, that's a little while ago. Working with Floyd Bloom, we showed that actually one of the peptides that had recently been discovered in the early 80s, VIP, was promoting a glycogenolysis in cortical slices. And that meant that by definition, the target was astrocyte because glycogen, as you can see here, is only present in astrocytes. And so we proposed that there was a possible regulatory mechanism for the local control of energy metabolism mediated by specific neuronal systems. Then a few years later, when I started my lab at in Lausanne, first at the medical school, then at EPFL, actually Luc Pellerin joined my lab, and we showed that another neurotransmitter, which actually is much more pervasive in terms of action because glutamate is released at about 80% of the synapses in the brain, that glutamate, a neuronal signal, would stimulate aerobic glycolysis in astrocytes. And aerobic glycolysis is really the utilization of glucose that results in lactate production in the presence of oxygen. It's called also the Warburg effect. And that so was a second mechanism that would couple neuronal activity to glucose utilization. And that gave rise to what is now known as the astrocyte neuron lactate shuttle, whereby glutamate released by neurons, active neurons, will act on postsynaptic neurons, of course, but then taken up and removed by astrocyte through a very active co-transport with sodium that drives the uptake. And then this costs energy to convert glutamate to glutamine and to extrude sodium. And so whenever astrocytes take up glutamate, they will decrease their energy charge, bring in glucose, and the big surprise for us was at the time that lactate was the molecule produced from this glucose uptake. It doesn't mean that there is glucose not going into the neurons. Of course, there are transporters, GLUT3, in astrocytes and endothelial cells. It's glucose transporter 1. But during activation, certainly, the uptake of glucose is mainly through astrocytes. So that's bring me to more recent work related to neuronal plasticity. A question we asked, was, well, this neuroglial metabolic coupling illustrated by the astrocyte neuron lactate shuttle, is it regulated or modulated by plasticity? In other words, will it be more efficient, less efficient? What will happen? And to do though, we actually did many experiments. I go straight to the main one, which I did with Cristina Alberini at the time at Mount Sinai, now at NYU, and we showed in vivo that actually, if one inhibits the lactate transport from astrocytes to neurons, there is an inhibition of long-term memory. So the test was pretty simple, inhibitory avoidance. We injected either DAB, which blocks glycogenolysis, so the mobilization of glycogen, or downregulated the expression of these monocarboxylate transporters, 1 and 4 in astrocytes and 2 in neurons. These transport allow the transport of lactate. And we showed that both manipulations actually block memory consolidation that could be rescued by extracellular administration in the hippocampus of lactate. All were rescued, so inhibition of glycogenolysis, MCT1 or 4 downregulation. MCT2 could not be rescued, which is not surprising because then lactate could not enter neurons. So the conclusion of all this work was that actually glycogenolysis and astrocyte neuron lactate shuttling are required for long-term memory formation. And a decade later, more or less, work done with colleagues in Milano, Elena Vezzoli, and then Corrado Cali in my lab, we used another learning paradigm and novel object recognitions. And what we found, if you look here at the number of spines per cubic millimeter, there is an increase by learning, and this increase is inhibited by DAB and rescued by lactate. Same for spine volume, increased by learning and inhibited by DAB, by DAB and rescued by lactate. So in summary, this data published, as I said a few years ago, show that during learning, there is an increase in spine density and volume, an increase in glycogen present in astrocytes in terms of number and size of glycogen granules, which is very interesting because in a way, this is like a marker for facilitated synapses, that DAB inhibits all this. And the rescue occurs in terms of volume and density for spines with lactate and very little effect on glycogen. So, summary of this, that the transfer of lactate from astrocyte to neurons is necessary for memory consolidation. Neurons need astrocytes to establish plasticity and memory. I like to say not bad for what were considered glue, the astrocytes and the metabolic waste product lactate. But the question was then, what's the mechanism? Is it just for energy or is it some other process? And after a few years of exploration, we found out that actually lactate promotes plasticity gene expression by potentiating NMDA signaling in neurons. This is work that was spearheaded by Igor Alamand at the time. And so just a quick data on gene expression, what we saw now going back in vitro, we showed that on neurons, lactate would induce plasticity genes such as ARC or ZIRF, also BDNF with some delay, but not pyruvate, not D-lactate, not D-glucose. So there was something different than just energy. And with the work of Hubert Fiumelli and a graduate student at KAUST, Michael Mergineanu, we found out that essentially there is about 20 genes whose expression is modulated by lactate in neurons. In green, those that are upregulated, and these are synaptic plasticity. So we found again ARC, BDNF, and ZIF is EGR1 or 2. Neuroprotection, interesting to see this upregulation of genes such as BGFF or BDNF, and downregulation of certain cell death genes such as BCL2. So the idea is that lactate enters neurons through MCT2. It's converted by LDH1 into pyruvate. This will provide energy. But in doing so, there is also a change in the redox state with an ADH formed that can, and ideally, that was a conjecture at the end, modulate NMDA signaling. And over the last couple of years, with work with Hubert Fiumelli at KAUST, what we found is actually probably the molecular mechanism of this facilitation by lactate of NMDA currents. What we found, first of all, that lactate increases the peak current of NMDA receptor-mediated currents. And it's reversible. And if we look into the pharmacology, what we found that this increase in the peak is actually inhibited by ifemprodil, not the PXQX. And ifemprodil actually is a selective antagonist of the NR2B subunit of the NMDA receptor. And what we found in terms of mechanism, we found that actually this change in redox intracellular state because of formation of NADH will reduce two cysteines on the intracellular loop of the NMDA receptor. And this will facilitate the interaction between the receptor and CAM kinase, and therefore increase the efficacy of NMDA signaling. So again, to summarize, lactate enters, it's converted to pyruvate energy, increasing NADH. NADH modulates this subunit of the NMDA receptor. We also show that it mobilizes intracellular calcium by acting on the ryanodine receptor, and all this brings the interaction between NMDA receptors and CAM kinase much tighter. And interestingly enough, work we did with Monica Di Luca and Fabrizio Gardoni showed that actually this increase in redox state and change in the NMDA receptor increases the interaction between NMDA receptor and PSD95. So all funnels to make NMDA receptor transmission more efficient. And this is consistent, of course, with the effect on memory consolidation that we showed earlier with Cristina Alberini. Now, just to finish up, some data on the concept of what does this coupling between astrocytes and neuron do in pathological conditions. Obviously, it's related to energy metabolism in terms of glucose uptake, but also of lactate-mediated signaling. And in fact, there are many diseases that are characterized what one can call hypometabolism. And I will briefly review these cases and show that, and these cases involve genetic disease, GLUT1 deficiency syndrome, Alzheimer, ALS, depression, for which we have examples. First, work done a few years ago with Jean-Luc Martin and Anthony Carrard, we showed that peripheral administration of lactate produces antidepressant effects in essentially three models of quote-unquote depression in rodents: acute forced swim test, open space forced swim test, and chronic corticosterone. And this effect of lactate are associated with changes in the expression of genes that have been shown to be involved in depressive states, serotonin receptors, S100 beta, neurogenesis, NO synthesis, and so on. And later we showed with the same colleagues that in fact, in order for lactate to express its antidepressant effect, neurogenesis, hippocampal neurogenesis was necessary. Also work done even earlier with the group of Jeff Rothstein in models of ALS. We showed that there is a decreased expression of MCT1, the transporters of lactate from astrocytes to neurons, in these SOD mice, but also in the motor cortex of ALS patients. And if one interferes with MCT1, in other words, hampers the delivery of lactate from astrocytes or oligodendrocytes even to neurons, then or axons, then there is axonal damage and neuronal loss. Finally, very briefly, work we did with Lawrence Hirt in the MCAO model of ischemia. We showed that ICV, but even IV administration of lactate up to 1 hour after reperfusions decreased the infarct size and improved neurological recovery. And in fact, with Lawrence, we have now an ongoing clinical trial proof of concept. Now, let me bring to the last phase of the work, which is related to translational work done by a startup company derived from my lab at EPFL. So I have to, of course, declare a conflict of interest because I'm one of the founders of Glaya Farm. And this startup, based on the work of my lab, what it's doing is developing molecules that will boost the astrocyte neuron lactate shuttle. In other words, boosting glucose uptake into astrocytes and lactate production. And what we found is a very interesting molecule, GP119, that does two very interesting things. First of all, it promotes glycolysis in astrocytes by targeting an astrocyte-specific enzyme, so more glucose in, more lactate out. But with some delay, within a few hours, it also increases the translocation of glucose transporters 1, which is specific for astrocyte, to the membrane surface, also not only in astrocyte, but also on endothelial cells. So the idea is that this molecule will increase how much glucose enters into the brain and the conversion to lactate. And it seemed that a genetic disease would be the paradigmatic disease for this, and that's glucose transporter 1 deficiency syndrome. It's a monogenic disorders due to a mutation of the transporter, which, as I said, is present only in endothelial cells and in astrocytes. And so we boost this transfer, and this heterozygous mice model of the disease have less glucose, glycogen, and lactate in their brain. And what happens is, if we did FDG PET, we can show a normalization of the FDG signal. We can see a normalization of the motor discoordination that these mice present. And interestingly, using organoids prepared from GLUT1DS patients that present hyperexcitability with increased burst duration and the burst frequency, there is an effect of the molecule in dampening the epileptiform activity. So quite promising for this disease. Also, with the notion that during aging, there is a decrease in glucose utilization, and that this is correlated with decreasing cognitive performance. So the idea is, let's improve glucose delivery to the brain to combat this hypometabolism. And interestingly enough, this decreased glucose utilization is essentially due to decrease in aerobic glycolysis, which is exactly the metabolic pathway that is expressed in astrocytes. And so using various models, I'll go very quickly, either aging or streptozotocin, intracellular streptozotocin injection of in mice, or the ApoE4 mice. In all these conditions, these mice perform much poorly than the controls, and this is improved by GP119 in all these models. So, and then also, I will go briefly because I'm getting to the end, we have positive effect in other hypometabolic states such as ALS. Now, the molecule is in an interesting level of development. We are essentially finalizing the enabling for IND, and hopefully within early phases of Q1 2026, we plan to do phase 1 studies. So in conclusion, I showed you that definitely I think there is a metabolic interaction between astrocytes and neurons, that this is due to stimulation of glycogen uptake or glycogen mobilization, all leading to lactate production, which has many positive effects. It's a long story, started a bit by chance over 40 years ago, that eventually brought me to study memory, plasticity, and even develop a therapeutic target. Many collaborators over the last few years, even now at KAUST, my lab here. And just to let you know that KAUST is a very interesting place to do a postdoc. And finally, just quick, I can't resist, just advertising this book, Il gioco della mente, that I wrote with my wife. It's a mystery novel about Alzheimer's disease. Thank you very much for your attention. Do you hear me?
From the audience, do you have? Okay, Professor Marsicano is asking a question. Can you give him, please, a microphone?
Hi, Pierre, very nice. I want just to lactate is doing many different things, right? And I was wondering if you think that in all what you described, the impact, for example, of lactate receptor could be present, or now we know with the interaction with serine metabolism, the phosphorylated pathway, and things like that. So could enter in this scheme.
Fully. I fully agree with you. Of course, I know well your work and in Bordeaux about this. Given the time constraints, I had to really go quickly to some key points that we did in the lab. But yes, definitely, I think your work on in which showing that lactate acts also on receptors for lactate and involves the serine production pathway, which is well known also to modulate synaptic transmission, is certainly another mechanism. I think the key point in both work, ours and yours, is that we have a metabolic pathway active in astrocytes that will modulate synaptic transmission. And I think this really is, in a way, it's, if I may say, the cherry on the cake of this neuron-glia metabolic coupling, which from a neuroenergetic point of view, I think now it's been quite well accepted. But I think the new concept is that this metabolic interaction is also a signaling interaction affecting synaptic plasticity.
Thanks. Are there any other questions? Okay, I can ask a question. So regarding these multi-roles of lactate, you talk about also regulation of gene expression and plasticity-related genes. So we know that many metabolites are actually the chemical moiety of post-translational modifications. You think that lactylation of histones could be related to this type of changes that you have seen?
Of course, of course. I mean, there is really overwhelming evidence over the last 3-4 years of the role of lactylation in epigenetic regulation. In fact, we're now working on this also, obviously. And I have no doubt that this is an important modulatory mechanism for gene expression.
Thank you, Pierre. And I think we should move on. Please stay with us if you can.
Yes, of course.
So we have the next speaker, that is Rosa Chiara Paulicelli from University of Lausanne. And with Chiara, we will switch a little bit the gear and talk about microglia. So lactate metabolism in the control of microglial function.
Yes, thank you so much, Paola. Thank you, Piervito, for organizing this symposium, for inviting me. It's really a pleasure to be here. I have to say it's also an honor to have the chance to speak in this amazing venue. So the view from here is really impressive. It's something. So as Paola said, I'm going to tell you something about lactate metabolism and how we think it may control microglial function. I will follow also a bit the flow of Pierre because he explored glia-neuron interaction, but what about microglia? So microglia, are the innate immune cells of the brain. They are multitasking cells. They rely on core properties and they are hypermotile. They move all the time. They are the phagocytic cells of the brain. They release soluble factor. And thanks to these skills, in a way, they can contribute to several biological processes in development and also in pathology. But what is very clear and very striking when you look at microglia is they move all the time. So they are very motile cells, never resting cells. And you can see if the video works. Well, anyway, I don't need to convince you that these cells move all the time and then they make contacts with all the neighboring cells. They also do, in this case, you would have seen a nanoscale surveillance of these tiny filopodia moving all the time. So the bottom line question here is that they take a lot of energy, so they really consume energy, but where do they take this energy from? Because it's a really continuous process. And, by looking at transcriptomic data, you can already ask the question, what are the signature, the machinery that are in place in microglia? And when we run this probing of metabolic pathways, KEGG pathways, we could see that most of them are there in microglia. So you have glycolysis, fatty acids oxidation, OXPHOS, and in a way, this could serve as a transcriptional basis for, in principle, supporting metabolic flexibility, suggesting that microglia are, in a way, ready to metabolize the energetics, the bioenergetics that are available. But this is based on the transcripts, and a few years ago, the lab of Brian McVicar gave a beautiful evidence of functional metabolic flexibility. What you see here is an example of microglia in acute slices that are in presence or absence of glucose. This is really a glycemia situation here. And what you would notice, in a way, it's nothing, because in fact, you cannot even tell that there is aglycemia. The cells are the same, beautifully ramified. They would move and do surveillance. First of all, suggesting that what they are doing is important because they find another way. They will not solely rely on glucose. But then if you come and block, for example, in addition to glucose utilization, also other sources, like here, pharmacologically blockage of glutamine, then you will reveal the problem. Then the cells become small, they stop moving completely. So in a nutshell, what is the message here is that microglia are indeed metabolically flexible, and even if there is not glucose in certain conditions, they could shift to other energetics, like fatty acids or glutamine. And I guess here the point is that they could shift to what is available at that moment. That is very context specific. So we start asking the question, what about other metabolites? What about lactate, for example? And for us, the question about lactate came from the transcriptomic data, because one of the top genes in microglia is LDHB. And LDHB, or LDH1 that Pierre was talking about, is this subunit, LDHB, that together with LDHA form the lactate dehydrogenase complex, which is an enzyme that catalyzes the reversible conversion of lactate into pyruvate and vice versa. But the LDHB subunit preferentially converts lactate into pyruvate. And so we're really thinking what these microglia do with this LDHB enrichment. When we look in the brain specifically, we could even identify microglia by the LDHB expression. If you could see here, these are P12 receptor positive cells in the hippocampus. So really we came with this question, okay, maybe microglia can import lactate and can utilize not only as an energetic substrate, but perhaps also as a functional instructor. And it's no news, in fact, that lactate can be shuttled among cells. We just heard a beautiful talk about the astrocytes neuron lactate shuttle. So astrocytes, we know they can produce lactate, they can feed synapses with these metabolites based on the activity needs, synaptic activity. But so what about microglia? Perhaps they could do the same. In certain situations, maybe they could, directly from astrocytes, we don't know, or in the extracellular space, take lactate and use it for their metabolic energetics, and maybe also for some functional instruction. And now for this functional aspect, I have to say we got very much inspired by the cancer field. There was this paper a few years ago showing that oxidative cancer cells indeed import lactate, and then in the conversion through LDHB, what they do is also, because this reaction produces protons as well, could fuel the VATPSS pump and the lumen of the lysosomes, so promoting acidification of lysosomes. And we know how much for microglia lysosomal organelles is really key, because it's at the basis of the degradative capacity. And so we asked this kind of question. So we said, okay, maybe microglia can import lactate, and perhaps we could see in microglia the same modulation of lysosomal acidification and phagocytic capacity. And then, if this is true also, and this pyruvate is produced, what happened to the disease cycle? What happened to the mitochondrial function? So in a way, we could also ask what are the effects on microglia, but in a way also non-cell autonomous, so in the surrounding tissue. So the first question is, clearly, do microglia import lactate? Which is not a very original question, because it has been shown that many cells can take and then release lactate. But it's true that in microglia, apart from cell lines, we didn't have so much of evidence. So what we did, and Katia Monsorno was a brilliant PhD student at the time, now is a staff scientist in the lab, what she did was to prove that in fact microglia, primary microglia, so ex vivo system, could import lactate when we expose to lactate in the medium. And this is an experiment of mass spec, simply incubating microglia with lactate or sodium chloride controls. And you see that after a few hours, you see almost 4 times fold higher in the microglia. We could complement this data also with fluorescent analog of metabolites, in this case, fluorescent analog from lactate, you see in green, that is even increased its internalization when glucose is depleted. So in a way, supporting this concept of metabolic flexibility, so glucose is less, lactate get imports more. But the final evidence was done with the experiment of labeling, carbon labeling of lactate, where you could see that after a few hours, 98% of the lactate that we found into microglia was the one coming from outside. So I guess here we could not argue that lactate cannot be imported. But the question is, okay, if this is happening, does it get metabolites? The first question was about the lysosome, in fact, and taking advantage of easy dye, like a pH-sensitive dye, like LysoTracker, we could do a cell concentration-dependent assay and see that already after treatment with 3 millimolar of lactate, we appreciate a significant increase in the acidification of the lysosome. And this is interesting because, and we could confirm that it's true, the VATPS is pumped because by using bafilomycin, which is an inhibitor of the subunit, you could completely abolish this effect. And importantly, this is really due to the conversion of lactate into pyruvate, because if you block LDHP or if you just provide pyruvate, in fact, in this experiment, you don't see any effect on the lysosomal acidification. So how does it enter? This was the next question. We know that there are several MCTs. We have heard already about MCT1, 2, and 4 that are all highly expressed in the brain. And in microglia, MCT4 is the most abundant. But what we found was that MCT4 is also the only one that is dynamically regulated, because when we expose microglia to lactate, MCT4 is doubling its amount, not only in the transcript level, as you can see here, but also at the protein level. After 24 hours exposure, the amount of lactate was really increased at the protein level. So this was convincing us that it's a good target, MCT4, as a modulator of lactate metabolism in microglia specifically. We were lucky enough to work with Luc Pellerin at the time, that was in Lausanne, and he has generated many tools and reagents, among those the flox mice for MCT4. So we crossed this with the microglia-specific macroglia macrophages, CX3CR1 Cre line, and you see here we could deplete very efficiently the MCT4 for microglia from microglia only. And the first thing we did was to characterize, to quantify, in fact, the lysosomal response. And you see that while the wild-type cells, once exposed to lactate, acidify their lysosomes, this response was completely lacking in the MCT4 knockout cells. So suggesting that you need MCT4, indeed, to observe the modulation of lactate on the lysosomal function. So we did some functional assays because this was, again, needed to prove that there is an impairment in the phagocytic capacity. And what you see here is the synaptosome phagocytic assay. You simply expose microglia, in this case wild-type or knockout, to a synaptosome isolated from the brain of mice, and fluorescently labeled, so in this case with pH Frodo. And you can appreciate that there was already a decrease in the amount of uptake, and then the total efficiency was also affected, impaired. And then this is the DSBQ assay, which is based on also enzymatic degradation of the substrate, suggesting an impairment in the ability of the microglia lacking MCT4 in processing this cargo. So now we had a situation in which microglia were impaired, were phagocytically impaired, and so we asked what happened in the brain, because you know that, especially in the early development, microglia are relevant for synaptic remodeling. So one of the questions was, okay, what happened to the synapses? And this is hippocampal data specifically, where Katia was screening a bit for different markers, and if anything was changing, was kind of an increase in certain synaptic markers. Here you see synapsin, GLUA2, VGAT. And we could show, in fact, that also by immunofluorescence, synapsin was, for example, significantly increased in the knockout, and this was correlating with an accumulation within the microglia that was kind of consistent with this defective degradative capacity. So in a way, we could describe this impaired synaptic remodeling, impaired synaptic pruning by microglia lacking MCT4. But still, we wanted to prove the functionality of this, because again, so far it's just descriptive and structural. So we team up with the lab of Professor Mameli at the University of Lausanne. They are electrophysiologists, so they could really help us to characterize the properties of those neurons. These are pyramidal neurons in the hippocampus. And what you see is that in the knockout cells, there was an increase in amplitude and frequency in the spontaneous excitatory postsynaptic currents, which was also associated with an increased excitatory inhibitory ratio, so an enhanced AMPA-GABA ratio. So altogether, this data was suggesting an increased excitatory drive, and I would like to remind you that the only modification, the only manipulation is removing MCT4 in microglia. So then we asked, functionally speaking, what is the relevance of this perhaps vulnerability, and we designed an experiment by exposing these mice to a subthreshold dose of an epileptogenic substance, so canic acid. So what you see here is that while, as expected, the wild-type mice were not really reacting to the canic acids. You see the score is very minimal. All the, and you cannot appreciate, but 100% here of the knockout mice were actually displaying epileptic seizures. So suggesting that this altered and impaired remodeling of synapses due to microglia defective MCT4 is in fact providing a vulnerability a susceptibility to increase excitation. So we also did some battery of behavior. I don't have time now to show you, but what we found in the adult mice, there is an increase in anxiety-like behavior specifically. So the last question that we wanted to ask, last of many other actually, but for today, is that if there is conversion through LDHB, what happened to this pyruvate? Is it efficiently metabolized in the mitochondria of this microglia? So first what we did was to go back to this experiment of carbon labeling, and what you see here is that not only all the lactate we found inside was the one labeled, but also the pyruvate was labeled and all the metabolites in the TCA cycle. So suggesting indeed that the lactate not only is imported, but it is functionally converted to pyruvate and fueled into the TCA cycle. So we could appreciate all the metabolites labeled. Then the next question was, okay, but functionally speaking, if we look now at the mitochondria, what is the relevance for the respiration? And Katia, together with Clement, another brilliant PhD in the lab of Nicolas Plas at UNIL, they embark in what I call the adventure of the Ouroboros, because this is the high-resolution respirometry approach that is quite complex, and now they are mastering pretty well this technique. What you see here, just simply, is in red, is the oxygen consumption, because you can take it as a proxy of mitochondrial respiration. And what you measure is the basal respiration here, then you can inject your metabolites of interest, and then you can follow over time how this changed the basal respiration, and then if you come with with drugs, challenges like, for example, FCCP, which is a mitochondrial coupler, you can see the maximal respiration is affected or not. So what they found, first of all, they could confirm that pyruvate, as expected, increased the basal respiration and also the maximal respiration. And they could also describe this for lactate. However, for lactate to behave like this, you need to provide malate. And so now we are still investigating, but looks that the malate aspartate shuttle is required for microglia to use lactate. And the other interesting thing that they found is that this effect on the mitochondrial respiration really depends on the LDHB. Because if you block LDHB through oxamate, which is an inhibitor, or through the MPC carrier, UK1599, you completely abolish this response. So showing that at least for the mitochondrial respiration is the lactate conversion into pyruvate that drives then the downstream effect. But then we keep investigating the mitochondrial function, and this is work that is in progress, and Gloria is leading this at the moment. It was interesting because what Gloria described is that while the mass of mitochondria was not really changing through TOM20 staining, the membrane potential of mitochondria was only increased by lactate. So in this case, it's not anymore a lactate to pyruvate readout, because when instead you give pyruvate, you have almost the opposite effect, and also with inhibitors like oxamate again and aspartate, the malate aspartate shuttle, you block it, this phenotype. So now we are going to a more specific lactate effect that we think is mediated by the redox balance. We can discuss this later if we have time. And the last experiment I would like to show now today is something more into the relevance and the translational effect of this lactate, because we know that peripheral lactate can also reach the brain very easily. And we wonder what is the effect, at least through microglia, what happened on microglia when there is this peak in lactate. So we did a time course. In this case, we were injecting IP, lactate, in pups, P15 animals. And what we see is almost in a way confirming the literature that already 30 minutes after, you have indeed a peak in the brain lactate level. So we choose 1 hour as a time point to sort microglia and to characterize their metabolomic profile. And we did this in collaboration with Angelo D'Alessandro, Colorado, the metabolic platform. So what we see here, and still we are digging into this data because are pretty interesting, but we could already see some alteration in the TCA flux. So microglia that are derived by hippocampi, in this case, exposed to lactate, 1 hour later already have a shift in the succinate to alpha ketoglutarate ratio, the malate to fumarate ratio as well, that are indicating some alteration. Now we are functionally investigating exactly what could be the readout, but in a way they already speak for a disruption in the redox intermediates. For the glutathione system, the ascorbate that is significantly reduced here. So at the moment we are still investigating the function, and for today my conclusion is really that I would like to also to quote Pierre before. He said that it's not all about energetics. So in fact it's true, because one is the component of pyruvate, and this is what we are observing, but there is much more than that. There is the regulation of the lysosome, for example. There is, I don't have time to show you, but we have very interesting data on the regulation of the membrane potential of microglia when we expose those cells to lactate specifically, epigenetic regulation. And what is important is that all of these differential readouts or modulation, they happen at the same time. It's not really one or the other. So it's really a complex regulation of microglial function. So with this, I would like to conclude. I would like to thank all the lab members, excellent scientists and colleagues, in particular Gloria Colombo and Katia Monsorno, who led the experiment that I shared with you today, our collaborators, the funding agency, and all of you for your attention. Thank you.
Thanks for this wonderful talk. Are there any questions from the audience? I can break the ice with a question, okay? So, since we were talking about, in Pierre's talk, about this hypoenergetic condition in the brain, so this is a story that also microglia is using lactate. Are there any evidence of a competition between neurons and microglia cells, like in some situation or conditions in which microglia is particularly activated, can actually take up more lactate than neurons, and this causes some impairment?
This is an excellent point, because it's something that we have been discussing a lot. Already in our knockout mouse model, one of the possibilities, for instance, also is a non-cell autonomous effect, that there is more lactate available if microglia are not taking in. And indeed, we did some proteomics on at basal level, Walter knockout, and we already see more lactate, for example, in the hippocampus of the knockout, but this is wall cell, we cannot discriminate where does it come from. But would point in that direction that perhaps is less used by microglia, and so it's more available. We have to design specific experiments now. I don't have any evidence, but this is one of the possibilities, for sure.
Okay, thank you.
Thank you.
Okay, Professor Morsicano.
Ciao.
Ciao.
Questo effetto del lattato sul...
In Italia.
This effect of lactate on respiration, on mitochondrial respiration. Did you go a little bit inside it? Could be receptor mediated or I don't know.
So receptor mediated, we have tried to detect the receptor, the GPR, but we don't see.
You said it's independent of pyruvate, right?
Yeah.
So it's
the respiration is dependent on the pyruvate conversion.
Okay.
It's the membrane potential that is not dependent on. It's actually opposite to pyruvate. The respiration seems to be. The respiration seems to be really like a easy guess of the lactate that we have converted, and then pyruvate, because when you block even the pyruvate imported in the mitochondria, then you don't see it anymore. So it's another, but the membrane potential is independent of pyruvate, and like also the membrane potential of the cell, which is redox possible, NADH, we believe.
If there are no other questions, I will give the word to Piervito.
Okay, thank you. Moving on. So next speaker will be Maria Teresa Dell'Anno from Fondazione Pisana per la Scienza in Pisa, and she will talk about metabolic trajectories in human neocortical development. So Maria Teresa, the floor is yours.
Thank you, Piervito, for the introduction. I'm delighted to be here today with you and share our most recent data on human neocortical development and metabolism. As you all know, neocortex is the outermost portion of the cerebral cortex. It is a laminated structure made of 6 layers of neurons, and it has been also considered the crowning achievement of evolution. In fact, it is the biological seeds for human-specific cognitive capacities that distinguish us from other primates. Of course, all these complicated functions come with a very high energetic cost. And already in 2011, Herculano Hausel established that although the human brain only makes up about 2% of the total body mass, it consumes at rest 20 to 25% of the daily calories intake. And half of this is used by the neocortex alone. But what do we know exactly about neuroenergetics and neocortical development? Well, in general, we have to say that much of the information that is currently available comes from murine models, so very little is known about human neocortical development. Therefore, before going into details, I will recap very briefly the main events that occur in human corticogenesis. So the neocortex originates from a primordial structure named neural tube, already present at the 4th gestational week, and it is made of a single layer of cells named neuroepithelial stem cells. Neuroepithelial stem cells are endowed with a very high proliferative capacity, but can also differentiate, giving rise to neurons. These neurons will take advantage of the scaffold made of radial glia cells and then will migrate until they reach their final destination in the layered cortex. When neurogenesis is over approximately half gestation, then radial glia cells and progenitors become frankly gliogenic and will form astrocytes, oligodendrocytes, and ependymal cells. Well, the main aims of these study were to examine specifically the metabolism on human neocortical development in two main dimensions, time and space. In order to address the first point about time dimension, we established a longitudinal study from human neural stem cells until they become functionally mature neocortical neurons. And for the second aim, we took advantage of neocortical tissue, human tissue, and cerebrocortical organoids. Well, let's start with the very first aim. So in order to study how the metabolism evolves over time, we took advantage of a neuroepithelial stem cell culture that was prepared from a 6 post-conceptional week neocortex. Cells were kept in vitro without any type of genetic immortalization and were differentiated for up to 120 days. The cells were characterized in vitro. We also confirmed that their karyotype was stable, even though the cells were not immortalized. And also, we characterized the progeny. At this stage, neuroepithelial stem cells are strongly neurogenic, so the vast majority of the cells that we obtain are actually neurons, whereas we obtain little astrocytes and oligodendrocytes, and they also express deep and upper layer neocortical markers. Most importantly, the reason why we had to wait for 4 months in order to obtain a mature progeny is that because at 120 days, we were able to observe ionic currents and action potentials. The first technique that we used to investigate the metabolism of these cells was fluorescence lifetime imaging microscopy, a technique that measures NADPH lifetime. So basically, NADPH as a coenzyme has a very short lifetime when it is free in the cytosol, whereas the lifetime is delayed when it is bound to an enzyme. Two major advantages are present in FLIM technique. The first one is that it relies on an endogenous marker, and the second one is that it is conservative, so it does not destroy the sample, which can be reassayed multiple times over time. Please bear in mind that typically during glycolysis, NADPH is free in the cytosol, whereas it appears to be bound to enzymes during oxidative phosphorylation. It is important to bear this information in mind in order to read the images that you are going to see. So since FLIM is a microscopy technique, when we apply it onto cells, we obtain an image, a grayscale image. Every pixel in the image will correspond to an equation describing the lifetime of NADPH, which can be mathematically transformed so that the image is decomposed in a cloud of dots. This cloud of dots, named phasor, will have a different position in the graph depending on whether there is more abundance of free NADPH or of bound NADPH. Assigning a false color to every dot in this cloud, we can reconstruct a metabolic map, and the color of the map will be indicative of a prevalence of glycolysis or oxidative phosphorylation. This is exactly what we have done on our cells, so you can see here the phasor plot and the metabolic map of human neuroepithelial stem cells derived from a neocortex. If you start a differentiation protocol, you will see that over time, the phasor moves from right to left, thus indicating a shift from short to long NADPH lifetimes. Simultaneously, if you reconstruct a metabolic map, you will see that also the color is changing from light blue to yellow and red, thus also indicating that we are assisting to a switch from glycolysis to oxidative phosphorylation. Glycolysis, in fact, although has a minor energetic efficiency, better supports the anabolic needs of progenitors, whereas neurons obviously mostly rely on oxidative phosphorylation, which better support the energetic needs of synaptic transmission. If you imagine to zoom into a phasor plot and try to condense all this image in one single graph, this is practically what you will see. So as you can see, the major metabolic rearrangement, the major segregation in between phasors occurs within the first month of differentiation, whereas in the remaining time points, although the cells are kept differentiating, there were just minor rearrangement in phlegm measurements. In order to try to dig further in what we saw by phlegm analysis, we decided to couple with mass spectrometry. So we isolated the proteins present in all time points from div 0 to div 120, and interestingly enough, the principal component analysis beautifully matched the data that we obtained by phlegm. In fact, also in this case, you can see that the proteomic landscape was massively changing from the div 0, when nest cells are still under proliferation, compared to the progeny at div 30. At this point, we wanted to go further in our investigation and try to analyze pathway by pathway, enzyme by enzyme. And this is exactly what we have done. Let's start with glycolysis. I will just show some of the pathways that we have investigated. The first thing that we can see from this graph is that glycolysis is not completely abrogated in the progression of differentiation. Please remember that when you look at this graph, the color that you will see appearing below each enzyme is indicative of the abundance of that specific enzyme. So light yellow is for low abundance and deep purple is for high abundance. In fact, you will see that although in the so-called payoff of phase of glycolysis, the abundance of enzyme is decreasing, with the only exception of enolase 2, that is a neuronal specific isoform, other enzymes are actually increasing. The second effect that we see is that Warburg effect is rapidly deactivated, which is basically the conversion of pyruvate into lactate. For what concerns pentose phosphate pathways, enzymes showed a very oscillatory behavior, but the trend overall over time didn't suggest major modifications, meaning that metabolites produced by this pathway are most likely to be necessary for both progenitors and the neuronal progeny. When the Warburg effect is deactivated, then pyruvate goes smoothly into the TCA cycle, and in this case you can see that there is a progressive increase of enzyme abundance over time, and the same can be said also for the abundance of complexes belonging to the electron transport chain, thus indicating that OXPHOS is taking over energy production. We also analyzed the anabolic pathways, such as fatty acid synthesis that is rapidly declined, as well as the cholesterol biosynthesis pathway. So they decline very quickly as soon as we start the differentiation, because as already said previously by other colleagues, typically fatty acids and cholesterol are most likely produced by neighboring astrocytes. We also investigated the glycogen synthesis and degradation pathway, in which we found a dichotomous behavior, because the synthesis was going to decrease rapidly, whereas the catabolism enzymes were being still active, thus indicating that the neuronal progeny was still keeping the machinery in order to degrade glycogen. We found very intriguing the data about this bimodal behavior of enzymes belonging to the glycolytic pathway. Therefore, we conducted a correlation analysis, and we found that enzymes that are in the very first phase of glycolysis, the so-called preparatory phase, correlate in a positive way with proteins expressed in neurons. Conversely, enzymes in the following phases of glycolysis have a negative correlation. Similarly, if we look at a correlation analysis of the same glycolytic enzymes with anabolic pathways, we found a negative correlation of the preparatory phase and a positive correlation with anabolic pathways for the following enzyme. When we translated these correlation graphs into a satellite graph, we found a common hub that is represented again by lactate dehydrogenase, which appears to be the protagonist today in this symposium, meaning that most likely the Warburg effect deactivation is switching off the nest cell maintenance machinery in favor of a neuronal differentiation. Metabolomic studies and fluxomic studies are ongoing as we speak in order to support better these preliminary data obtained by mass spectrometry. Then finally, in order to translate the information that we obtained in time over space, we took advantage of a human fetal brain sample staged at 11 post-conceptional week, kind of mid-neurogenesis, because in this sample we have the coexistence of progenitors in the so-called ventricular zone and post-merotic neurons in the so-called cortical plate, recognizable respectively by BCL11B and SOX2 immunostaining. Also, in this case, we performed a FLIM analysis, which beautifully shows a progressive switch in the depth of the developing neocortex from glycolysis, more abundant in progenitors, compared to the OXPHOS that is way more abundant in the cortical plate. But we were not satisfied with this data, so we decided to utilize the BCL11B staining and the SOX2 staining to perform a laser capture microdissection of these two specific areas and perform, again, mass spectrometry analysis. So by a direct comparison of the data that we collected from a real neocortical sample and the data that we obtained in vitro in our longitudinal system, we found a very nice match of our data. In fact, all the proteins that you see here appearing with the blue dot show a very similar trend in the differentiation. And interestingly, as you can see, the glycolysis also in a real neocortical sample is showing a bimodal expression of the enzyme. That is also validating the strength of our in vitro system of neocortical neuroepithelial stem cells. Although, of course, human neuroepithelial stem cells of fetal derivation are a gold standard example of neocortical progenitors because they were not in any way genetically manipulated, we were also asking ourselves a question. So is our system suitable for cells that are derived from iPSCs in order to open the path also to patient-derived cells? So therefore, we applied a neural induction protocol on human iPSCs, converting them into neuroepithelial stem cells, and then again into neocortical neurons for over 120 days of differentiation. So we applied again the FLIM analysis on these iPSCs-derived progeny, and we could confirm that both iPSCs and neuroepithelial stem cells were sharing a prevalent glycolytic metabolism, whereas as soon as we started the differentiation, we could see the movement, the shift of the phaser, indicating again a progressive switch from glycolysis to oxidative phosphorylation. At this point, again, in an attempt to see whether we could move to more complex models that are currently used to model disease, we also prepared from iPSCs cerebrocortical organoids, which were differentiated for 120 days. This is an example of the organoids that we were obtaining, showing a very sharp segregation of the neuronal progeny that is located within the organoid in the shape of neuronal rosettes and surrounded by the progeny that is positive for the marker RBFOX3. So we decided to perform on living organoids the FLIM analysis. So we sliced the organoid and applied FLIM on a living section. And again, the FLIM map was showing this very sharp segregation of progenitors relying on oxidative phosphorylation compared to organoids, compared to neurons that were mostly relying on on oxidative phosphorylation. And finally, and my conclusion, so we could show in our bidimensional system, in the longitudinal study, that metabolic pathway rearrangement largely anticipated the functional maturation of neocortical neurons. So rearrangements are occurring within the first month, whereas action potential were detectable only at 120 days. We also found that, to our surprise, that glycolysis was not abrogated entirely or in a progressive way, but we found that some of the enzymes in the glycolysis, and especially the enzymes in the very first part of the pathway, also support neuronal differentiation. Our metabolic rearrangements found in vitro were also confirmed in a real human neocortical sample that was staged mid-neurogenesis, and also we provided proof that FLIM appears to be suitable for the generation of the metabolic map derived from in iPSCs derived cerebrocortical organoids. And with this, I wish to express my acknowledgement to the institution I belong to, Fondazione Pisana per la Scienza Onlus, and to the Tuscany Health ecosystem. I wish to express my deepest gratitude to all the people that allowed us to realize this project, and also to our collaborators, both at the University of Pisa and at Scuola Normale Superiora. If you have also more technical and detailed questions, please see Gianmarco Ferri at the poster session today in the afternoon. Thank you again for the attention.
Thank you, Maria Teresa. We got some time for a couple of questions. Any questions from the audience? Okay. So a curiosity from my side. So which is the space resolution of your FLIM? So which is the real resolution?
Okay, so you can definitely have a single cell resolution also within tissue, and you can, depending on the microscopy technique that you applied it to, you can also discriminate organelles within the cells. So yes, definitely it allows you to go quite deeper within the cells.
So it's like a system that can be suitable for other cells type, right?
Absolutely, absolutely. And probably the most interesting thing about this technique is that it provides you a measurement without killing the cells, because it's conservative. So you have a real measurement of what is happening, even when you, for instance, treat the cells with a drug or any type of metabolite.
Thank you. Are there any questions from the audience? Okay, I think we can move on.
I can ask a question.
Oh, yes, absolutely.
Okay. Thanks for the great data that you showed. So it's just a curiosity. In neurodevelopmental disorders, are there any evidence that during the corticogenesis there are alterations in this metabolic switch that you observed?
Yes, absolutely. So for instance, typically the switch from glycolysis to oxidative phosphorylation is necessary for neuronal maturation. So if this switch does not occur, obviously the cells cannot maturate. So you may have, for instance, the occurrence of conditions such as microcephaly, because of course you don't have enough maturations of the progenitors. But there are many other conditions where if the glycolysis of OXPHOS is blocked, you can have obviously an impairment in neurodevelopment.
Thank you.
Thank you, Maria Teresa, for this fantastic talk. Okay, we can move on with the last speaker, Enrico Luchinat from University of Florence. He will talk about cell phenotype classification by NMR spectroscopy of intact cell lines. Please, Enrico.
Thank you, Piervito, and thank you also to Paola for organizing this symposium and inviting me, giving the chance to talk to this audience, which is very different from my usual audiences, so I'm quite excited, by the way. This is now, I'm definitely switching gears now. Let's start from the beginning. I'm a chemist. I work in Florence, where we have a very big facility of NMR spectroscopy. So what is NMR? Now, I guess most of you are familiar with magnetic resonance imaging, which is a technique which is actually the most important technique to investigate the brain in vivo, and it has a lot of clinical relevance. So NMR spectroscopy is just the same thing, but instead of taking an image by exploiting the signal that comes from the nuclei of atoms, nuclear spins, we separate them in frequency. So what we get instead of an image of the sample is a spectrum. It works, I mean, so the physical principle is exactly the same. However, this is I guess, one of the most important techniques in chemistry, and also very important in structural biology, because with this technique we can investigate the chemical properties of matter and the structural properties of molecules, including biomolecules and macromolecules. So it's one of the ways you can actually obtain structures of proteins. I will not talk about this, however, today. It has also many other applications, including the analysis of complex mixtures with thousands of different molecules. Because every single molecule, every nucleus inside the molecule can resonate at a different frequency. That is why it's so powerful. Also, it is non-destructive, which means that we can apply it to samples without destroying them, and we can apply it to intact cells. Now, this is a bit of background of what I do usually. So among these applications, we can use the set of techniques of NMR, biomolecular NMR tools by working with high field spectrometers to study macromolecules instead of purifying them, study them inside living intact cells. And we have been specializing ourselves in Florence to study proteins inside human cells. So what we do usually is exploiting some cell line, which is actually HEC293 T cells, which are very good for protein expression, overexpress the protein of interest, label this protein with isotopes of nuclei, so in a way that we can only look at that molecule, and then obtain information about the structure, conformation, and interactions inside the cells. So we can study these proteins as they do their work in the cells and observe the conformational changes due to protein folding, maturation, aggregation processes, interactions, redox state changes, and also the binding of small molecules to these macromolecules. Then there is a very different kind of application, which is not my main field, which is metabolomics. Because NMR at high resolution can separate signals in the frequency of thousands of different molecules, NMR, just single monodimensional proton NMR, so where you see hydrogens of all organic molecules, can be used to analyze mixtures in solution, like biofluids of any kind, urine, serum, so from human origin, but also cell extracts from cultured cells, where we can see the metabolites of the cells, or even food extracts. This is done on milk, on coffee, or anything you can think of. If you can get a solution of molecules, you can do NMR metabolomics. This is done in two conceptually different ways. Maybe something which could be more familiar to you is so-called metabolic profiling, where we are actually looking at specific signal from molecules, from certain molecules. And once we identify these molecules, we're sure we can measure the intensity of signals and get information on and quantify these metabolites, and then make a statistical analysis like a univariate analysis. Now I got the point. This is the univariate analysis. Now, another approach, which I think is very powerful, and it will be the focus of my talk today, is the so-called fingerprinting. In this approach, this is an untargeted analysis approach, where we are not asking who is who in the spectrum. We don't care. As long as the spectra from different samples are different, then we can do statistical analysis. There is some data processing involved to simplify the analysis, but you can just take the raw spectrum and take every single intensity, every single peak, every single point in the spectrum as a variable, and then do multivariate analysis on this multidimensional set of spectra, each of which is made of so many variables. Now, because when you do it in vitro, so in vitro on clear solutions, this works really well because you can separate so many different molecules. But can we do it in intact cells or tissues? Now, the simple answer is no, at least not like this, not in this way, because we lose the high resolution. Even though, some of you may know, if we go to higher magnetic fields, you improve the resolution of NMR spectroscopy. This is true for solutions of molecules dissolved in water or organic solvents. But this is not true anymore if you put intact cells or tissues in the NMR spectrometer, because the tissue itself or the cell pellet itself will cause inhomogeneities in the magnetic field. That in turn leads to line broadening, which is not due to the intrinsic relaxation of the nuclei. That's a different kind of line broadening. You also have that. But this line broadening comes from the simple fact that we have cells in the tube. So the magnetic field strength is different in different points of the sample at microscopic level. Therefore, instead of obtaining such a nice, highly resolved spectra, this is a cell extract, we get these broad shapes in the spectrum. And even if we go to higher fields, we'll still get these broad shapes. But the question remains, are these shapes revealing something about the content, the molecular content of the sample? And the short answer is yes. But no one has ever tried to actually make a sense of these broad shapes. And an interesting point, which is the beginning of this journey, is that the empirical observation that the spectral resolution we get, even at very high fields, I'm talking about 20 Tesla and more, these shapes are very similar to the spectra obtained with MRSI, so magnetic resonance spectroscopic imaging, where you take voxels of brains and then you take the actual proton spectra from each voxel. And you get much noisier result, but the shapes are overall very similar. And so Professor Luca Massacesi, who's a neurology professor in Florence, came to Lucia and myself and asked us, can you tell if I give you different kind of cells? Can you tell me from the spectrum which cell line it is, which kind of cell it is? And the short answer was no, absolutely, we cannot do it. But then thinking, we started observing the fact that different samples of cells, even from the same cell line cultured in different ways for different days, they were slightly different. So hidden inside these broad, very highly overlapped shapes in the spectrum, there are the metabolic content and also macromolecules. So is there a way to extract this information from those poorly resolved NMR spectra? And if the answer is yes, then that would mean that it should be possible to discriminate the cell phenotype based on the spectral features. And this would have quite important implications, I think, in the analysis of brains in vivo with MRI, actually with MRSI, because possibly with the next generation instruments, with higher fields, we should get enough signal and enough quality of spectra to really see differences in different voxels, and this would be a way to extract information out of them. So this is the workflow of the analysis. We started growing different kinds of cells, and this is where we needed also help from people here in Pisa, from Marco Onorati and Claudia Dell'Amico, who gave us, they made a tremendous effort to grow neural progenitor cells and then differentiate them and make neurons and also glial cells. Then we ourselves culture our HEX cells, which are very fast to grow, so they're easier, we can make a lot of them, and also some HeLa cells and URCAT cells, because they're also easy to grow. So they represent different kind of phenotypes. And each of these samples was analyzed by NMR spectroscopy at high field, so we could ensure high signal-to-noise ratio, but as I said, not very high spectral resolution. So we collected this data set, and then we started to play with different kind of analysis by using machine learning approaches. And here we collaborated with Carlo Mengucci in the University of Bologna. They do metabolomics, so he was really expert in this kind of analysis. And this is how we basically grow the cells. You're very familiar with this. So incubator, 37 degrees, then they reach confluency. We can grow, if we can grow enough cells to fill a glass tube which is needed for NMR analysis at high field, then it's possible to obtain a spectrum. This is done by detaching the cells, washing them, and then we chose an inorganic buffer, phosphate buffer, saline, because there are no additional signals from the buffer, no organic molecules. So the only signals we get are from the cells. And then we transfer them to a glass tube. I will have a picture later on. Make a cell suspension, but these cells would just naturally go to the bottom. So by spinning the tube, we can get a nice pellet here, which is our mimic of a tissue. And this is placed in the instrument, and we obtain a spectrum. This is how the spectrum looks like. Not so different from some of these voxels in MRSI at high field, at like 7 Tesla, even though the field is very different. And these are the thousands different molecules hidden behind these broad lines. We did this for about 160 different samples from 4 types of cells. And then we also kept going and made some additional samples, more details in a minute. We mixed together 2 different cell lines in the same tube and see what would happen with the classification. And then with Marco Onorati and Claudio Dell'Amico, analyzed some neural progenitor cells, neurons, and astrocytes. And then finally, we also changed the field and went to lower fields and saw how the spectra would change by changing field. And these are our results. So these are just the raw spectra. This is just hex cells. You can already tell that they are not identical. Some peaks change quite a lot in intensity. And then if we keep going, Urcat cells, neural progenitor cells, they all have different fingerprints. Now the challenge here is to separate these samples, but not just by the difference in the spectra, but by the interclass differences. To do this, and these are HeLa cells again. So what we did with Carlo Mengucci was develop a classifier of cell types. He tried a lot of different multivariate analysis approaches, machine learning algorithms like PCA, PLSDA, and long story short, we found that PLSDA, partial least square discriminant analysis, which is a way to discriminate samples where we know the classes of samples. So it's the class is one of the variables we give. This was working quite well to discriminate these 4 different cell types. And then scalar vector machine was used to cluster somehow, to find the borders of these 4 clusters in the final plot, which is this one, and use these borders to classify additional samples, test samples. So here you see each dot, each round dot is a sample of the train sample, and each triangle is a sample of test. So if the triangles fall within the correct color regions, then it means that it works. And the F1 score, which is the mean of precision and recall, was quite high, was close to 1. So the performance is quite good. Once you have a good training set, then you can test it and get a good performance. Then the additional challenge was cell mixtures. Now, here we found that PLSDA was actually working awfully, really bad. Because if you train the PLSDA on single cell types, then the mixtures will fall between these borders. The triangles are the cell mixtures. And then there is no way to tell from which of these 2 cell lines the mixture comes from. So this is a confusion matrix where the expected outcome is here, and the real outcome is the actual sample is here, and these numbers should be all on the diagonal, and as you can see, they are not on the diagonal, so it's really bad. So then we switched to a completely different algorithm, which is a Boruta. It's a feature selection algorithm. It works in a different way, and then it also uses a random forest classifier for fine-tuning. So this is a way to select specific spectral features and then just use those, clean the samples in a way, and then use the relevant features with a random forest classifier. And here there is no plot, so you have to trust me, but the performance is much, much better. So now 23 were correctly assigned and 6 were not. So we have a much better performance. Now, this is relevant because in the human brain, you have a lot of different cell types, all mixed together in a single voxel. So it's good to know that you can sort of separate them if it's a mixture. Then here is the classification of neuronal cell types. So we tested early neurons along the differentiation pathway from the neural progenitor cells, together with the progenitors, late neurons, and then iPSC-derived astrocyte progenitors and mature astrocytes. These samples, they had a technical challenge because now the sample size is much, much smaller. We had 1 or 2 million cells per sample, and they took months to prepare, I mean, several weeks. So we needed a way to get a decent spectrum out of these, and we managed by switching from normal 3 millimeter tubes that we use for bigger pellets of cells to capillaries of 0.4 millimeters of external diameter. So we took a while to figure a way to fill these capillaries with a pellet of cells, but then we succeeded. And the spectra look as nice as the others. So we were quite satisfied with the spectral quality. And so again, for each category, you have different spectra. And if they look different when you look at them, it's a good sign that you will be able to classify them correctly. This is again the PLSDA analysis, which shows a quite clear separation between the progenitor cells, early neurons, and the late neurons, and also the astrocyte progenitors and mature astrocytes. They are on a totally different path. So one can imagine like the differentiation paths diverging from going up and then down in this plot of abstract dimensions, but this is enough to distinguish them, to separate them. And then finally, what I think is maybe most important, can we switch to lower fields? Because MRI instruments don't have such high field as we do have. And the answer is yes. Now, these are same samples collected at 950, 700, 400 megahertz, and they basically look the same because of the inhomogeneous line broadening. So now, by using a training set at high field, 950, and then testing samples at lower fields, we actually got similar F1 scores. That means we can almost approach the 7 Tesla, 300 megahertz spectrometers. In principle, we should be able to do the same analysis at such lower fields. So this is a very preliminary work, I think, because it's very far away from a real brain. But we can somehow decode the broad spectral features and use machine learning to assign single cell types, classify them, and also mixtures of 2 different cell types. We can also see the differentiation of CNS relevant cell types, and we can also transfer it to lower fields. So the hope is that in the future, this would lead to more advanced ways to analyze brains in vivo with MRSI. And with this, these are all the people involved in this work. So I will leave it with this slide, and thank you all for your attention.
Thank you, Enrico, for the great talk. Is there any question from the audience? Okay, I have a question. Actually, some comments. So, do you say that there is some kind of potential application in neuroimaging field? But at the same time, you said that you need collection tubes to ensure geometry of the cells. So, how can you deal with that in neuroimaging?
That's a very good point. And the naive objective at the beginning was to build a database of every single spectra of every single cell line from the brain, which is very, very challenging. Because also, what does it? I mean, I would not be sure that the actual shape of the spectrum would be the same, because in a living brain, the metabolites would be different. So in the end, I think the training set, a real training set, would have to be done with a high-field MRI instrument on an actual brain, or maybe with biopsies. That's why I say we are at the first stage. Yeah. We have the idea. We can use these statistical analysis, machine learning methods to tell apart features which are otherwise very broad. But then the training set is probably not the one you would use in the end, yes.
Okay. Thank you. Thank you, Enrico.
Thank you.
Okay, we are closing the session. Thank you, everybody. It's been a great talk from everyone. So please enjoy the lunch break and see you later.
Bye, Pierre, if you're still with us. And thanks for your talk.
Sì, sì.
the adapter or shall I bring one?
Vittorio ha piegato, c'era un cavo accademico.
Ok.
Allora, di parlare mo, no? Di parlare dopo.
Sì, però okay, I bring my adapter.
Adapter?
Yeah,
Yes.
I have one. Shall I-
Sì, va bene. Qui tu lo metti. Questa è la terza. No, non abbiamo fatti questo. Però abbasso. Ok. Che stanno così piegati che si. Ok. Quindi. Oh. Quando lo metti in amici? Ma chi è che voglio vedere se come si legge per vedere la risoluzione. Ma questa qua che devo parlare chi è? Non lo so. Ma so che tu. Guglielmina? Guglielmina.
Yeah.
Ok. Oh. No, non è questo. Attenzione, attenzione, attenzione. Facciamo un attimo, vedere se si vede di là. Piano. Lo vedi? Sì, 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 sì. Ora?
Yeah. Okay.
Ok.
Wait. Okay. The lights will be down. Las luces las podemos apagar. Ok. Que es todo en negro. Gracias.
Prego.
Sí. Sí.
Quando parlerà lei vuole le luci abbassate.
Sí. Esta.
Ok.
Porque es.
Ce lo dobbiamo avvisare a quelli del... Immaginate quando parli. Qua, che cosa è questa? Non capi quando parli. Adesso no. Il nome, l'hai visto il nome, scusa? Guglielmino. Questa qua. Ok, Guglielmino.
Magari quando io dico good afternoon mi proietta. Ma lei può anche non proiettarmi, non è che sono spettacolo io. Allora ci metto un po' di adob, un po' di sfumature carine, cioè, bisogna fare anche quello. Queste luci sono fantastiche, una miseria. No, vabbè, ma questo, ne state regalando a tutti, ecco perché vedevo tutti quanti questo. Adesso mi sono davanti una risposta, grazie. Andrea, mi dite voi quando posso iniziare? Perché
perché Monica mi ha detto aspetta 5 minuti. Quindi io aspetto un segno. Ok, va bene. Ah, Monica è lì, no? Sì, tranquilla. Good afternoon, everyone, and welcome to this special lecture. Before introducing our speaker, I really want to highlight the novelty of this initiative. Our president, Monica Di Luca, and all the committee of the society, Italian Society on Neuroscience, strongly wanted this new activity in our society and in our conference, giving them a meaning, a special meaning, also the importance of translate our research, our basic research into the clinic. So having a clinician, a lecture about the clinical aspect means a lot for our society. And in this way, in this key lecture, I want to introduce Prof. Andrea Di Bartolomeis. And it's really a great pleasure and a genuine honor for me to introduce Professor Andrea Di Bartolomeis. He is a medical doctor with a specialization in psychiatry, and let me use my glasses. And so he's a full professor in my department. That's why I'm also a little bit emotional in this point, because I know Andrea since I was just a PhD, and I was discussing before with another colleague about how much he appeared to us like a genius at that time when we were just students, and he was already a researcher with a large experience in psychiatry. He always worked, focused on schizophrenia, and of course during the year, I'm not a PhD anymore, so he's a lot more than a researcher now. And then, he learned how to approach the problem from different point of view, in particular focusing on the drug-resistant forms of schizophrenia. Andrea spent a lot of time, let me say better, he spent about 10 years in USA, in Maryland in particular, and he had the privilege and the fortune to collaborate in an important discovery. So he actually was part of the team that discovered how much the dopamine release in schizophrenia plays an important role in the genesis of the pathology, and actually also in the treatment, because this discovery provided
provided us the possibility to approach the pathology differently, and it did by using PET in humans. And PET was a pretty new procedure at that time because it was not so much used. And so this is the real breakthrough that Andrea did in the past. And when he came back to Italy, I was a PhD student. That's why for me it was just a genial, enthusiastic, creative, and he always was funny and open to collaboration with our department, because actually we are in the same department. So I was saying before that he's a medical doctor. He received the residency in psychiatry and the PhD in methodology and communication science. He's also a member of the UNESCO chair for health education and sustainable development, still in Naples. And of course, he published a lot of articles. We are approximately around 200, and we received a lot of citations. We are about 10,000, maybe even more. And so really, please join me in extending a warm and sincere welcome to Prof. Andrea De Bertolomeis.
Good afternoon to everyone. Thank you, Professor Matrone, for the kind introduction. Thank you all the committee, especially Dr. De Luca, Professor De Luca, for inviting me to great my honor. So this presentation will deal basically with the mechanism as well as the pathway that leads to response or no response to antipsychotics. Just let me give you a very wide paramount of the situation. If you ask around how many people are affected by Alzheimer, everyone will tell you something that more or less close to the reality. It's about 26 million, just speaking about Alzheimer, not all neurodegenerative disorder. However, if you ask how many people are affected by schizophrenia, probably very few will tell you that the number is very close to the one of Alzheimer's disease, 24 million. And we're speaking about very young people with a chronic disorder that they will basically deal with treatment with antipsychotics for possibly all their life. And this is the first time it's also making one of the greatest, unfortunately greatest experiment that human being has never done on human brain, from the appearance of the monoamine system that's much older than human being. Monoamine system in invertebrate species appears almost at Cambrian age. That means 500 million years ago. And when manipulating dopaminergic system, it's kind of different what's happened with other psychotropic drugs, especially because with antipsychotics, there is a lot of chance, unfortunately, that people will get the treatment for all their life. So say that, this is the outline which I will try to move from without the pathophysiology that in some way represent the rationale to use antipsychotics, and then I will move to some imaging study that probably can say something why people do not respond to treatment. These people with hallucinations, people with delusions, so it's a quite strong situation. And I will try also to give some flower of which can be a way to model with all the limitation of the modeling of animal for a psychiatric disorder. We don't believe that there's a schizophrenia in animal. I've never heard about a rat that tells me they has a hallucination. However, I think that we can model some small stuff and get from some information. And then I will try finally to give you some news regarding different possibility to explore synaptic plasticity in psychosis as well as to modulate some basic mechanisms that probably are related to the response or not response to antipsychotics. Let's say the general framework will be very clinically oriented. We should try to do things that are meaningful for the people. Otherwise, we go in with beautiful, very elegant science, but then nothing happens with our patients. So I will speak about specifically with treatment-resistant schizophrenia. That is a significant story because think about it, reflects 1/3 of all patients that suffer of this disorder, 30%. And I will try also to show this possibility to capture macro and micro scale of the mechanisms that are behind the use of antipsychotics. And finally, thanks to all the young people working in the lab, I have no responsibility for anything. Now there are young people doing much more than what I do, and I'm grateful for them. What will have been envisioned as decay function radiomics. As you know, radiomics is basically, especially for connectivity, which you speak about on structure and functional. Things about MRI, things about fMRI. That's a real structure you can deal with. In the case of PET scan and in animal model autoradiography, we do not have a real structure. Think about that, just to have an example, what happens with Google Map. In Google Map, you have basically a computational system that applies what is latitude and longitude. If you move in astronomy you have what we call right ascension and declination. It's exactly the same thing then, but then it's the stars. But when you go up, you don't see the earth, you don't see the ground, you see just lights. And if you will make connections, become much more difficult because it's just light, there is no structure. But it is this exact what have done astronomers for years, and this is the reason for which we have constellation. They just did before us this kind of interesting story. Okay, say that, this is my disclosure, but I will not speak anything that relate to this nowadays. And let's go with the first part. It is the landmark of use of antipsychotics. As you know better than I know, there is no really any biomarker in psychiatry. Whatever you do in psychiatry in terms of diagnosis is clinics. So forget what every day I will see on newspaper, sometimes scientific newspaper, we have the gene for schizophrenia, we have the gene for depression. First of all, these are polygenic disorder. We have no gene, we have genes. In the most cases, it are also, besides the polygenics, they are also multifactorial, so the context, the environment makes a lot in the onset of the disorder. But there is one landmark that, thank you, Professor Matrone, for mentioning it. I have no very merit for this. I was very lucky, I was very young to be on the board of this story. And the landmark is that almost, not all, almost large part of schizophrenia patients have a condition of hyperdopaminergia in the brain. And this can be, in a living situation, demonstrate giving amphetamine or other stimulants and showing that schizophrenia patients get much higher release of dopamine and striato compared with the normal controls. This is an experiment that's been demonstrated in different kind of samples in patients under treatment, in naive patients to antipsychotics, different gender, a different duration of disease. Obviously, when we speak about the antipsychotics, everyone knows that these drugs, mainly, despite the fact they have a very complex profile, if they do not occupy dopamine D2 receptor, they have no antipsychotic effect. Now there are coming some exceptions very recently. By the way, one of the person that was the first author of the paper they are showing there regarding the increase of release of dopamine under amphetamine is exactly the same person that after 30 years, few months ago, was able to release the new drug xanomeline that's probably not completely the only non-dopaminergic drug that's never been released for schizophrenia treatment. It's Alan Breyer. So let's go, just think that when we speak about dopamine occupancies, things are not so clear, that things are not so simple. We have different kind of dopamine receptors in terms of function. We have high affinity, low affinity, and for many years we were dealing with D3 receptor as D2, D3. And the D3 receptor has been a kind of theoretical possibility. Now we know that's probably not true anymore because we have what we call preferentially acting at D3 receptor drugs such as cariprazine and other drugs are coming on armamentarium for the therapy. Just last things regarding the possibility that really there is a rational background for the use of antipsychotics, the fact that despite being demonstrated for schizophrenia, the increase of dopamine in striatum makes sense that you give a drug that reduces dopamine function. This is not probably something that relates only to schizophrenia, but relates also to delusional disorder that's very difficult to treat, and also affective psychosis. This is, in other words, seems to be a landmark that is more appropriate to consider properly of all psychosis. Now, say that, just I would like to remind, there's a lot of young people it is very, very nice, that we are also used to think that action of antipsychotics is based on the occupancy of the dopamine receptors. It's absolutely true, but other mechanisms have been going through in the last few years. One of these mechanisms is the one that we have been discussing this with Professor Correlli, other people here like in the audience, just Professor Barone, the possibility that this drug, usually, especially two of them, that's clozapine and aripiprazole, can work differently by trapping as a chaperone the dopamine receptor inside the cytoplasm. So the receptor does not navigate through the surface, and therefore what happens is that basically you have less receptor available for the dopamine. This can be one way. The other consideration should be done, that is the concept the pharmacologists know much better than I know, that the K-on, K-off, that's been for many years the one has been separated, the so-called typical and atypical antipsychotics. I would just remind that typical atypicality has nothing to do with biology, has to do only with the clinics and with the appearance or not of atypisk situation, that is basically the appearance of extrapyramidal disorders. But now we know that probably the drugs that recycle many times on the surface of the cell, in other words, have a high lipophilicity, can stand on the receptor higher. So that means that probably what can separate typical from atypical is also some kind of moiety physical stuff. And finally, the observation done by a group of German-Italian researchers, the Italian researchers now work in Cincinnati, it is Davide Amato. We just speaking yesterday about this story, and it was interesting. They have been showing that antipsychotics can be trapped in the vesicle that include the neurotransmitter. Exactly. This is very strange, but people have been demonstrated with macrodialysis that antipsychotic can be contained inside the vesicle that contains dopamine, and therefore can be released with the dopamine, and when it's released, it's going to act on sodium channel and therefore switch off the release of dopamine. This is an alternative mechanism. So now, despite all these nice things, 1/3 of the patients affected by schizophrenia do not respond to the treatment. That means that we have a lot of young people around that have delusion, that have hallucination, they have different treatment with 2 different antipsychotics, nothing happened, even if it's been done for several times. And what this means? That means that these people probably need to be treated differently. Now, after almost 60 years, we continue to have only one compound that can be not always, not all the time, but many times significantly different from the other one and take care of these young kids. And this compound is clozapine. And now it's a drug that doesn't cost nothing, it's very few dollars, and it's one of the 100 drugs that have been listed by World Health Organization as the essential drug, together with a few other psychotropics. Now, this is a guideline for which we're interested that we made a few years ago, and I believe still the only guideline available that gives you the criteria to define patients affected by schizophrenia with the condition. By the way, the treatment resistant is not a diagnosis, it's a condition. And unfortunately, despite having guidelines, what happens that we need almost 5-6 years to treat properly these patients. That means that we introduce clozapine sometimes when it's too late. Now, if this treatment-resistant condition is so different, it's because probably we do not have in this condition the increase of dopamine that we observe in all other forms of schizophrenia. And this makes sense, because if you have no increase of dopamine, there is no sense that you expect that the drug that works against the dopamine function could be effective. And probably, but not completely, because the studies are not well balanced and not always get in the same direction. The magic box is, the Schwidbehopen is the one of the glutamatergic system. Who is in neurology and basic science, he knows very well. Many years ago, Professor Olney in St. Louis University made a famous hypothesis that's been carried out by other people over the time. We did some job or two. And what is interesting is that basically glutamate drives the hypodopaminergia, but probably in case of treatment-resistant schizophrenia, this mechanism is not the usual one. And this is the reason for which if you use drugs that work only on dopamine, unfortunately they are useless. Okay, this is a very bad condition. I just want to show you one clinical study that many that we have done. People working in the lab, in the clinics have done. I really am thankful to all researchers. And this one that showed you a few things. First, that with machine learning, everyone does machine learning now. You have seen a study in the last 20 years that does no machine learning? We do machine learning too. So say that one interesting thing is that we can do some prediction, but what is interesting that there are very clear clinical statements that can be made. First is people mainly are driven by the dissociation factor. These people have very peculiar characteristics in terms of clinics. They have high level of what we call negative symptoms. That means flatness of the affectivity and social withdrawal and avolition. And on the top, the most important things, Professor Iasevoli has very well demonstrated this in a clinical study a few years ago, that basically these people function very badly. Among all the patients, if you catch patients with schizophrenia, patients with bipolar disorder, and patients with treatment-resistant schizophrenia, these one are unfortunately the worst performing. So now, what we did, we tried to do something. It was possible to tear apart and make stratification of patients that respond from patients who do not respond to antipsychotics. To do this, we did something relatively easy or relatively simple in idea, was extremely complicated because just remember that these are patients many times, despite being compensated, have been strongly hallucinated and strong delusional, and on the top, they should lie in a PET scan suite. That's not the easy thing you want to do. I've done many PET scans by myself because I've been twice cancer, and I tell you, it's disaster being there. So I understand when people don't want to do, but they were so graceful to us to do this, and therefore what has happened is this, that we were able to reproduce some results done before with lower number of patients. We have here 70 patients that divide in 3 groups. One group is represented by people who respond to antipsychotics. Another group is people that are treatment resistant. The third group that's normal control. Now, to make short, not so much complicated, but long story, this was happened, that we were able to separate people from control condition to the one affected by the disorder, so overall all schizophrenia patients. But what's most interesting was the fact that we were able to separate by PET scan the people who respond with the people who do not respond to antipsychotics. And so very well, what happened is this, there are 2 points that were very specific, and this was that superior frontal gyrus that basically was showing hypometabolism in patients with treatment resistant, and fusiform gyri that was on the opposite condition, showing hypermetabolism. I just will remind you, I should remind myself, because let's be honest, we didn't know so much about the fusiform gyri when we started watching this data. And the fusiform gyrus, it's quite interesting in schizophrenia because, as you know, it's basically correlated to the capacity of human being to recognize human face and especially recognize emotion. And one thing that's been always demonstrated, I would say, very well replicated in neuropsychological study of schizophrenia is that people with this disorder have very difficulty in recognize the emotion of another people, what we call theory of mind, TOM. So that makes some sense, and at one point something more complicated came out. We thought, why we do not try to understand if this has any relationship with the neurotransmission, with the fact that these people probably not having hyperdopaminergia have something different that is related to the glutamatergic system. And as I know, I'm making very short, complex story. Professor Magistretti, that is here, was at the Congress, has been one of the principal leading scientists behind this theory and this scientific commitment. There is a possibility that when we use FDG, that stands for further glucose, obviously, and we use to measure glucose in the brain of these people, it's probably over a statement say measure glucose, but just to be direct, probably we're doing something that's also to do with the glutamate. It seems to be a stoichiometric ratio between 1:1 between these two moiety. Now, I think there's a lot of drawbacks, and these have been discussed, but there is one thing that's interesting, that clozapine may induce widespread cortical glucose hypometabolism, altering the GLUT1 transporter expression. I should say that with us this evening is one of the scientists, the first shown on molecular psychiatry, Fiorenzo Conti, that clozapine could affect glutamate transport. Is correct? Okay, so let's go ahead. And so what we decided, let's do, could we do some connectivity, see this spreading effect on antipsychotics beyond the dopamine occupancy that can be tracked down with graph analysis? Again, I was telling before, the graph analysis has been applied everywhere in MRI, functional MRI, but I think that very few studies, never in schizophrenia. There is some study from San Raffaele group in Alzheimer's disease, but not in schizophrenia. And so, thanks to the great application of Giuseppe De Simone, with that, he had a very beautiful presentation, like all other researchers in the lab, in the clinics. So we decided to do connectivity in PET study and on PET images. So this is the very complex way in which we decide to go for the first statistical comparison to permutation testing and then applying machine learning. I just showed you what's going to be the results of, again, comparison between TRS patients, that means treatment-resistant schizophrenia, and patients who respond to antipsychotics. Okay, if we look for what we call global strength of the connectivity, unfortunately, we are absolutely not able to separate and differentiate these two categories of patients, the one who responds, the one who not responds. However, if we go to consider specifically other kind of measurement that put together all the patients with schizophrenia, independently which is the response, we are able to separate completely schizophrenia patients from control based on connectivity on PET scan, not just a structural one. And the region are most affected by connectivity loss in patients, all schizophrenic patients, the frontal regions, the DMN, the default mode network, and the dorsal dopamine pathway. Giuseppe was a very good insight to tracking on, again, which is not a real image, it's just a spot on a PET scan, what could be the reflective of the dopaminergic system. Now this can be done also with the Halser atlas or with Halser methodology, but at that time we just were looking for what was available. Okay, but then again, if we go on the nodes degree, weight degree, that means to try found difference between some of the connection strengths linked to a specific node of the connectivity network. Again, we don't find difference between TRS and not TRS, okay? We find difference between controlled schizophrenia. But then, if we look exactly for very specific comparison, in this case with per age comparison, the difference appears and emerged in frontotemporal hippocampal connectivity as well between the cingulate cortex and temporal and frontal areas. Now, this is makes quite sense. Why? Because we know that TRS patients do not perform well under the cognitive point of view. And if you will resist till the end of this talk, I will show something that has to do related with specifically to the cognitivity, again, done with the PET scan. Now, let's make things a little bit more faster and say that also applying machine learning was possible to separate these patients, especially on the basis of the frontal regions, temporal, cingulate gyrus again, and the hippocampus. I would just say that cingulate gyrus has been around a long time to discriminate patients affected by treatment resistance compared to the one that respond to antipsychotics. And this probably was a kind of, not surprise, but was for us surprising, the fact that machine learning was, at least in part, doing prediction and was working in the right direction. So say that there's a lot of limitations, there are some strengths. I leave you this, and I'll just tell you that we have a relatively good number of patients considered PET scan study. I would say that in the intersubject network maybe is one of the major limitations, because maybe prevent inference about hierarchical or causal relationship. And also there is another reason for which we believe there is a big limitation in this study, and I'll show you later on. And this one was a study that was magically performed by Marta Matrone. Why? Because she did everything during the COVID and did everything with young kids. Why do we were looking for young kids? Now, one of the things of the previous experiment seems to be relatively well designed in terms of sample, because we have normal control, we have people with schizophrenia who respond to the drug, and people with schizophrenia who are resistant to the treatment, but there is something that is lacking, is missed, and this. All these people were being treated for many years with antipsychotics, so what we observe, it's very difficult to understand is what's come from the disease, what comes from the treatment, whatever. On the other side, this is a kind of 22 catch problem. Whatever you put the situation, it's complicated. Why? Because if you should consider treatment-resistant patients, you can consider treatment-resistant patients only the ones that have been already through antipsychotic treatment. So I thought, what could be a solution? And with Marta, we came out something that was interesting, we never thought before. Why do not look exactly what happened in the brain of very young people that have been diagnosed with treatment resistant, and the problem for the very low age, they have been receiving treatment for relatively short time. And so what we did, Marta did, and we have patients with early onset treatment resistant, patients with late onset treatment resistant, obviously we have health control. And the interesting things for this that when we look at the glutamate levels of glutamate creatinine, we show that were increased in anterior cingulate cortex in both TRS late and early onset. But then, so the glutamate was there, something, even if we're not able to stratify the patients. And then, which was interesting, that diffusion tensor, that's what kind of connectivity, it's not really matter of graph analysis, but you have a connection for sure, was showing a clear reduction of structure at the level of superior longitudinal fasciculus in patients affected by the disorder. So now, we can modeling antipsychotics. Again, I don't believe there is any animal model of any psychiatric disorder. We have a lot animal model, very good animal model of mechanism, potential mechanism, or putative mechanism of mental disorders. And to make things very quickly to the end, we have been looking for many years to this structure that is postsynaptic density protein that, as you neuroscientists know much better than I know. And looking at this structure, why? Because this is a kind of hub in which the dopamine and the glutamate mechanisms and the signaling may converge through some proteins that have different function, can be adapters, can be scaffolding proteins. And also there are a lot of proteins that are directly linked to the receiving signaling system. That's an NMDA receptor, AMPA receptor, and mGluR receptor. mGluR receptor are linked directly with some of this protein in the postsynaptic density, specifically with the protein that has this strange name. We've been investigating for more than 20 years. I was in the United States when it was discovered. Johns Hopkins, his name is Homer. This morning, Dr. Lisa Bonagura has done a wonderful presentation of which are the background to consider this protein interesting spot for the drug action of antipsychotics. I am going to show you in very summary way that basically we've been demonstrating almost 20 years that all antipsychotics in different way. I don't pretend to believe that these are real very fingerprint for the antipsychotic, but they stand there. All antipsychotics can change the expression and the protein of postsynaptic density protein. So in other words, antipsychotics do not work only on the system, and we're expecting this in some way, that's the dopaminergic one or other system then related to the very complex receptor profile of many of our antipsychotics. All antipsychotics basically are very complex, with one exception, this the sulpirides and amisulpride. That's a basic D2, D3 receptor. No one knows what means the 5-7 antagonism at that receptor, but basically it's like that. And so, interesting things that for many years we thought that this was an intriguing aspect to have a fingerprint of antipsychotics, but then after 20 years we started the story. What happens? That was demonstrated with JWAS, with so known preconstituted concept and just scanning all the genome, that some of this protein, specifically the one we have been looking for many years, Homer, at the time we didn't know anything about the pathophysiology of these disorders and the implication of PSD in these disorders, are considered of the top ranking for schizophrenia. And we have been showing this also, it's been shown this in the lab of Professor Usiello, with which we have been teaming together, and this has been shown that PSD95, that's one of the major components in terms of quantitative presence in PSD of all PSD proteins, can be a major distinctive molecule for the pathophysiology in terms of separating normal control. This is a study from schizophrenia. This is a study done in two different post-mortem brain samples, one from Harvard University, from Bethesda. And again, looking for network analysis, one of the things that came out that was not surprising, but was difficult to believe that was in that direction, that just with the connectome analysis, with the network and Fourier plus application, the VGLUT2 was one on the top ranking gene in post-mortem brain analysis that correlated with appearance of schizophrenia and can predict one. So what we did at this point, we tried to replicate what we have been done in PET scan in animals. Let's say I'm saying something, I am a liar, because we did exactly the opposite. We start first with animal, and then we move into PET scan. And then we went back again on animals. So what's much more complicated was the people who speak very well, they speak about the reverse translation. I never understand which translation is, what is the reverse translation, but this is what we did. So what we did something simple. We gave alloperidol to the animals, and we showed that giving alloperidol, using just the image of the transcript of Homer genes that is in the synapse, it is relevant for the dendritic spine constitution as well for the architecture and maintenance of the dendritic spine. Look what happened when you give alloperidol to animals in terms of connection based on the expression Homer 1. This is when we do the most strict calculation of the connection, in which on the left side there are the condition of the treatment, on the right side the vehicle, just this is a kind of reconstruct brain. And look at this when we in some way release the, let's say the P for the valuation of the connectivity story. Again, there is a much different, profound difference if we consider small, very small condition responsible for the difference in terms of connectivity. But these are normal animals, so we decided to do something that, again, is not a real model, but we have done this many years ago, and then we replicate very recently, and it's treating animal with ketamine. As you know, ketamine NMDA antagonist. Ketamine does a lot of things. It's also a good antidepressant, different doses, obviously. What we showed that even with the pre-treatment creating some kind of psychosis-like condition, we were able to individuate difference when we were giving, by the way, not just a pure D2 antagonist, but the drug that we were recognized to be one of the few drugs, like a clozapine, equimolecular acting at D2 and D1 receptors. This is one big mystery. There is increase of dopamine in the brain of schizophrenia patients. Okay, but why? Why? Which is the reason for which if you give D2 antagonists, you are able to counteract delusional hallucination? If you give D1 antagonists, considering the dopamine D1 receptor has much higher number in the brain, you don't see anything. We have been done this with the story of NMDA blockade also with using an animal model in which there is a perturbation of the aspartate that's considered relevant for the story. We have done this also in giving phencyclidine, that's the homologue of ketamine, and again we were able to see both, even in the transcription on the right of the diagram, and also in PET scan, this scan from Trento Lab of Professor Bozzi. But this is also what happened with the clozapine. Since we're speaking about treatment-resistant schizophrenia, and so what we did an experiment in which we were using aripiprazole, as partially agonist, clozapine, and vehicle, together with the most common antipsychotic used for, at least among the first generation antipsychotics, the haloperidol. And again, also with this compound, we were able to observe both this case acute and chronic. Usually the previous study were done in acute, this is done in acute and chronic, that we could modify giving aripiprazole or clozapine in differential manner. So they don't do the exact same thing. What you expect from a drug that is partial agonist and other drugs that is not very high potency, full D2 antagonist, but has some D2 antagonism as well. Okay? But now something comes. Everything seems to fit relatively well. We have a model in human, we have a model in animal. Antipsychotics seems to do what they were supposed to do. And in other words, seems that the hub constituted by the PSD is really the best things that we should have to test. As have been seen with different antipsychotics, we have a different fingerprints. But Natura is not any simplex, like Isaac Newton was saying. Probably it's very complex. And so we, many years ago, we teamed together with Bethesda and the lab and with the neuroscience lab at University of Tübingen. And what happened is, it was interesting, that basically what we show, we knock out our animal that were showing depression after KO of adenosine 1 receptor. We show that basically this animal were profoundly depressed, sorry for the similitude, and the condition could be altered, given to us this animal imipramine or ketamine, but non sleep deprivation. At this point, we thought that, okay, what's going to do with the PSD? What we should demonstrate that the target was exactly the PSD, and exactly was the Homer. In fact, if we delete also Homer gene, all antidepressant effects are lost. So they say this, that this probably do much more in terms of response.
Andrea.
Okay, so I'm going really to the end, and I was promising you, I will show something that we really believe is a novelty in schizophrenia. As you know, schizophrenia patients may have profound cognitive alteration. And in some cases, the alteration of schizophrenia patients can remind the condition that we know as MCI. So we thought there is any overlapping between MCI and cognitive impairment in these patients. And we decided to do something that is common things to do with MCI patients. So try to spot an amyloid in the PET scan. And we use flumetazole to do this. There's a radioligand that binds amyloid. What happens? It's an interesting story. We did not find any kind of amyloid deposit in the schizophrenia patients. But then something happened that was very intriguing. As you know, when we give this radioligand to bind the amyloid in the first part of the scan, you can see an image that reminds exactly the image of the FDG. No one has done this before. So what we try to do now is to do this in a large scale of patients using the first part of radiotracer administration just to show the metabolism of the brain, and the second part, the binding to the amyloid. Okay? We also doing things in different strategy. For example, we try to separate patients using TMS, not as a therapeutic tool, but as diagnostic potential tool. It has been shown in terms of probing in vivo glutamate excitation and GABA inhibition by Dr. Barone. It has been published in Nature Schizophrenia a few months ago. And we also try to work on something that directly comes possibly from the human brain in patients affected by schizophrenia, in patients who respond and not respond. And there are the blood extracellular vesicle. The one you see on the left, a real vesicle isolated from the blood and coming from the brain. I have to say that we are very disappointed with this, not for the results, because we were scooped. And we were scooped in a very bad way. The people who scooped us wrote a wonderful paper, Australian researcher, a wonderful paper on Nature Communication. Last things really is this one. We have been speaking about structure, we have been speaking about connectivity, we have been speaking about PSD as a hub between, in the synapse between dopaminergic and glutamatergic system. But we're wondering if really there is any possibility to manipulate these proteins. This can be done. There is now a compound, there's a benzotiazole aniline, and can be used in animal model, and in animal model it's showing that it's able to capture the structure, the mushroom structure that's been altered by several treatments, such as ketamine or something other. But what's most interesting thing is this. For the first time, there is ongoing clinical trial, phase 2, in the United States and in Australia, in which this drug, at oral formulation, is given to schizophrenia patients, and with the idea that for the first time, instead to use only a G protein coupled neurotransmitter-related small molecule, we can use a molecule that goes directly to the structure and the most intimate structure, the core of the dendritic spine. So these are just few of the people that have been working all these years, very hard workers. I should say that without these people, nothing could happen. And together with them, all people all in the world, for which I have been very lucky to be sustained among these people that have been makes the story of schizophrenia. And I am deep in debt to all of them, young and old. Thank you.
Thank you, Andrea. It is a really great job, and we wish all the best for this patient and for you indeed. And thank you all for your attention, and we can proceed with the next lecture, the plenary.
Okay, good afternoon to everyone. It's a great pleasure for me to introduce Professor Guillermina Lopez-Bendito. Guillermina stands out as one of the most inspiring voices in contemporary neuroscience of sensory circuit development. She graduated in biology and earned her PhD in neuroscience in 2000 at the Institute of Neuroscience of the Spanish National Research Council in Alicante, where she is now full professor and leader of the Development, Plasticity, and Reprogramming of Sensory Circuit Group. After a postdoc at the University of Oxford until 2004, she next joined the Spanish National Research Council as a PI in 2007 and became head of the Developmental Neurobiology Department in 2020, and in 2024 she was elected co-deputy director of the Institute of Neurosciences. Lopez-Bendito's laboratory investigates the development and plasticity of brain circuits. Her team discovered that sensory representations arise while circuits are still being assembled during embryonic life, and that spontaneous activity plays a crucial role in shaping these early networks. In summary, their research has pioneered three fundamental aspects of neurodevelopment. First, they have helped to define the molecular mechanisms underlying the construction of sensory circuits in the brain. Second, they demonstrated the role of spontaneous brain activity in driving this formation, the formation of these circuits during fetal development. And finally, their research agenda on plasticity and cell reprogramming in the developing brain is directed toward recovering circuits after the early loss of a sensory organ, with the long-term goal of designing strategies capable of restoring defective neuronal connections in patients with sensory deficits such as blindness or deafness. Professor Lopez-Bendito has led as principal investigator 6 consecutive national projects of the Spanish State Research Plan, 2 Prometeo projects of excellence from the Valencian community, 1 Human Frontier Science Program grant, and, well, 3 consecutive ERC projects, a starting, a consolidator, and finally an advanced grant. Unsurprisingly, she has received several prestigious national and international awards. She is an AMBO member and currently the chair of the AMBO Young Investigator Program Selection Committee, and she's also a member of Joseph Altman Award Selection Committee and of the scientific expert of the Museum of Arts and Science of Valencia. And in 2022, she was elected corresponding academic of the Royal Academy of Exact, Physical, and Natural Sciences of Spain. It is therefore a great pleasure and an honor for me to introduce her lecture today entitled Building Functional Sensory Circuits, Unraveling the Role of Spontaneous Activity in Modality Specificity. Thank you.
So, thank you very much for this very kind introduction. So I'm very happy to be here, so thank you to the organizers for inviting me, especially to Monica for inviting me here. And this is, I think, is my first time I give a lecture in such an amazing place. So I must say that this is incredible. You guys have a super beautiful, history. And I must say that last week I declined to go to the Spanish Society for Neuroscience and to give a talk there in the Canary Island, and I think I don't regret to be here. So, but don't tell the Spaniards, they will be very upset of me. Okay, so I decided to give you an overview of what we have been doing over the past 5 years, mainly concentrated in new unpublished data. So this is what I will be giving you today. And everything starts, I will say, with this picture, which is the major challenge in neuroscience, is to understand how this incredibly amount of neurons and connections is actually made, and how all this complexity emerges and works. And I think everybody agrees that this is one of the greatest challenges, and it still is. So maybe what we can do to approach this question is actually choose one, or let's say several of these connections that work together, such as in one function that has to do with the processing of sensory information. And this is what we have done in my lab. So as you know, sensory circuits and perception relies on well-defined circuits that handle different types of sensory cues separately. So understanding how this is achieved is one of the major challenges. And not only that, but these circuits are actually being specified so that within each modality you have maps that they represent and are absolutely fundamental to represent and to process sensory information within the separate sensory circuits, such as the somatosensory map in the somatosensory cortex, the retinotopic map in the visual cortex, and so on. So still we don't know what are the developmental programs that allows these maps to be formed and these specific cortical territories to acquire sensory modality specification. And this is the major goal in my lab, is to understand how cortical areas acquire their sensory modality specification. That means how an area becomes visual or somatosensory and how this process is influenced by spontaneous activity, genetic factors, and environmental programs. And this understanding is obviously crucial not only for developmental neuroscience and neuroscience in global, but also to understand conditions of altered development or neurodevelopmental disorders. So we approach this system from the periphery to cortex, subcortex, zooming in specific cell types and circuits. And for the last decade, my lab has been concentrated in understanding specifically the role of subcortical structures such as the thalamus and other subthalamic structures such as the superior colliculus, as you will see in a minute, that are known to be specific regulators of cortical development. So they have a strong power, a strong to say, on how the cortex develop. Okay. So one of the major, do I have a pointer or do I point here with this? Do you see my mouse or not really, no?
Yeah,
you see it? Okay. So one of the major questions, as I was saying, is how cortical areas are functionally parceled in different sensory territories. And this is particularly an important question because we know that the embryo in humans is the same. Cortex emerges as rather homogeneous structure, homogeneous in terms of limits. And then what has to happen is that during development, with mechanisms that are probably relate genetic programs and activity-dependent mechanisms, first spontaneous activity and then incorporates evoked responses, this actually makes the transition from, as I said, a relatively homogeneous cortex to a very functional parcel with a purpose cortex. And a milestone in my lab over the years, and we put a lot of effort in the lab, is to understand how genetic programs and spontaneous activity interact in early stages of development. And I would say that this was a milestone because, this we know very little in the field because of lack of tools that allow us to really record spontaneous activity in early life. So a few years ago, 4 or 5 years ago, in the lab, we put together to work this technique in which you can have living embryos, and then we can stimulate the very first immature circuits in vivo. And these allow us, as you would see, to really answer some of the fundamental questions that were still in the field to be answered. One of the questions had to do with when maps are formed by real, functionally formed in the developing brain. And to answer the question, we use this technique I just showed you, so we can stimulate distinct parts of the peripheral territories, in this case, the somatosensory input in a mouse. This is an embryo before being born. And then what you can do is stimulate different territories, and the question was, is there already a map as the neurons and the circuits are being built? So here I show you the movie, so you will see how peripheral stimulation happens. So we go in and we stimulate different portions of this animal snout, and then you are recording the activity that is linked to these stimulations in cortical neurons in S1, and this is because neurons are encoding GCAM6. So then the point here is that we were able to show that yes, indeed, sensory territories and sensory maps are being formed early in life, so they don't need experience-dependent activity to be unfolding, which was a very important point. So we show that actually the formation of these broad maps depends on a subcortical station, actually the activity of thalamic neurons during development. And we show a few years ago that thalamic neurons have spontaneously these waves of activity that coordinate distinct sensory systems. And if we perturb this activity during development, we are able to actually perturb how the maps develop. So this will be the normal scenario. You will have the thalamic waves. This will help these cortical neurons to be organized in columns. And this is the pre-step for the anatomical cortical mass to be formed, which is the basis of normal sensory behavior. So as I said, if you perturb these activity waves in the thalamus genetically, you remove the synchronous activity and make the thalamus to talk in another language, then you will have forever a perturbed absence of anatomical mappings in these somatosensory fields. Okay. So this shows that, yes, the brain has all the tools to make maps as develops, and this was a major point. But then we wonder if, when making these maps, already sensory territories are specified to a single modality or not. So what I'm asking is, do sensory circuits emerge as sensory modality specified? So in the field, and I'm sure some of you have even done these experiments, we know very well that sensory cortices have to work in a unimodal fashion. So this means that when you stimulate a sensory pathway, in this case the whisker path, you should, in this case, have a contralateral unique somatosensory S1 response. So only neurons in S1 should respond to this stimulation. So the question here was, is this how circuits actually emerge during development? And this was actually unknown. So we performed the same experiment now that we have the tool at embryonic stage. And, sorry, yes, it's me, remember. And then what we found is actually was very surprising. So when you stimulate the whisker path in an embryo, you have a multimodal response. So both S1, but also bilaterally B1 cortices, the visual cortices, are activated. So this means that sensory circuits emerge multimodal, and there is a unimodal switch during development. The question was, what is the mechanism? And this is what Teresa was looking at. So I will go very fast into this part because it's already published. So to answer this question, what Teresa did was, okay, because we thought that this switch from multimodal to unimodal has to do somehow with the developmental program of the retina being colonized in the distinct stations, what Teresa did was remove the eyes embryonically and look what happened with the responses. So this is the control experiment. Again, you stimulate the whisker path in postnatal life and you have a unimodal response. But if you remove the eyes, these animals remain unimodal for a week. So no matter what you do, in every single case that you remove the eyes embryonically in blind pups, the cortices remain multimodal to touch. Okay, this means that the eyes somehow are needed. This is just the quantification, so you see it's a very robust phenomenon and happens in 100% of the cases. So to make a long story short, so what we found is that this multimodality not only happens at the level of the cortex, but also happens at the level of the colliculus. And we wonder whether the colliculus have anything to do with this multimodality. As you know, the colliculus, the superior colliculus, is a multimodal station that receives both visual and somatosensory inputs. And what Teresa did was put TTX bilaterally in the superior colliculus, and she was able to rescue from multimodal to unimodal the responses. So really the superior colliculus was key for this mechanism to occur. So here I show you the developmental program. So this is what happens as there is a switch from multimodal to unimodal responses that happen during development. And what we have found is that the mechanism occurs at the level of the colliculus, so there is a refinement of axons from the superficial layers where the retinal axons are going to kick in, then somatosensory inputs that were developmentally there are retracted. So we think that the arrival of the retinal axons to the superior colliculus kicks out somatosensory axons that are developmentally there, making a space for the unimodality to occur. So then there is this window of time, I didn't show you, but there is a window for all these mechanisms that is depending on the arrival of the axons from the eye. So during development, you may know that the eye is not silent. There is something very important occurring in the eye, in the retina, which is that the retina is full of spontaneous activity called retinal waves. And there are 3 types, 1, 2, and 3. Type 2 and 3 are very well known, and their role have been involved in visual processing and the refinement of visual circuits, and actually many labs have perturbed these waves and shown an important defect in how visual circuits are developed. But regarding stage 1 retinal waves, no much was done because these are very early, are almost embryonic, and to perturb these waves embryonically was actually a challenge. We know that these waves are gap junction mediated, so what we did is to block these waves by carbenoxolone, so this is the control. And this is when we put carbenoxolone and block these retinal waves, look what happens. You have a multimodal response. So really, stage 1 retinal waves, and only stage 1, because if we block stage 2, nothing happens, are important, crucial regulators of this segregation to occur during normal development through the same reorganization of axons in the superior colliculus. So we were able to show that this has an impact. So actually, if this doesn't occur on time, and this was work by Mar in the lab, then this has a long-term consequences for how visual axons are developing. And here I show you eye-specific segregation by 2 colors in the superior colliculus, and as you compare with the control, if you perturb these retinal waves, you have an impact during development and until adult in how these circuits develop. Okay, I show you that there is this mechanism that allows sensory circuits to be sensory modality specified during development that occurs partially in the superior colliculus, and we have reasons to believe that involving GABAergic cells. Okay, now the question I would like to talk about is this one, that relates to how really sensory cortices acquire sensory modality. So I told you that this depends on a bifurcation or segregation that occurs at the level of the superior colliculus, but really this is something that at the level of the cortex was really lasting for a long time. And this question relates on decades of research, and many labs have shown the expression of morphogenes and other genes, transcription factors that are expressed, ingredients are set the broad boundaries of sensory cortices during development. And then activity-dependent mechanisms are thought to really delineate or refine these boundaries. So what do we know so far? Actually, very little. If you compare, the field has advanced a lot with single-cell transcriptomics and so on, but not a really detailed or direct, let's say, direct comparison between developing cortices, CES1 and B1, for example, hasn't been really done. We know really data from in situ hybridization and all microarrays screen that have shown different transcription factors and markers that are expressed either in S1 or B1 during development. So yes, there are markers for these areas that we all use for in situ hybridization, for example, to label these regions. But what do we know about the function? I just told you right now that during development, these cortices, although they express different markers, they work together at least for a window of time. So then it doesn't make much sense. There is a contradiction here. If we look at the genes, yes, there are differences among these developing cortices, but if we look at the function, these cortices seem to be working together. So we need to really solve this issue. So this is what we wanted to do in the lab, and to revisit this old question with new tools and new approaches. And what we think is that we should look at the process of cortical arealization as a layer process. So let's say that we have one layer, let's say layer A, that has genetic programs, and layer B that has spontaneous activity patterns. So depending on the layer you are looking, that's where you, in principle, could get this contradiction. But as I will show you, there is no contradiction whatsoever. So the question we are asking here is, maybe what happens is that early in development, these genetic programs are not so strict, and that's why everything is less specified, and you will have a multimodal response, and then this is getting fully specified, leading you to the unimodality that you need for the functional circuits later on in life. So is this the case? So everything started by the work by a very talented postdoc in the lab, again, Teresa and Lorenzo, a PhD student. And this is unpublished data. We are close to submit now this work. So what we did was to really compare directly by single nuclei transcriptomics S1 and B1 at P4 to start with, which is an important postnatal day when I show you that also functionally these areas are working independently. So when they compare these, to make a long story short, the point here is that they found that the cell type, if you color code the different cells that you find in these cortices, the cell type, this seems to have the weight of the difference between S1 here in orange and B1 in blue relays on the glutamatergic neurons. So the glutamatergic neurons seems to be the powerful cells that say you, S1, are different from you, B1. And when we did the GO-term analysis and molecular function, the surprise came, and we found that the majority of the genes, that differentially expressed genes, codify for activity-dependent genes. So activity-dependent genes tell you are different, S1 from B1. So then the question was, this might be telling us that perhaps what occurred here is that S1 and B1 are different because their genetic programs are different, that are saying that your patterns of activity should be different. Is that the case? So then we did mesoscale calcium imaging day by day at the postnatal life. Here I show you P6, and this is mesoscale in EMX1, glutamatergic neurons. And Fran and Danny were putting an effort for 3 years to be able to develop tools that can, by machine learning, unbiasedly determine the patterns of activity that you can extract from these movies. Basically, you give these movies, and then it gives you the patterns. And we were able to extract 3 types of patterns. 2 are stationary, so this means events that do not move, are like spots. And 1 is propagating, this means a wave. And when we color-coded in a UMA for S1 and B1, S1, no surprise, a spot-like activity as this one, and B1, wave-like activity as that one. So this was very reassuring because we know that B1, the visual cortex, is full of wave-like activity coming from subcortical stations such as the retina. But what is the real locus for this difference? So what is the neuronal type for this difference? So what we did was deep network analysis trained by publicly available data from the Paola Slaughter's lab, and what we did was fraction the glutamatergic neurons in the different subtypes. So then here I show you the different subtypes. You have the deep layer, for example, cortical projection neuron, upper layer, layer 4, and so on. And then if you might already spot it, because you are following me very carefully, that layer 4 and corticothalamic projecting neurons are the 2 cell types where the weight of the difference is more apparent. Actually, you can do differentially abundance analysis, and that is looking for neighborhood relation among cells and differentially expressed genes, and really layer 4 and corticothalamic projecting neurons are the weight layers that tell you are S1, you are B1. Then some of the genes are here that really are layer specific for S1 and B1, and here we did the UMA for some of these, and beautifully you see, for example, neuregulin 1 is an S1 layer 4 specific gene, or TSHR3 is an B1 layer 4 specific gene. This was confirmed by Allen Brain in situ, and now we have the spatial transcriptomic data, but this is beautiful. We want to keep it because you can beautifully see also the expression gradient for these genes in these cortices. Okay, so what I'm telling you is that you have postnatally S1 and B1, and they express differentially expressed gene in a layer-specific manner. And these layer-specific manner genes are very important because they define which sensory cortexes you are in. But if all our thoughts are right, then we should see these patterns of activity also in the layer specific. So this was a real challenge for Lu Wei. She's a fantastic postdoc that was trained in Japan to do two-photon imaging in mid-stage postnatal life. So when she came to my lab, I said, look, Lu Wei, I know you did P6, but now you need to do P0. So, and you know, it really looks like numbers, but I tell you, this is a real challenge. So you need to open a little window in these little paps, in this little brain and little head. And these cannot move over time of the recordings. These animals are non-anesthetized, so you really need to get all this information. And beautifully enough, Lu Wei was able to do these experiments and show, as you can see here, spot-like activity in L1, in layer 4 for S1, and wave-like activity in layer 4 for B1. So we are super happy because now our two-layer hypothesis seems to be right, in which you will have the two layers, and now we have to look at the interaction among these parameters. And yeah, we are really, really happy to say that, you know, when we look at where the differentially expressed genes are, as you know, these are thalamus-recipient layers. These are layers that receive input from the thalamus. So the fact that the majority, the weight of this differentially expression relies on these layers really suggests that the thalamus, or maybe something from the thalamus, is important to get this sensory and modality specification. I told you that the way the symphony that the thalamus is actually singing is important, that, you know, it has to be in synchrony, and if you perturb the synchrony, you actually perturb things very important in the cortex, such as the barrel field map. So I'm not gonna show you this, you will need to invite me again. I will leave you with this, but we have a very nice evidence saying that when we perturb the activity in the thalamus, we have a strong impact in the cortex, but beautifully enough, this impact is not destroying the sensory modality specification of the cortex, which makes sense, because when you perturb thalamic activity, still the neurons from the different thalamic stations get to the right place. So this is even more interesting. So I give you a glimpse on this. So we now have all the activity, thalamic activity-dependent genes in a layer-specific manner. We have the genes that are independent on thalamic input, which are the most interested for us. And for some of you that work in developmental disorders, so we have found an incredibly amount of genes that have to do with schizophrenia, bipolar disorder, and so on, in a region-specific manner. So what are these protogenes for that are independent on thalamic input? So what Isa in the lab is looking at, which is a very, challenging idea, is that maybe there is a code, guys. Maybe, thalamic immature neurons and cortical immature neurons are talking to each other somehow, and they are making this code for synaptic partnership. And this is what we are looking. Actually, Lorenzo in the lab is looking at this code in the thalamus, and just 2 slides to show you what we found, which is very beautiful. This paper is in bioRxiv and is currently in revision. So what Lorenzo found is that, yes, there is a very nice, by single-cell multi-omic analysis, he found very nice strategic plan in the thalamus. So you have 2 lineages, lineage 1 and 2. Lineage 1 is the sensory, and lineage 2 is the associative and motor thalamic neurons. And he found beautiful markers and transcription factors that are expressed in either of these lineages, and actually give you some of them. Lorenzo found that sensory nuclei development depends on some of these genes, and non-sensory nuclei depend on this other list. And beautifully enough, within the sensory nuclei, he found two branches, branch 1 and 2. And these two branches, by tracker-seq analysis in utero electroporation of the entire signature and clonal analysis by barcode, what Lorenzo found is that this gives rise to visual and somatosensory nuclei. And these are very specific nuclei, as I told you. One is for visual function, the other one for somatosensory. But the most beautiful part of his story is that visual and somatosensory cells in the thalamus are coming from the same ancestor. Actually, are coming from a high-order state that later on is bifurcated in a first-order function. And he found that this role of one specific transcription factor, SP9, is very important for this transition. I have no clue how I'm doing with time, but I will continue as I look in your faces and you seem to be following me. So you can stop me whenever you think you're bored. So the next thing I wanted to tell you is a little bit what another part of the story in the lab, which actually had to do with the spontaneous activity, and is whether this sensory specificity only relies on neurons. Okay? I told you that yes, glutamatergic neurons, layer 4, are very important for this, but are the only players in this. So this work I started from data that we published a few years ago that beautifully showed, and this was, I tell you, completely unexpected, that astrocytes and neurons from the same region show molecular imprinting of the origin. So when we look at transcription factors expressed by neurons, classical, GBX2 in the thalamus, EMX1 in the cortex, we show that astrocytes from the same region show, in a lesser extent, the same levels of specificity. So this means that neurons and astrocytes might work together. And this is what Francesco is looking in the lab. So we bought this technique in the lab so we can do dual color mesoscale imaging, so recording astrocytes and neurons at the same time from the same brain. And here you have neurons in magenta, astrocytes in cyan, and you can beautifully see, you know, every time I look at these movies, I'm more excited. You can see retinal waves here in the superior colliculus and how one seems to be following the other, which is really exciting. So this functionality that I showed you before, this functional modality specification of cortical patterns, seems not to be only occurring at the level of the neurons, but also to sibling cells such as the astrocytes. So here I show you that indeed in B1, you have retinal waves in astrocytes, and in S1, you have a spot-like activity. And there is an interaction between these cell types, because if you perturb the astrocytes activity in B1, and you can do this by perturbing a connexin 45 or 43, a specific connexin only expressed in the astrocytes, you actually perturb how neurons behave functionally in this cortex. So we have reason to believe that this astrocytes activity is crucial for the development of these neuronal circuits during life. Okay, I think I have a couple more slides. I did not resist to tell you about this because I think you guys are also interested, of course, in plasticity, and we also are interested in plasticity, which means how the brain copes with an early loss of sensory input, which is a major issue because, you know, not only for congenitally blind or deaf conditions, but also for some of these neurodevelopmental disorders that are known to be linked to sensory disorders as well. So, okay. So I will just tell you one single story from Mar, that Mar was looking into these windows of plasticity in early life. So this is before active sensing, before eye-opening, before all this occurs. So how early the brain notices that something has occurred, and really, what does it do? So basically, Mar wanted to look at this question by looking into the relationship of two neighboring cortical areas that have barrels, yes, they have representation of the whiskers, but their function to sensory behavior is very different. So here, the magenta area, the posteromedial barrel sulfil, is the proper barrel area, the big whisker related, that is important for discrimination task for the rodent, okay? So maybe you haven't even heard about this anterolateral barrel sulfile, but there is a barrel-like structure near to that one that really represent the little barrels, the little whiskers that these animals have in the lip that are used for secondary function. So my question was, so is there a window of plasticity in which the function related to the anatomy and related to the plasticity of this area can be replaced without touching, let's say, the remaining sensory receptors? So can actually the brain centrally cope with having little whiskers but has to do complex behaviors? That was the question. So what Mar did was even more complicated to what I told you before. So Mar was able to cauterize, remove the whisker pad in an embryo, only the magenta part. Here I show you this. So the magenta area, you have these big follicles that will represent the big whiskers in the future. This is E14 embryo, okay? So when she cauterized only that magenta area, leaving intact the number of little follicles, and then later in life we measure the size of the hairs and are untouched. So what she found, and I show you this piece of data, was incredible. She found that only when she does this removal embryonically, the brain can cope with that. What it does is that make the cyan area very large, takes over the other part, and not only that, that can be understandable, you are removing one area, the other one is larger, but what she found is that the barrels are larger. Individual barrels are larger and more defined. But this is even if you have only little whiskers. So she found that this cortex can support complex tactile stimulation in the adult, and she did some behavioral experiments. So she compared the control with just trimmed whiskers, and then with these animals that have without the larger whisker for life. And basically what she found is that these animals can do complex tactile discrimination. And this is, if you think about it, is a very nice mechanism for the brain to be able, for a window of time, make reorganization, re-escalation of the cortical territories in terms of anatomy and function to be able to do this complex behavior later on. Okay. I like this quote that we have in the lab: the developing brain orchestrates its own maturation through genetic blueprints and waves of spontaneous neural activity, and these elements define spatial territories and shape future sensory function. And this is very important because not everything that I have tell you is important for mouse development, of course, but it's also important for human development. And actually neurologists, and we are starting to have a collaboration with a hospital with pediatric neurologists in Spain, are recording spontaneous activity in preterm babies and newborn babies as a prognosis factor of development. So now they are making links where when this activity is not right, that later on some of these kids will develop neurodevelopmental disorders. So we really believe that the spontaneous patterns of activity, and that's why we put our effort, are predictive factors for this, and maybe in the future will help to design tools to redirect this plasticity. With this, I finish. Thank you very much for listening, and yeah, I'm advertising a position here in case you are interested. Thank you.
Thank you very much for the very nice talk, and thanks to all the audience for your attention. I wish you a good afternoon with the rest of our scientific program. Thank you.
apriamo noi.
Sì.
Questo va avanti.
Vale?
Qui, questo
Ok.
qua grande.
Ah, va
Questo qua è
automaticamente.
il pointer per
Ok,
andare.
perfetto.
Ok. Si vedrà anche su.
Ok, perfetto.
Questo qua per tornare indietro. Questo
Quello,
piccolino.
perfetto.
Però questo qua va avanti.
Puntatore, va bene, grazie, semplice. E ci chiamate voi, vero?
Sì, vabbè, moderatori. Appena sono pronti i moderatori.
Dovrei fare il talk alle 3:30, penso ci sia io adesso, giusto?
Com'è il nome?
Emanuele.
Va bene, la credo. Mica usa il suo computer?
No, avevo messo la chiavetta là.
Tutto a
quindi-
posto.
qui.
Già qua, già qua.
E quindi...
Allora, la presentazione la apriamo noi.
Ok.
Qua va avanti questo grande. Questo qua è per il laser. Che è elettronico, quindi lo puoi indicare anche qui, non devi andare per forza lì. Ecco. Tenendo premuto.
Sì, bravissimo. Va bene.
Buonasera, lei è Giordano. Ok.
Posso bere un sorso?
Certo,
Grazie.
è suo. Ha già visto come funziona?
Sì, me l'ha spiegato. Io mi siedo lì davanti, aspetto che mi
Sì.
chiamate, giusto?
No, fa tutto in autonomia.
Quindi io aspetto già qui.
Come preferisce, sì.
Grazie.
Siete autogestiti, ho
Va
capito.
bene, va bene. okay. I don't see anything there. No. Ah, ok, so it's moved by people.
Yeah.
Ok, lo caricate voi.
No, pure se si apre adesso, ma non si chiude.
Ok.
Poi lo mandiamo.
No,
Quando al
vabbè, basta
momento-
che ci sia qualcuno che va bene.
No, no, no,
pensavo
no,
si
vediamo
potesse
tutti,
mettere
perché abbiamo
prima.
le telecamere.
Pensavo si potesse mettere prima già il titolo, così la gente vede che.
No, perché c'è il tappo. Questo qua che c'è adesso fa parte di questa sessione qua. Quindi quando
devo
va-
aspettare proprio loro.
No, ma io penso che il moderatore.
No, non ci sono, hanno detto.
Non ci sono
Perché io
moderatori.
si vede che perché sono esibitore,
Ah,
non ci sono i moderatori.
con me possiamo. Allora ti parlo
Se
con,
si può
con la
mettere
prima
già
cosa.
la slide, la seconda magari. Vabbè. Because it's different from standard people with the moderator people asking things.
Right, okay.
I don't have them. Okay.
Okay, that's good.
Okay. Maybe I took the title of the presentation now. Grazie.
Abbiamo un altro settaggio che con la telecamera che riprende il relatore. Se vuoi andare in giro ti possiamo dare
No.
un microfono palmare.
Va bene qua, grazie. Così posso cambiare, sì, vabbè.
Sì, sì, puoi andare.
Lasciamo così tra 5 minuti.
Va bene.
Ok.
I microfoni sono già accesi, eh? I microfoni sono già accesi, quindi.
So you sent?
che sono tutti al caffè, la sala...
No, sai cos'è? Che forse fanno adesso le presentazioni dei poster di sopra. Che è un po' sbagliato, vabbè.
Mi dispiace. Possiamo provare a buttare un po' di gente dentro.
Good afternoon, everyone. My name is Emanuele. I'm the field application scientist at Molecular Devices. And today I'm going to show you some example of how to obtain quantitative data from neurotoxicity and screening assays. So before I want to mention my company, Molecular Devices, is part of the Danaher Group in the life science sector, part of one of the 9 companies in that sector. The last one added was Abcam very recently. And there is also a biotechnology, diagnostic, environmental, and applied solution sector that very recently joined in one single business. Molecular Devices is spread all around the world. The headquarters is in San Jose in California, and the European headquarters is in UK. Our customers, they go from pharmaceutical, biotech, universities, and government. In Italy, for example, we are present in universities like Milano, Roma, Padova, Firenze, Napoli, and not only, and in centers like Istituto Italiano di Tecnologia, Human Technopole, ICGB, IZS, e per esempio varie, and for example, different biotech and pharmaceutical companies. Our mission is to be the innovation partner that empowers scientists to achieve landmark discoveries. And how can we help scientists to achieve these goals, in particular, in this case, in neuroscience? Our solutions, they go from microplate readers with GXP compliance services, but also imaging and high throughput screening systems, 3D biology system and services, instrument for clone screening and selection, digitalizer and amplifier for patch clamp, and all our solution can be integrated with customizable workflows. Now let's dive into the topic, neurotoxicity and screening assay. So the nervous system, as you know, is sensitive to toxic effect of many chemical compounds, environmental agents, and naturally occurring substances. This exposure can lead to neurotoxicity, and you can get temporary or permanent damage of the brain or peripheral nervous system. You can get damage of directly activity of neurons, activity of glia that can lead to inflammation and chronic disease and contribute also to neurodegenerative diseases. So it's very imperative to get a screening tools that can be optimal and very reliable to define and find some drugs that can compounds that are that can affect the nervous system and so the human health. So how can we support you with these screening assays? Basically, with high content imaging systems and microplate readers, those can be applied for different cell-based assays. Are systems that can generate quantitative data, quickly perform complex analysis, and allow to dramatically speed up your screening. Let's see first the high content imaging. As I mentioned, they provide not only qualitative, but also quantitative data for screening, because, for example, you can image 96 or 384 plates. And here I'll show you an example of high content screening of neuronal toxicity using iPSC-derived human neurons. And this type of assays that I'm going to show you, you can, for example, easily do with our instrument Image Express system. For example, you can quantify neuronal networks integrity through the neurite outgrowth assay. You can see on the left, so we have control cells and cells treated with mitomycin C. On the left of the images, you see the cells stained with beta-3 and tubulin, and on the right side, the analysis tool. So with just few clicks, the software of the instrument is able to quantify, in this case, the total growth and create a dose response curve against the concentration of the compounds that you test, and extrapolate very quickly the, for example, IC50 of these compounds. You can also evaluate the mitochondrial membrane potential, and this is the polarization of the membrane potential is an early factor for toxicity. In this case with JC10, that is a dye that enters and accumulates as aggregates in mitochondria in healthy cells, but then when there is an issue in the potential of the membrane, the fluorescence from orange shifts to green. So you can image this. Okay. So again, images taken with the imager, and another type of module that can, in this case, measure the granules or mitochondrial per cells and calculate the dose response curve also in this case. Third one, you can monitor live cell toxicity using dyes that they can measure in real time the toxicity effect of some compounds, like calcine. And in this case, the cells were stained with calcine, so the same type of module of the analysis was applied, so the new rate of growth test to check, in this case, the toxicity effect of methyl mercury and other type of compounds, and to get in the same way the dose response curve for these compounds. Plate readers, so for assessing drug and treatment effect, one of the primary assays are cell viability, cell proliferation, and cytotoxicity. And this can be done with colorimetric, fluorimetric, luminometric assays with standard microplate readers. Nowadays, with multimode microplate readers, it's easy to basically perform any type of assay of these technologies. In this example here, we can see the assessment of viability of malignant glioma cell treated with different type of compounds and different concentrations. This was a promega cell titer glow assay, for example, that works in luminescence, and it detects basically the level of ATP, that is an indicator of metabolic activity of cells. In this other test, this is called the real-time glow annexin 5. At the same time, in kinetic mode, you can measure both apoptosis, reading in luminescence, and necrosis reading in fluorescence. So you can alternate the two read modes for multiple time points. In this case, up to 24 hours was done this test. Another type of screening assay, very important, is the calcium assay. We know calcium ion is really essential in neurons and generate a multitude of intracellular signals and control key neuronal function. So understand how the receptor and their agonist, antagonist, they can alter the calcium level is really important for neurotoxicity studies and drug development. So compared to standard traditional calcium imaging on microscope, calcium assay on microplate reader is way faster, allowing automatic and simultaneous measurement of multiple samples. So in this case, more suitable for screening. Molecular devices can provide this type of dye that are internalizable by the cells, and inside they become active. They bind calcium upon some stimulation by a compound, and you can check the increase in fluorescence. Is no wash reagent, and you have a very big signal to noise ratio. This type of assay can be run on a standard microplate reader, just equipped with injectors, you typically are two, and has to be bottom read capability for fluorescence. This because the activity of calcium is upon stimulation with a compound is very fast, so you need simultaneous inject and read at the same time from the bottom in real time to follow the kinetic. On the left, you can see a typical trace of the kinetic. This is a response over baseline already calculated with the software of astrocytoma cell line that are treated with an injection of different concentration of acetylcholine. And on the right side, you can see the same response over baseline, but against the different concentration of compounds. So you can extrapolate this data directly, quickly from the software automatically, and you can appreciate better the effect of antagonist or agonist on your cell line. We saw then that we can increase the throughput compared to the calcium imaging on a microscope, because using just two injectors, you are able to do well by well, but in automatic way. You can further increase the throughput. We call here now medium throughput using the FlexStation 3. That is our unique multimode plate reader with integrated fluidics. This is specifically designed for cell-based kinetic assay, because it has 8 or 16 channel pipettor, and it can add up to 3 different transfer of compounds from a 96 or a 384 well source plate that you position above your assay plate inside the instrument. In this way, you are able to transfer a full column, so you are able at the end to do in one single test, one single kinetic, a full 8 or 16 sample at the same time. In this example here, primary cortical neurons are tested, and in the first injection was injected TMDT, that is a potent neurotoxic compound that overstimulated the release, the increase of calcium, and then with a second injection was tested different type of drugs. And beside, you can see a typical analysis of one of them. In particular, in this case, was the ketamine trace, where on the software you can analyze the, for example, area under the curve, peak amplitude, peak frequency. If medium throughput is still not enough, we have the workhorse for high throughput kinetic screening. This is called Flipper Penta. It's the industry really renowned system for GPCR and ion channel screening, because this has a pipettor with 96 or 384 or 1536 tips. So it can transfer directly in one single transfer from multiple source plate compounds directly in a full plate. So at the end, you can get up to 1536 well tested at the same time in the same few minutes of your kinetic. So I said is really the leading instrument for GPCR, ion channel, but also for cardiotoxicity and neurotoxicity, because it has a camera that can read up to 100 reads per second. So with also with the software, with a specific analysis tool for peaks, you are able to not lose any, not primary or secondary peak. You are able to get all the information from your experiment. I mentioned ion channels. Of course, these are very, very important targets for drug discovery. The gold standard has always been touch clamp recording, but this technique requires really high-skilled operators. It's very low throughput, so for a screening purpose, is not ideal. So much faster method uses voltage-sensitive dyes. These can be measured on plate readers with injector capabilities. So for example, our membrane potential assay kit detects ion channel modulation by increasing or decreasing the fluorescence based on the membrane, if it's a depolarization or hyperpolarization. This is compatible with all ion channels, as is really a generic response that is measured. So you need, in the case, to be 100% sure your system is built to respond specifically to the ion that you are studying. Now, I didn't put in the slide for screening, but on the market there are also dedicated, always been developing dedicated ion channel sensor, like there is a sodium, there is a chloride, and we sell also, for example, a specific one for potassium. Here you see still an example of the membrane potential, where on the left we see an increasing signal in fluorescence, meaning a depolarization of working on the sodium channel by veratridine, and on the other side, tetrodotoxin that induce hyperpolarization. We sell two different type of kits, the blue version and the red version, because there are some ion channel that respond better to one, some other to others. Now, in the recent days, of course, the 3D system has become really rapidly developing, and because it's really important also in neuroscience. In this case, we are talking about neural 3D organoids from induced pluripotent stem cells to study brain development or neuronal diseases. However, the formation of these organoids is very complex, is a long process, so it's not that suitable for screening so far. So now a question, this is applied of course also to these cells. So how many of you have spent hours and maybe stayed late in the lab or even went on the weekend to make feed cells, split the cells, count the cells? So this is really annoying steps. We have the solution. This is the Cell Express AI automated cell culture system. This is an automated workstation for reproducible cell lines and organoid cultivation. As a built-in incubator, a liquid handling, an imager, and so can automate the processes with machine learning assisted monitoring, feeding, scheduling, imaging, and so data analysis. You can set up with one single software whatever you want to do, and you will get an automatic. In a typical workflow, where you can use 3D models, so in this case, testing iPSC-derived 3D neural organoid to check for a spontaneous oscillation of calcium is a response to different neuroactive compound. As a type of screening, you can then grow your organoids using the Cell Express AI. You can do a morphological characterization via neuronal markers on the Image Express image system. And then you can do your functional analysis of the calcium oscillation on the flipper penta. So you can see on top different markers, beta tubulin, GFAP, and progenitor markers with the imager. The trace is taken of the calcium oscillations taken with the flipper penta, and below some analysis specific on the peaks. Just to conclude, I want also to add that there are some other there are some readers like our multimode Spectramax i3X reader that can give you more from a plate reader, like also imaging and Western blot. In this specific case, to validate some cells edited with CRISPR, was used the imager that can be built in this microplate reader to check for the efficacy of cell transfection. And we have a powerful algorithm in the software to automatically calculate and count cells in bright field, and also, of course, if they are stained in fluorescence. And later you can check for the expression of your protein with the Western blot that works with time-resolved fluorescence. So in this case, you get great sensitivity, and the signal is very, very stable over months. Just to conclude, then, we saw in this presentation very different type of screening approaches. You can go from low to high throughput, and usually the combination of multiple approaches is the right key to get a complete and optimal type of screening and good screening. So molecular devices can provide you with all these solutions, and not only because we saw instruments, we saw reagents, we spoke about the software, but we can offer, of course, also scientific consultancy, validation services, application, technical support, and also specific customizable solution for your needs. Lastly, I want to just show you the big Italian team of molecular devices. Myself and some of my colleagues you will find on our booth that is at the first floor. So for any question, any information, you can go upstairs, and that's it. So thank you for listening, and if you have any question, let me know. Thanks.
I was curious about the system that is, checks basic activity in the cells, no, with calcium. I was wondering, but how can you do this in parallel? How many, for instance, culture dish can you analyze, and the sort?
So for the calcium assay that I showed, the instrument, so I showed you from, let's say, mid throughput is the flex station to high throughput, that was the flipper, and those works with microplates, so from 96 to maximum 1536 wells plate at the same time. So the flipper, for example, up to 1536 in a single kinetic, so usually calcium you record for 2-3 minutes. So those 2-3 minutes are done at the same time with the camera. Injection the whole block of compounds from a different plate to the assay plate at the same time. And the advantage of the speed is that because if you use otherwise a standard microplate reader, you need to read well by well, because you inject one thing and you read from below. So you will need anyway to wait that 3 minutes of the kinetic of the single well. In that case, in one single injection, you have injected in all the plate.
Still on that type
Yes.
of assay, because you mentioned, disease that could involve changes ion channels or on activity on the cell. But maybe it's a stupid question, but how do you discriminate between you know, impairment in activity or the fact that the cell is really suffering. So maybe there is an upload in calcium. I mean, can you do this? Can you say, I don't know, calcium levels are constitutively high, or simply the cell is not active? I mean, this type of...
the discrimination
yes.
that you mentioned, of course, I mean, I guess you cannot do directly on the system because you need to know what you are looking, and the system will give you just a response of an increase or a decrease, or maybe peaks that then you see a decreasing signal, or you can measure, for example, with multiple injection and agonist and antagonist, so you can see if the signal, there is a permanent opening of some channel, but then you need to know and build your system, of course, to be able to discriminate yourself what you are really looking, because here we see traces, we don't see images, we don't see the morphology of the cells, or what is inside, if it's stained. This is not imager. There is a camera, but you get values, let's say, from that. Yes.
Thanks for the contribution
You're
to our
welcome.
understanding. So, just a curiosity, how thick the tissue can be under analysis in your platform? So, for example, you talk about neural organoids. So, what is the ability to go deep in tissue analysis?
So I mentioned different type of instruments. So for the organoid, of course, you might mention, you might refer to the imager, because the flipper of the calcium oscillation is just a camera that measures a signal in the center, let's say, base center of the well. So this cannot change the stack in vertical. But the imager, of course, you can do with a confocal. I have to tell you that I'm not the specialist of the imagers, so I'm sorry I cannot answer how, so the nanometers that you mean. So, but if you can come upstairs, we can let you speak with experts, and we can answer your question, because I am the responsible more for micropage readers.
Okay, thank you.
But
So much.
good question, and come and I'll let you know.
Good afternoon, everyone. So first of all, I want to thank the committee to give us the opportunity to present our company today in this Congress. Today I want to talk about the cell analysis and how to improve the lab work and productivity. I'm Maria Ferreira, product specialist at the ACSPA. We are in Milan, And ASI is a compound of professionals in the biomedical sector that provide premium instrument equipment to the pharma, biotech, and healthcare sectors, and we support our customer in every step of the cellular workflow. We have many instruments, but today I want to focus and introduce to you the Seahorse XF analyzer. This instrument, so as you may know, the cells are not static, they are dynamic, but many tools analyze in a single time point, very few analyze in real time, and using a single time point analysis, the researchers sometimes lose the data. So with Seahorse, you can interrogate and understand how your cells change the behavior after treatment, after intervention directly on the cells. And this technologies is an instrument that analyzes the metabolism of living cells by using live cells analysis with 2D and 3D plate options, the label-free multi-parameter sensor that quantify protons and concentration of oxygen in the medium surrounding the cells, using the integrated injection ports for real-time compound testing, real-time kinetic measurements, ready-to-use kit and reagents, and lastly, an advancing software and support from Agile and Fast. So the instrument analyzes simultaneously to measure energy-producing pathway of the cells, the mitochondrial respiration and glycolysis. This is done by measuring the oxygen consumption rate, the OCR, and proton, the extracellular acidification rates that is performed by proton efflux rate, the ECAR. So, understanding the metabolism, this provides a clear window into the key function that drives cell signaling, proliferation, activation, toxicity, and biosynthesis. And the metabolism is a key to understand some mechanism in mainstream research, like cancer research, in which cancer cells redirect their metabolic program to support rapid growth and adapt to tumor microenvironment, in neuro disorder and aging, in which neuron function is highly dependent on mitochondrial function, in obesity, diabetes, and metabolic disorders, where the metabolic dysfunction drive insulin resistance and diabetes progression, in stem cell biology and regenerative medicine, in which metabolic programs control stem cells pluripotency and also influence the stem cell fate. And lastly, the immunology, in which the metabolism is important to drive immune cells lineage, commitment, and function. So, going back to our instrument, the instrument is composed by a microplate for cell culture, designed just for the Seahorse analyzer, a dedicated medium, a solid state sensors that is formed with the cartridge. During the assay, a microchamber is formed, then allowed measurement of oxygen consumption and extracellular acidification just in few minutes. And each well can also treat up to 4 compounds because the cartridge is composed by 4 injection ports. So in this mode, you can add inhibitors, stimulants, and substrate. The injections are defined by the users. We have three models of Seahorse, the Seahorse XF Pro with a 96 well, suitable when there are some multiple conditions to analyze and a need for screening, accommodates adherence, suspension, and spheroids model. The XF Flex, the new one, this is launched 2 months ago, is 24 wells format, accommodates 2-dimension and typical model, adherence, suspension, cells, but also 3D models like isolated tissue, organoids, and other 3D models like zebrafish. It's easier to handle because support larger wells and larger injection ports. And lastly, the EHS Mini with 8 wells format, best for limit materials, and is compatible with HS Mini plate that is high sensitivity. So I wanted to focus on the X Flex, that is the new one that we have, this model. This model is not just an instrument, this is an all-in-one solution because the instrument comes with dedicated consumable and also protocols to cover 2D and 3D models and analyze the metabolism in all of these type of models. So there is 3 upgrade in this instrument. The Seahorse X Flex is more robust. We have more consistent data because we have reduced interplate variation through the enhance of temperature control inside the instrument, better sensitivity of the concentration of oxygen and protons in the medium surrounding the cells in the wells, and more user-friendly because we have automated data quality control, new features in template design, and also dedicated assays view. Moreover, Agilent produces 2 types of microplates with different sizes. The cover, this is for 2D models, adherent and suspension cells that cover low respiring model from 25 to 1,200 picomole per minute, and also another microchamber with higher chamber sites that cover the high respiring model. Also, the Seahorse X Flex come with dedicated kits to perform real-time ATP rate assay that assess global cellular metabolic phenotype change. You can quickly determine bioenergetic pathway and also easily screen metabolic modulators. We have another kit like the cell mitostress test and substrate oxidation stress that get a complete mitochondrial respiration profile with multi-parametric output and determine fuel dependency for a specific cellular function or phenotype. And lastly, the Seahorse leukolytic rate assay that accurately quantify leukolytic activity in real time. This kit is designed for and using for 2D cell culture model, but we have also consumable that can apply to 3D application like tissue, organoids, and small organisms. We have a microplate for tissue, cell cluster, and also zebrafish. Mitostress kit dedicated for the 3D model, and consumable for organoids. So with this model, you can interrogate and understand the metabolism after your intervention in real time. This is a typical protocol of an assay with seahorse and tissue. The day before the assay, you have the preparation of the cartridge, prepare the medium, and prepare the assay template. The day of the assay, dissect your tissue or your sample, prepare slice or punch. We have many tools to prepare also this part of the protocol. Deliver the tissue punch to the well, and then run the assay. This is an example of the result that you can get with this instrument. This is an example of rat brain tissue and with a thick of 200 micrometers, in which you can see that the sample would respond to all modulators, for example, oligomycin, FCCP, and rotenone antimycin A, compared to the control in green that is quite stable. Also, in this picture, you can see how the basal OCR is quite linear to the tissue sites. So, if you want to know more about this technology and this model that we have, please contact us. You can write us. And don't forget that in ACPA, we can provide all the instruments you need for your lab. We cover all the cellular workflow, for example, for tissue dissociation and cell isolation, cell count and characterization of phenotype of your cells, real-time imaging and plate readers, and for analysis, the Agile Intelligence and Seahorse, and much more. Thank you very much for your attention.
Hi, and thank you for your presentation. So I was wondering what would be the format of the data that are going to be downloaded from the Seahorse XF model, and if there is a specific software for data analysis.
Yes.
Thank you.
Yes. The Seahorse X-Flex that I presented was launched 2 months ago, and is covered with 24 wells, and there is a dedicated software online, is open, in which you can analyze your data that you get from the instrument, and we have specialists and FAS that give you a course on how to analyze the data, how to work with the instrument. It's Wave Pro. You also can, if you have some data from other Seahorse, you can register on the software. It's free. And if you have some question on how to analyze, write me that I can take you in contact with our FAS from Agilent.
I have just a very naive question, because I don't know exactly the system how it's working in several applications. Is it possible to do also imaging and metabolism basically together?
Together with the X-Flex come a machine that does normalization, is the citation of biotech, but it's just to scan the plate after the measuring of the metabolism. So you scan the plate before and you scan the plate after the assay, and this give you the idea of the number and the morphology of your cells during the quantification of oxygen consumption rate and the ECAR, the extracellular proton flux.
Okay, okay. Thank you so much.
Thank you. Thank you.
Parlo io, parlo. Hanno fatto un cambiamento. Allora, non parlo benissimo inglese.
Ma sicuramente parla meglio di me.
Però quando faccio dei congressi quando giro mi mandano sempre qua a comprare le cose, faccio ma io non parlo inglese, ma tu le compri, tu ci riesci.
Ti fai capire, mia.
Sono di Napoli, capito?
Napoletano.
Ti devo far capire un po'.
Allora, hanno deciso che ci sarà una seria da capire chi.
Quello lì che verrà a parlare adesso, diciamo che non vuole la faccia in
Ok.
mettere al suo computer perché ha le note di laurea. Ho detto, ha detto che se no senza le note la presentazione dura 5 minuti. Ho detto ok, non si può mettere perché così
Si fa prima. Sì, ma quelli adesso sono i pompieri, Sì, no, ma... quelle due. Adesso sono i pompieri. I calci. Qui c'è la Certosa. E qui di Pisa, un paese Sì, lì è... quello sta più fuori. No, il tecnico.
Pensavo che fosse già...
Sì, perché...
ho visto che era...
È Bruno.
Infatti, visto
C'è stato una coincidenza.
Cercare di infiltrarmi, ma
ma basta il microfono.
Infatti
Lo sa usare già?
si stava appunto
Su Bruno con Becky. Ma non c'è?
No, ancora no.
quindi parlerà.
Su Bruno con Beckes.
E quindi al CERN non ci sarà nessuno?
L'avevo chiamato sperando che arrivasse in tempo, se no mi ci appoggio io.
Non è che hanno cambiato qualcosa, perché adesso avevo anche il nome Alessandro. la mettiamo lì? Ce la mettiamo qua? Che cosa? Ah, per la presentazione. È perché questo è un programma che non esce in condivisione schermo, non è, invece qua lo mettiamo in estensione.
Ah, ok, quindi il signor Bruno non c'è.
No, io sono nello stesso stand, ma
Ok.
mettiamo appoggiato senza chiudere questo. C'è anche il mouse ok, così vediamo un poco se possiamo fare. Ok, sostituisco.
Ok, ok.
Tanto qua c'è proprio quel livello di umidità.
Va bene, allora.
Lasciamo vedere un attimo.
Sì, esatto. Magari annuncio io allora.
Sì, aspetti che stiamo. Scusami, è sporchissimo. Faccio io, dai. Che, forse non serve mettere. Password? Sì, che poi la vede quando vuole lui. Già, no, adesso l'hai connesso al. HDMI? Sì, no, guardavo se. Allora lo devi fare prima e poi lo apriamo? No. Deve essere già quella lì, quella più recente. Questa vero? Qua? Il tomo più presentazione, sì. Allora, un attimo solo.
Ma ce l'ho in carta, la prendo io.
Sì, chiudi pure. Vediamo, ancora non si è staccato. Posso abbassare tutto? Se no chiudi tutto. Si sarà bloccato, aspetta che adesso. Sì. Ok, mi serve qua. Questa luce qua deve essere proprio. Sì. Perché se no. Sto sudando 7 camicie così. Infatti. Non ho guardato neanche il cambio. No, quelli credo che non sia in essi. Ah, lo fai tu? Fai le stendi? Sì. Ah, giusto. Poi lì se si vede l'estensione di là. Tu? Se non c'è qualcuno.
Lui sarà il nostro chair della sessione.
No, ma anche se non fai domande va bene lo stesso.
No, noi le facciamo da giù le domande. Le facciamo da giù le domande.
Ah, ok. Benissimo, sono proprio contento.
C'è il programma stampato? Giusto per dire.
No, c'è la diapositiva. Perfetto, così. Il nome ve lo facciamo. Un'altra cosa. Per andare avanti, andare con la. Facevo io, sì, grazie. Se no ci colleghiamo l'avanzatore. Tra l'altro secondo me non sono 56 perché alcune secondo me le salta, almeno le vedo nella presentazione che non ti so dire quanto duri. Se mi volete fermare a un certo punto. No, allora magari la tiro veloce e lo cambiamo. Premendo il tasto con la barra, sì, va bene. Per tornare indietro nel caso? Le freccette. Avanti e indietro. Ok. Parlo di Tomo che un po' all'inizio, la tecnologia e dopo l'HTX. Cerco di andare, perché non so, a me anche 20 minuti sono più che sufficienti. Cioè, tanto dovevamo partire. Partiamo magari tra 5 minuti, sì. Vediamo se arriva qualcun altro. Sì. Perché poi quelli che sono lì non si vedono. Se c'è qualcuno in galleria. No, nei parchetti sono i poster, mi ho fatto questi poster. Sì, c'è qualcuno però, siccome sono al buio, non si vedono. Sì, secondo me stanno a scrivere. Il tempo era alle 50. Quello che dico, io purtroppo non ho avuto tempo di provarlo da dire Guarda, dovremmo alle 5:20 dovremmo fermarci. Sì, sì. Sì, sì. A parte io poi vado a metterli perché che ridi? Sotto mentite spoglie. Possiamo andare, Alessandro?
Sì.
Ok. So, good afternoon, everybody. I've been hired to chair this session. From the original program was Bruno Combates that couldn't get it. So Alessandro Tellarini will tell us about, from TomoCube, will tell us about this holotomography technology. So no stains, no labels, just truth. See what you've been missing. Grazie Alessandro,
Okay,
and
thank you so much.
Enjoy this presentation.
Thank you for the introduction. My name is Alessandro Tellarini. Nice to meet you, everyone. I work for Schaefer Southeast Europe, Italy. We are the Italian distributor for TomoCube devices. TomoCube is a South Korean company. They work since 10 years. Actually, they are having the 10th birthday on Monday. You can follow that what's happening via LinkedIn. There will be some excellent news. So today we'll speak about holotomography, and holotomography has got an extra way of doing label-free imaging. It's a proprietary technology. It's a mix of tomography and holography. Now, as you can see, there's a CAR T assay where CAR T cell attacks a cancer cell. These are not fluorescence images. These images are obtained by the index, the refractive index of the structures contained into the cell. So the sample has got 360 degrees of light beams, and the system collects the information about phase imaging and works them into tomograms, and the software made the tomograms at 360 degrees. So this is a real 3D imaging. Just to give you an information technical spec, the HTX1 has got a 140 micron zeta stack. So you can imagine also not all the organoids, but a very wide range of them. TomoCube started in 2015, but their first, sorry that I didn't put the glasses on. The first device was put into the market in 2016. This was just a holotomographic device without fluorescence. What they understood almost immediately was that they needed fluorescence to confirm what they were seeing with the refractive index. And what they confirmed was that the refractive index was perfectly fine. So they could put color on the structures, like different colors based on different refractive indexes. There is a limitation that I can tell you right now, that not all the structures into the cell can have different color, because, for instance, organelles inside the cell has got the same refractive index, more or less. So they had to introduce a fluorescence engine into the new HT2H in 2016. And so they maintained the fluorescence engine with the new device, which is the STX1, which is multi-well device. So you can measure until 96 samples at the same time. And 96 samples, if I remember correctly, takes 30 seconds, but we will see it in another slide. So conventional holotomographic can be classified into two classes. One is typical with coherent light. The first HT tomocube had the immersion objectives, and this was kind of difficult. So a microscopy skilled user could use the device with no issues, but like a rookie couldn't. I mean, a person who doesn't work with microscopes all the day or is not so skilled. So now with the STX1 is super easy to take imaging. And so another thing that made them change the light source to the LED, to the low coherence, as you can see in the slide, the holotomography STX, is the fact that with the LED light, there is no speckle, which is another problem for the quality of the images that you get from the system. Okay. Sorry. Okay. So the principles, I try to be very fast. So bright field, so let's see what are the most common imaging technologies. So bright field is a very old technology. The problem with the bright field is that detects the color of the light. So you have to color the samples. Phase contrast microscopy visualizes the thickness of the specimen, so there's an advantage in observing transparent cells without staining the sample. Fluorescence, you have to stain the sample. So for instance, you lose some structure of the cell when you imagine cells with fluorescence. And because of the staining of the cell, it can have a short life. The principle with holotomography, with the tomocube devices, is that there's an incubator, so you can see what happens overnight. Overnight, I mean, is the shortest time of acquisition. We have experiments that has gone through like weeks. So it's one of the main specs that makes the tomocube unique, I would say. The first one is that it's real 3D measurement. I mean, sorry. So these are what happens. I mean, the problems that you can have when you work with 3D live cell imaging. So sample preparation, phototoxicity with the fluorescence, the fact that the measurement is qualitative, while the measurement with tomocube is quantitative. Just to give you an example, I can see that the nucleus is growing up, but I don't know how much. With TomoCube, you can get that number, like it's 35%, for instance. Okay, this is just a table. So we can see different cells with different technology. So you can see, for instance, that it's very difficult to see something with the bright field imaging. You see the stained cells with fluorescence microscopy, and you see the whole structure with the holotomography. Same here, so we have, like in the middle of the slide, we have the holotomography, the cell acquired with holotomography. You can see here like what's the real difference. So bright field shows the cell, but the resolution is low, even if we are imaging a single cell. So you can only imagine what's the difficulty in imaging a 3D sample. Holotomography can resolve every particular of the cell, including organelles. This is the principle, so you can see the sample and you can see the wave of light that passes through the sample is collected by an engine in the microscope and is collected from 360 degrees. So you build 360 degrees of tomograms and then you put them together and you have the 3D image. If you can see the next slide, the next slide, here is a cell and the user decided to see what's inside the cell at different depth, like 0 micron, 2 micron, 4 micron. So what are the advantages of holotomography? So no invasive labeling, safe monitoring, so it means the incubator, faster acquisition speed. So for instance, the minimum acquisition speed of the new devices is like half a second per image. For some 3D holotomography, this is the best that you can get. It's not the mean velocity, the mean acquisition time is the best that you can get. So it's not always like this. So this is the new device, HTX1. So just a fast explanation of what happens. So they decided to use the LED, low coherent light. And as you can see, the sample is irradiated by light beams from like 360 degrees. And so, as I told you, you can get tomograms and acquire them together and reconstruct them into a 3D redistribution image. Yeah, as I told you, the most important part of this slide is that the light is a low coherence light. So this is an acquisition, so you can see all the part of the cell. So we can see every single subcellular structure. There are no interference pattern, no speckle. As you can see, that's the benefit of the LED light. So let's go to the next. This is the HTX1. So you can see all the specifications listed. So the glass goes down, the black glass goes down, and you put the sample into the microscope, and it takes, like as I told you, a very short time to get the final image. And you can also put the sample into the incubator and leave there like hours or days or even weeks. It depends on the cell. There's the new HTX1. The idea behind this microscope is that you can get images with high contrast, with low coherence, and you can work with multi-well plates. So another nice feature is that you don't need to calibrate, whereas with the previous device, with the H2TH, it works with immersion objectives, needs to be calibrated, needs a skilled user. So you can also set up protocols, and you can use the stage to generate a preview image and then choose where you will acquire your image. You can also choose to acquire, to image the whole stage. You just have to tell the microscope and it will get images to cover the whole stage, and then it will just do stitching, sorry. So as you can see, here the device works in zeta stack. So it's going to zoom to magnify, and you can see the lipid droplets. You can see the lipid droplets and then demagnify from. So you can move through the image and see what you got. Okay, this is about the well plates. What's important is that the bottom is transparent, so it can be glass or polymer, doesn't matter. There are the Tomo dish, but you can use also commercial plates, and also TomoCube HT Ready 96 well plate. You can use a microscopic slide. It's open to many different means to get your imaging, to obtain your images. Software is very user-friendly, stage is fully automated. You can also set up different protocols. You can see in the bottom right image. You can choose to do holotomography every, for instance, 5 seconds, 10 seconds, 20 seconds. It's up to you. This is an example of imaging. These are time-lapse. So we have collected a lot of images and put them together to obtain a video. On the left, we have the differentiation of adipocytes with images took every hour, every 20, 30 seconds. On the right, there are hepatocytes took every 20 seconds. And there's, as you can see, super long term. As I told you before, you can decide to do time lapse overnight, or you can do time lapse for like even a month. It happens regularly on a regular basis. So it's something that you can do with the incubator and the very low phototoxicity of the TomoCube fluorescence engine. This is just a table, is in an examples of different application of HTX1, where they have quantified the effects of different drugs on cells without any staining. So as you can see, you can use the 3D fluorescence. The gray level of holotomography is below the fluorescence. That's what you obtain with the holotomography, and then they have applied the fluorescence. So we just have confirmed the object. As I told you before, the fact they introduced the fluorescence in the first step to confirm what they were obtaining with the holotomography itself, with the staining, the coloring, giving color to the structures based on the refractive index. This is the HTX1. It's got HTX1 Plus, sorry. This has got field of view is bigger, and there's also AI. It helps to establish protocol, to differentiate structures, to select them. So, as I told you before, I told you of wrong information, obviously, because it was really unreal. So, HTX1 Plus analyzes an entire 96 well plate in under 30 minutes, which is, by the way, is a very short time. We have the external fluorescence mode. There's also the possibility to add an external confocal spinning disc. So you can add the confocal imaging. So I will go right to the image just to so you can see the quality of the fluorescence with the confocal mode. We have a collaboration with Crest Optics. This is the top-level model of the HTX1 catalog. It's really new. We're working with the Italian company Crest Optics, and this gives you the best fluorescence images that that TomoCube has ever obtained. Oh, you can see. I'm almost done, so you can see what you get here, like very fast. This is a live worm. Is the C. elegans. So this was live in the well. And there are so many applications. So thank you very much for your attention. And I just want to tell you another thing. There's a new microscope coming soon, and it will be revealed on Monday because there's this celebration for the 10th year of TomoCube company. So you can connect to the page of TomoCube in LinkedIn. There's a link to register to the event and see what's new. We honestly don't have a clue. So we don't know if it's like a higher top level or if an entry level with a nice price and almost same features. If I can talk about price just a second. So the entry level price for the HTX1 is 220K euros. The HTX1 Plus is 260 and 280. So Crest Optics is around 320. So you just have because it's a confocal and holotomography. So I think it's a very nice price. So thank you again. Thank you for attending the presentation.
Thanks, Alessandro, for this interesting presentation of the instrument. I don't know whether somebody has questions. I have one curiosity, probably you said, but in case I missed, the thickness of the specimen that you can still...
The Zeta stack?
Yeah.
The Zeta stack is 140 microns.
So what happens if you have a thicker specimen?
Well, Bruno would have answered your question, but honestly, I don't know, to be honest. But if you come to my booth, I can take your, like all of you, I can take your information and put you in contact with our specialist. Unfortunately, he couldn't be here because he's busy with another congress. So we have like 3 congresses this week. So. Okay. And Bruno couldn't be here, but yes, the limit is 140 microns. We know that not all the organoids fits this thickness, and so this is one of the limitations. This is why I said I don't know if the new device maybe can go higher than that, It can be. So it's
Okay.
really, we will have to wait until Monday to see.
Thanks. Other question? Marco.
Oh, thanks for the
Professor
presentation.
Thank
so I'm waiting for the Tomo Cube for 1 millimeter, 1.4 millimeter. Would be great to have imaging for thick tissues. But anyway, no, it's a great equipment. Just a curiosity about recommendations for the media culture, because I see that you can do long-term, basically time-lapse imaging. But is there any recommendation for the culture medium? So you can use standard medium or, I don't know.
No, there's no recommendation.
Okay.
Obviously, if you use fluorescence, phototoxicity is a factor, but so it can limit the life of the cell. But other than that, I don't see any other guideline about the incubator. So it's just we have a video. If you come to the booth, we have a video, and in the legenda you can see how long does the incubation was, 20 hours, 2 days, and every second, every 20 seconds you get an image. So then you build the time-lapse video, and to be honest, I don't think there are limitations to using the incubator. But
okay.
Feel free to ask. So you can come to the booth, same as before.
Okay.
So this is not like I'm not 100% certain on what I want to be honest, 100% honest with you. So I don't know. It's better if I put you in contact with people who knows better than me.
Thank you so much. Thank you.
Thank you.
Any other question? So if not, thank you, Alessandro, again.
Thank you. Thank you. Thank you, everyone. Sì, sì. Grazie Massimo, io ho fatto quello che potevo. Ho fatto quello che potevo, ma l'ho saputo venerdì. Grazie. Allora, c'è anche il coso del... Eccolo lì, c'è il mouse in tasca. Grazie, ciao.
Questo mi? Questa finestra blu si vede qua. Insomma. Tanto fino alle 5:40 non c'è nulla. 5:50, 5:40. 5:40.
Sì,
5:40 dovrebbe iniziare la prossima sessione.
che è uno solo. L'ultima.
Sì.
L'ultima lettura e poi finisce.
So. Io devo finire.
Dimmi.
puntatore, quindi se deve pigiare il primo. Ok, vabbè, quando c'è la presentazione.
No, ma
Questo è per
non
il
sono io, ma
puntatore. E questo è per andare avanti.
vabbè,
tutto
ma
qua.
quello è basta, cioè, quindi...
Sì, no.
Ma porterà il suo... no, penso che avrà il suo computer. Perché la presentazione non me l'hanno data. Userà il suo computer, gli parla adesso.
Ah, forse è andato al
Sì.
centro slide allora.
No, perché mi hanno caricato una slide di tappo, quindi...
Sì.
Sì.
È il ciclo. Ah, vabbè, possiamo
Vabbè, ora vediamo quando arriva.
Allora, ci dobbiamo dare questo. Vabbè. Allora, noi siamo. Spero di sì.
Grazie.
Ce l'ha l'adattatore? Sì. Meno male che l'ho portato. No, lo troviamo sicuramente. Se ce ne ha... Sì. Sì. Perfetto.
E magari prendo anche l'alimentatore?
Se c'è... Vabbè, al
Avete
massimo.
qualcosa?
Ok. Allora, ho collegato anche questo. Arriviamo laggiù? Sì?
No.
No, c'è un ricordo.
Ok, magari così. Vabbè, così adesso vado?
Vedo un attimo se c'è. Ok, perfetto. Sì, allora l'avanzatore, questo per andare avanti.
Sì.
L'unica cosa che non funzionerà è il puntatore.
Che lo faccio sul schermo.
Sì, È perché no, perché non è installato il software, questo è quello lì digitale. Però lo può fare con il
Però con
mouse.
questo mouse
Sì,
riesci.
sì, sì,
Allora,
sì,
l'altra
con-
cosa è che la fotocamera perché sono molto alto. Magari controlliamo.
Sì, ok.
Sì, questo non si può.
Per me sì, ma dica lei. No? Sì. Avevo ragione.
Purtroppo l'hanno inchiodato perché è abitato, quindi non posso
Questo mi fa un po'
toglierlo.
paura, se mi agito.
No, 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 è abitato.
Ma se io cado,
No, no,
nell'agitazione della presentazione.
l'unica cosa è molto largo.
Facciamo la selfie.
Cioè anche te. Siamo boomers. Ce la fa anche lei, ho detto, ho chiesto a lei se ci fa la foto.
La mando subito a mia moglie. La mando subito a mia moglie.
Inizio. So, please, if you take your seats, we can start. So it's really a pleasure to introduce Cornelius Gross, really a friend, first of all. Cornelius started his path from its undergraduate training. Started his path from Berkeley, where he did his undergraduate studies in biophysics and spectroscopy. Since then, he went to Yale for his PhD, and then he became a teacher in New York City, so going out of research. And probably this also increased his skill in communicating also with people that are not really scientists. But then, luckily, I would say, he moved back in science, and he joined the laboratory of René Hahn at Columbia, where he did the first discoveries about mechanisms of action of antidepressant and the role of serotonin in the development of the anxious behavior. In 2003, he moved to Italy and to the EMBL outstation at Monterotondo, where he set up his own lab. And actually, I think this was really a fortune for the Italian neuroscientific community, because he in addition to being a great scientist, he was able to really participate to the Italian community of neuroscientists. And his contribution is really important in improving our community, also modernizing our community. And in Monterotondo, he continued to work on serotonin, on the role of this neurotransmitter, for instance, in maternal care. But typical of him, he started other curiosity-driven, let's say, research that were very, very fruitful. For instance, he was one of the first to discover the role of microglia in shaping synaptic rearrangements during development. And then, again, he decided to study the hypothalamic circuit for social or predator fear. So you see how he is not, scared in trying to challenge new field, and has been very successful. Indeed, just to mention that he got 2 ERC advanced grants, and he, of course, got funding from NIH, when there was still some funding from NIH, and is now head of the MBL outstation in Rome. So I'm really, really, very happy to have him at our meeting. And please join me in praising Cornelius.
So thank you, Tommaso. Thank you. And I have to say, reflecting on what you say, it was the best decision of my life to come to Italy for many reasons, including science. And it's great to be at this meeting because it's just a reflection of that community, and it's fantastic to see friends and acquaintances and meet them here. I also want to thank the organizers, Monica Di Luca, I think, who really took on the leadership of organizing this event. Thank you. I know it's a lot of work, and she had many people around her, so thanks to all of you for making these things happen. It's really important that people take out time to contribute to the community, and it's very appreciated. I thought I would take just a few moments, I think I have 2 slides, to tell you something about the institution where I work, and that's because it's an unusual part of the Italian landscape, research landscape, and I think often I find people are not aware of the opportunities and the resources and services that EMBL offer to the Italian community. And I just want to start by telling a little bit what EMBL is. It's Europe's Molecular Life Science Institute, so we're an international organization. We have sites in 5 countries. We have 6 sites. And some of the unusual features that EMBL follows, if you're not familiar with it, is on the one hand, we do both service work, so service to the research community. In fact, 50% of the staff at EMBL is involved in providing services to the research community, and the other half is involved in doing fundamental basic research. Maybe that might be the part you know more about. But this service part is very important, and that includes work, that includes core facilities that are open to the public globally. It includes all of the data science tools and databases that we manage. We're now the leading global repository for molecular life science data. You might know AlphaFold, for example. That was a collaboration with Google DeepMind that was based entirely on EMBL's commitment to open data sets and actually is presented to the world by EMBL. So I think I put down some of the data here just to say each day there are more than 100 million hits to EMBL websites of people who are seeking information that we make openly available, and we have a long-term commitment to open science. Also, we have a somewhat unusual staff structure and contract structure. We hire people for essentially a maximum of 9 years. We provide our research groups with internal core funding, and we, as a result, are seen as a place where people train. So all over Europe in particular, but all over the world, there are people who did time at EMBL, and some of them are in the audience. And the idea is that one of our values to the community is that we give back people, actually. People are trained, people who spend time at EMBL, and there's many institutions all over Europe. I've listed some here that were founded by people who left EMBL, and in many cases that have taken on some aspects of the EMBL model. The Max Planck Young Group Leader, for example, system is based on EMBL and was started by people leaving EMBL and starting that at Dresden, for example, and so on and so forth. Janelia Farm mentions EMBL as one of the models for its mode of operation. Okay, so that's just to tell you a little bit about why we're slightly different. I think that also influences how we operate. EMBL is known for being collaborative. Because we have young group leaders who have a limited period of time and have core funding, they're seeking to interact with other people. And so it has this kind of atmosphere of encouraging people to work together. I also just want to remind you, we are publicly funded, so the money comes from taxpayers, it comes from you, it comes from 29 member states that voluntarily join EMBL. And in that sense, we're part of the Italian landscape. We work together with Italian institutes and national institutes across Europe. So as I said, we're a distributed system. We have this site in Rome, where I work. We have 6 sites across Europe. The big sites are in the UK and in Germany. And the site in Rome, we're outside of Rome, about 20 kilometers, Monterotondo, we're focused on mammalian systems physiology. We focus a lot on the use of laboratory mice as a model system. We're very interested in environmental adaptation, organismal adaptation to the environment. We do a lot of technology development, a lot of collaborations with imaging companies, for example. And I just want to highlight at the end a new development, which is being spearheaded by the CNR, which hosts us in the Monterotondo site. We've just completed a feasibility study for a very ambitious research and training partnership and center on that site, and we hope that we can tell you more about that in the coming years as we move ahead to try to make this a reality. And I think this will be very important for, hopefully for the Italian landscape, but also for EMBL to really maximize Italy's leverage of what EMBL has to offer. And just finally, there's a number of job openings, maybe to highlight in particular our postdoctoral fellowships. There's 2 of them that are internally funded. They're co-funded by the European Commission, but they're actually internal EMBL fellowships. One is for interdisciplinary research, it's called iPod. We have calls every year. And the other one is for something called Arise. It's a new fellowship. It's focused on technology development. So if anybody's interested in pursuing a technology idea, you're more interested in technology than necessarily biology. That can be from software to optics. Come check it out. Check out this opportunity. It's quite unusual. It funds you for 3 years. And also just to point out that these fellowships can be done in collaboration with national laboratories. We're also looking for new group leaders. So anybody's interested in potentially launching their independent career at EMBL, we will have our next one probably next year. Okay, so I wanted to start my talk by just emphasizing the importance of anatomy to understanding the brain. I'm a big believer in the importance of understanding function based on structure. And this is just a little bit of an illustration of how sophisticated and how complex the brain really is. And this also started my journey, as Tommaso mentioned, deep into the brain, into the hypothalamus, into this ancient part of the brain that we share with very simple bilaterian organisms. It's the brain, forebrain, cortex, striatal, nigral pathway, striatal pathways are all things that were invented much later. In the beginning, there was hypothalamus. And that journey actually came out of a realization of, and a kind of revisiting of the kind of old classic literature on the site of control of aversive behaviors, defensive behaviors, behaviors that are associated with fear states in humans. People and animals also sometimes call them fear behaviors. And when we think about the anatomy of fear, typically we think about the amygdala. That's the first area of the brain that comes to mind. And I just want to tell you that that is true, in fact. This is probably here, I'm presenting probably the most compelling evidence for the importance of the amygdala and fear. This is a subject who had a neurodegenerative disease that was selectively affects the amygdala. And that person had a dramatically reduced response, emotional response to film clips on the left side that contained emotional images that were related to fear, but emotional responses of other types were not affected. Also, that person, when they were taken into, it's actually a fascinating paper. If you haven't read this, it's really worth reading because there's lots of details in the paper about how they took this person to various real-life situations. They were taken to pet shops and to haunted houses, and it makes for a great read. It's just a single person. And she then proceeded in the pet shop to actually immediately step up to try to take the snakes out of the cages, and she wanted to touch the spiders. And in the haunted house, she laughed when all the other people in her group screamed. So it seems the amygdala is, in fact, very, very important. It's central. It's essential for humans to actually develop both fear emotional states and also aversive states, in other words, the avoidance that goes along with these fear responses. But there was an interesting follow-up paper, partially from the same group, where they decided they would try to use a somewhat different challenge, a somewhat different stimulus to elicit fear responses in this subject. And by that time, they had actually accumulated 2 other subjects, so they had an N of 3. And what they did was to challenge the people with an inhalation of CO2. And I don't know if you've done that before, if you put your head into some dry ice bucket or something. It's kind of aversive. Eventually, you feel like you're asphyxiating. And people typically have an aversive response, and some people who have particularly people who have a history of panic disorder and anxiety, they actually go into having a full-blown panic attack with hyperventilation, and typically they rip the mask off of their face. And what they found is that these individuals with amygdala lesions, first of all, they had a strong emotional response, this aversive response, which was at first surprising, given that they didn't have other types of responses to threatening situations. And secondly, they had this exaggerated response that was very similar to people who had a background of panic attacks. So what this is saying, or at least saying to me at the time when I saw this data, is that amygdala is essential for these sort of responses, but there must be some other, presumably a downstream structure that's actually responsible for creating or is able to create the emotional states and the aversive responses and avoidance responses that come with these sort of threatening challenges. So potentially the amygdala is but a conduit, a sensory conduit for these stimuli that are later then actually formed into some internal states deeper in the brain. And that brought me to the work of Newton Canteras. Who's one of my heroes, an anatomist. He works in Brazil. And he studied for many, many years the connectivity and the anatomy of the medial hypothalamic system. And I'm just showing you a very simplistic cartoon here. This system, which he associated with predator defensive responses. He studied rats who were exposed to cats in the laboratory. This pathway begins in the amygdala, just as we would suspect from the human data, and then passes to the medial hypothalamus, which is a series of interconnected nuclei. And from there, its major output is the periaqueductal gray, well known to be involved in producing flight and freezing. And from there down to the medulla, to the motor system, and to the muscles to produce behaviors. His work is all based on CFOS activation and PHAL track tracing, so classic neuroanatomy. And he essentially figured out this pathway and assigned it to responses to predators. And so that's where we started our studies. We first set up a behavior test. We wanted to do this in mice, so we could use all of the molecular genetic tools in mice. We exposed them to rats. We did that in mice, in fact, to get these really strong, powerful responses. I'll show you in a second. You need to highly habituate the animals, so the animals have to actually live in the chambers. The animals live in this chamber A for several days, and each day they're exposed to this corridor in the far chamber. And then on day 4, they find out that in the far chamber there is a rat. These are natural predators of mice. They will kill the mice if you allow them to directly interact. And you can see then this classic flight behavior and freezing behavior, which are the typical rodent fear responses that we can measure in the lab. Also, we added a day 5 to the protocol as a kind of memory day. So we actually looked 1 day later in the absence of the rat. We removed all the olfactory cues, and we found again that there was a sort of a contextual fear response. It's somewhat weaker, obviously, but we can also measure this sort of this quantification of a fear memory to the context. What does it look like? I hope you can see here. So this is an animal, a mouse here, who's in his home chamber. He is going to be exploring the corridor. He's moving out. You can see already the mouse is in this stretched, classic kind of defensive stretch approach posture, which you can measure. And then the animal, when he gets close to the rat, makes this decision to escape back to the safe chamber. And this is the behavior we can measure and quantify in the laboratory. We do all the experiments in the dark. We just turned on the lights so that you can see it better. Okay, so the first classic experiments was to go in. We used dreads at the time. They had just become available, so this was very convenient, and viruses, and we essentially blocked various nodes in Newton-Canteris' pathway and actually successfully showed that they were essential for these defensive responses of mice to rats. So that was really comforting. Newton's anatomy actually turned out to make sense. And we then proceeded to dissect in a bit more detail, now using this memory feature, right, to understand whether animals required these nuclei also to respond or form memories of fear. And that's important because when you do contextual fear memory, the stimulus is no longer the predator. It's no longer a kind of innate simplistic response. The stimulus now is the context, which is a theoretically neutral context, which has acquired some aversive properties. And so we actually also were able to show that the medial hypothalamus, I didn't tell you this yet, but we're actually focusing on the kind of one of the major nodes, this ventral medial hypothalamus, that this is important not only for the production of the acute defensive response, but it was also important for the production of the defensive response to the context. So this suggests that this medial hypothalamus also is able to process information not just potentially from amygdala areas or areas that bring sensory information, but other areas, memory areas like, for example, the hippocampus. And I'll talk about this later, but actually there are direct connections. One of the major outputs, in fact, of the hippocampus is through the lateral septum, and it's mapped exquisitely onto this ancient hypothalamic motor system, both for approach and avoidance systems. This first data also, I think, allows us to sort of make a statement, sort of categorize the amygdala on the sensory side, right? It's required for the acute response, and the medial hypothalamus, on the other hand, is already moving to another node where it responds both to immediate predator sense cues, but also to contextual cues. And then if you go to the output, to the periaqueductal gray, we actually found this is important for the production of the behavior, but is actually surprisingly not required for the memory. So you can block the PAG, the animal will not show the defensive behaviors, but they're perfectly able to produce a memory and then express that the next day when the PAG is no longer blocked. And so this really suggests that the PAG is already on the motor side. It's really about the output, right? So blocking the PAG is like giving a muscle relaxant, right? You just can't express the behavior, but you still are able to create the internal states. And so we have the beginnings already of a kind of hierarchy, I think, of synaptic connections, and these are connected with single synapses, synaptic connections between these structures. Okay, what about humans? Unfortunately, there's very little data on humans implicating these regions in fear behaviors. It's essentially restricted to a small number of electrical stimulation studies where humans have been electrically stimulated, right? This is typically in preparation for surgery to remove brain regions. And people have stimulated what we defined and Newton-Canteris defined as the predator part of the medial hypothalamus on the left. This single individual, was a paper published in 2010, actually reported an intense emotional response to this activation, and then went on to show hyperventilation and show a panic attack. So in line with the idea, at least, that this structure is somehow able to produce aversive and also, importantly, emotional responses. Here in the field, I'll just throw out, there's some controversy about whether the hypothalamus is just an area for the production of physiological responses to threats, or whether it's also able to produce emotional states. And I think this is strong evidence for its ability to produce conscious emotional states. PAG, right, which is this motor output area, you also get fear-related responses. They're a little different. You know, you can speculate. This person described how they felt like somebody was chasing them. You know, I would say that's more motor-like, but I think this requires more work. So perhaps these areas are also critical for human defensive responses and internal states. So we need to go in and understand, of course, not just whether these areas are required, but how do they process this information? How do they encode the information? And then what we wanted to ask is, how do they transform that sensory information into a, hopefully, exquisitely controlled and adapted, ethologically adapted behavior response, a decision to flee, for example. And when we recorded, now these are single units, so it's single unit electrophysiology recording in the medial hypothalamus, this VMH, the predator region, in animals that are approaching a rat. And what we found was a major population of cells, we called them assessment cells, which increased their activity up to the point where the animal made this decision, and then the activity of those cells precipitously dropped and then came back to the baseline levels during the time when the animal was actually escaping. Actually, we showed that the activity is directly proportional to the distance to the rat, and so the idea somehow is that this might be encoding the sensory threat information that the animal is receiving, probably multisensory information. But interestingly, the animal shuts off, or these neurons shut off when the animal makes this decision to act on that threat. We also recorded in the periaqueductal gray, and here we found a major population that looked a little bit different. They were actually quiet, or they fired at baseline levels all through the approach period. And then at the period where the animal made this decision to flee, you saw this dramatic increase in firing rates, which then eventually subsided. And the peak of the firing is actually before the peak of the velocity of the animal. So you see this kind of firing of the neurons, it comes down, and then the animal takes off. So it's in line with the idea that the PAG is sort of a trigger structure, potentially, functionally, for these flight behaviors. And so if you put that together, in the kind of first synapse or first structure, you have this buildup of sensory information, of threat information, until the decision point, and then in the downstream target structure, you have the expression of the motor response, or an impulse that presumably would then lead to the motor response. The problem with this simple model, I mean, we were very enticed by this idea, right, that you would build up the threat and then you would threshold that threat and in a nonlinear manner then make a decision to trigger a flight response. The problem actually was that we found in both regions also a second major population, so that both areas had assessment cells and flight cells. And we can talk about that if you like. I have some speculation. I just think it's interesting that you see a distribution across multiple brain regions at multiple levels in this behavioral control hierarchy that seem to encode important salient features. And I think it also relates to a lot of the work that's come out now, where people have really started to look more unbiased across the brain and have found encoding all over the place of important salient environmental stimuli. So I'd be happy to talk about that more. But it appears that the brain has at least the information potentially for making decisions at multiple levels of this behavioral control column. So if you put those together, if you just superimpose them, right, there's this kind of we're calling it an escape switch. It's associated with the escape decision, and there's a switch in the firing activity. Essentially, all of the firing in one set of neurons suddenly shows up in the other set of neurons in the same nucleus. And we know now that those neurons are intermingled in the core of this behavioral structure. And I won't go into more. We have a paper where we tried to develop some theoretical models, some computational models, to see whether we could recreate, at least, with a certain kind of feedback inhibition, feedback excitation, switches like this, nonlinear responses, and we can. And we're trying now to take some of the lessons from those and then, of course, test them. I mean, a model is not useful unless it actually makes predictions that you can then test to improve your model. But so far, I've been telling you about predator threats, right? We know what they are. We've thought about them. Maybe in a movie, we've experienced predator threats. Maybe some of us actually experience predator threats, maybe a dog or maybe other sorts of animals. But typically, humans are no longer exposed to predators. What's really important for humans is social threats, social interaction. I'm very convinced that social interaction is just at the pinnacle of what our brain is adapted to do. Knowing what we can get out of other people is extremely important for us, and we're constantly scanning to try to understand how do we relate to them, how do we bond to them, what possibilities are there with other members of our own species. And threat is a part of that assessment. It's a very important part of it. So is this system also involved in processing social threats? And so already at the very beginning of our studies, we had set up the same behavioral assay with a social threat, which in mice really works well. Among male mice, they're very aggressive. And we placed an aggressive, we use CD1 mice that have been selected to be aggressive, into the far chamber. There's a short fight between the mice, and then we allow the experimental mice to escape. And the mouse will show identical defensive behaviors, so repeated approach avoidance behaviors to that social threat. as they did for the rat. And so we're measuring the same behavior, but now we have a conspecific rather than an extra conspecific stimulus. And to our confusion, initially, we found that blocking the medial hypothalamus had no effect whatsoever on social threat responses, shown on the left. And after a bit of thinking, actually, and looking at the literature a bit, we realized that there was another pathway, actually a parallel pathway, had been described also by Newton Cantaris, largely non-connected to this defensive pathway. He called it the reproductive network, because it had been previously associated, or some of the nuclei had been associated with lordosis behavior in females, so preparatory sexual behavior in females. Actually pioneering work by Don Pfaff at Rockefeller, who really did sort of the first molecular behavioral neuroscience work back in the 70s. And also, more recently, it's been associated with aggressive behavior, so aggression. And we wondered whether that system might be important also for defensive behavior, so social defensive behaviors. And actually, when we block that region, so this is now the ventrolateral part of the VMH rather than the dorsal medial part, shown on the top left, we block that. We could actually block social threat responses, and we had no effect on the predator responses. So we had this double dissociation. So we were very excited about that, just the idea of being able to identify potentially independent pathways for conspecific and intra-conspecific and extra-conspecific fear, to me, also has importance for psychiatric disorders. I don't think this is something that has entered into the psychiatric kind of literature or thinking. What about in humans? Well, in the meantime, we were working on this actually a couple of years ago, 3 years ago. A very interesting paper came out, again, an electrical stimulation paper in humans by Joe Parvizi's lab at Stanford. And he had a patient in which one of the many, many electrodes, I think 115 electrodes, was placed into the ventrolateral part, so into what we defined as the social threat part of the VMH, close to the tuberal nucleus. And this individual, in response to stimulation, again had this intense feeling of emotional response, constriction of the chest. And when they were asked afterwards to describe the emotion, interestingly, it was somewhat different than the stimulation in the predator part. This person described an intense sensation of shame and embarrassment, along with sadness and fear. Now, I don't know, I assume you have all, or most of you have experienced shame. It's considered by psychologists to be one of the most powerful emotions. If you want to get somebody angry, or if you want to, expose somebody to something very aversive, find a way to shame them. And it's intensely social. It really depends on the gaze of others or the perceived gaze of others, and it's intensely negative. So it's only a single individual, but I think this is interesting, I think, and potentially lessons we learn from these highly conserved nuclei in mice could be relevant also for these emotional states in humans. And I think it also reinforces the idea that the medial hypothalamus is able to produce conscious emotional states that are relevant to humans. Unlike some who have argued that it is strictly a physiological output that's associated with defensive states or threat states. All right, we've gone back into the social part and the ventrolateral part. We've recorded their single units. We find, again, assessment in flight cells. This is recent work from a PhD student. So it seems to encode these decisions to flee in exactly the same way, which is really interesting. But in the process of recording there, we also, in this case, we've done this with miniscopes, which have certain advantages because you can look at the same cells in a more reliable way across many days. And you can also record more of them in these deep brain nuclei. It's just much easier. We discovered or chanced on something that was unexpected, and I just want to set the stage. Those of you who have worked in the hypothalamus, the idea is the hypothalamus is typically a sensory response region, primarily, in fact, pheromonal response region, the medial hypothalamus. It gets direct inputs from the accessory olfactory bulb, for example, rather than the main olfactory bulb. And so the idea in the field really was that these areas should be responding to acute exposure to some of these pheromonal or pheromone-related instinctive cues. And what we found was that they actually showed dramatic responses during this memory exposure, during this contextual exposure, when there was absolutely no rat or no social threat whatsoever. And I'm just showing you a couple here. We called them, so the most prominent ones are far chamber cells and home cells. So they really show this strong upregulation just at the boundary when the animal would enter into the far chamber or back into the home cage. And that intrigued us, because it appeared to be a sort of encoding of the context of some sort. And this is just showing you a different version of it. It's kind of a dimensionality reduction where we actually record up to 30 cells at a time. So each spot now on the graph is actually it's a frame in the video. And it's just to go to show that you can use the information, very much like place cell recording, right? You can use the information to tell you where the animal is. So you know that if the values are over on the left side, well, the animal's got to be in the far chamber. Okay, so that was intriguing. The first question we had was, well, okay, place cells form when the animals explore an environment. They actually usually form quite rapidly within the first minutes or hours or tens of minutes when animals are actively exploring. And so we decided to go back to the first days of our test because these animals have been highly habituated to this environment. So do these form on the first days the animals are in the apparatus? Or is this something that depends on later experience? And what we found, if you look earlier, if you look during the habituation phases, you find there's a dramatic reduction in this encoding of space, and in fact, there isn't a significant encoding of space. So it seems to somehow require the experience. So different than place cells, and I just want to, classic hippocampal place cells at least, just to kind of contrast the two, to sort of set up the next set of experiments. So we're tentatively thinking they're contextual or territory cells, and I'll tell you more about that in a moment. They seem to have larger receptive fields, which is this entire chamber. We don't know whether they respond to larger spaces yet. And they seem to depend on this, presumably on this defeat experience, rather than just forming spontaneously during exploration. And of course, they're in the hypothalamus, which is an area of the brain which people have not traditionally found, or not found and not traditionally looked for encoding of contexts. So to tell you a little bit about this, sort of part of this is now modeled. Some of these are going to be experiments aimed at testing this, but our initial idea was that this contextual information could get to the hypothalamus via this pathway I told you about before. So the hippocampus actually projects directly to the lateral septum, and as I told you before, has this kind of topographic mapping onto the hypothalamus. And the regions that then map onto the medial hypothalamus come from the ventral hippocampus, from a very specific part. They've been all mapped by work from Larry Swanson and his colleagues. And the ventral hippocampus is very interesting, actually. There's not a lot of place field recordings that have been done there. They appear to have much larger place fields. There's some really exciting work from the Mosers where they actually had rats on, 10-meter runways, and were able to find place fields in the ventral portions of the entorhinal cortex that were much larger, up to several meters. The second observation is that unlike the dorsal hippocampus, these areas respond dramatically to social interaction. There's a whole literature on ventral hippocampus social interaction. And then, as I said before, it has these major connections down to the hypothalamus, to the lateral septum and hypothalamus. And lesions of the lateral septum, for example, you probably know that, classic lesions of the lateral septum called septal rage. So these areas seem to be inhibiting some of the major outputs of the medial hypothalamus, like aggression and defensive behaviors, for example. So that's the hypothesis. And if we put that together, the idea would be that threat information would come from amygdala. There's many different inputs. We could talk about that, that bring various types of polymodal threat information. Some sort of contextual information could come from ventral hippocampus. Those would be integrated in some manner such that the medial hypothalamus would have traces of both, and that would then allow the animal to properly adjust its flight and potentially freezing behavior to the context. And these circuits would be able to make this kind of nonlinear thresholding of these inputs in order to support defensive outputs. And it got us to thinking about territories because, as you may know, mice are very territorial. And so typically in the nature, if you look at some of these papers where people have looked at commensal mice behaviors, a single aggressive male will dominate and actually patrol and control a territory. Usually there's a whole bunch of other males who live outside of the territory together, but you have these territories of the dominant males, and when another male comes into the territory, they will attack that animal. But if they themselves venture out of their territory into the territory of another animal, they will escape. They will show these defensive behaviors. And so our idea initially was that, well, maybe the VMH has been implicated in both aggression and defense because, in fact, it controls them in a way that's appropriate for the context. So when this switch, let's say, is on, in this simplistic kind of model, the animal would be, it's going to be on, or it's going to be particularly sensitive, or the threshold will be low when the animal is outside of its territory. When the animal returns to a territory, the threshold would rise, and the animal would be unlikely to trigger the switch, and would be more likely to support activity in the region, would support other types of activity. And there's actually some evidence. People have recorded during aggression, and this is actually what you see. You do not see the switch when the animals transition from social investigation to an attack. You see a sustained activation of assessment cells. So that's a bit the hypothesis, and there's various directions we're going. One of them is to try to set up territories, territorial behavior in the laboratory. So I had a postdoc spend about 4 or 5 years doing this, and these are some of the results. The beauty is that when you increase the space, I don't know if you've done this in the lab, but if you give mice space, they just start behaving in amazingly ethological manner. I mean, the behavior gets very complex, it gets sophisticated, it becomes very clear. So on the left side, you can see 2 animals that are matched, well matched by chance. About 10% of the animals show this behavior. They're showing all of the classic tail rattling behaviors, defensive behaviors, aggressive behaviors. They briefly venture into each other's territories, but they avoid each other in each other's territories. And I think on the right side, if I can start down the right side, you see what happens in actually the majority of the animals in this particular case. You'll see one of the 2 animals is dominant, and you see this really dramatic chasing behavior, aggressive, persistent aggressive behavior, where one animal is dominant over the other. And the idea, of course, is to do this in a manner which is compatible with electrophysiological recording and manipulating of neurons, which is something that hasn't been done before in these type of behaviors. And so it's appropriately small enough to do that, and big enough to show these behaviors in a robust manner. So just some of the things we observed, just looking at the behavior, so this is published last year, as you expect initially, so this is now the cases where you have one animal that's dominant. On the left side, I'm showing you sort of the data from the first 20 minutes. There's a back and forth, kind of the animals seem to be equals. They're both attacking, they're both showing flight behavior. There's very little hiding behavior, they're actively seeking each other. And then as you move forward, if I'm showing you the data from the last minutes now, by the end of essentially 2 hours or an hour and a half, you see this really dramatic, very clear behavior in which one of the animals is attacking, the other animal is primarily hiding in order to suppress those attacks. So there's very clear dominant-subordinate hierarchies. You can take all of those behaviors that you record and you can do a dimensionality reduction. Actually explains about 60% of the behavior, which is, so it's, I think really takes home the idea that it's very clear. And essentially you have 2 components. One component you could call sort of the dominance behavior, which splits the 2 animals. And I'm showing you lots of these pairs here connected. And then the other one, which is a sort of a sociability interaction kind of index, in which you can kind of you can explain the differences between the pairs. One of the observations we made, and actually this took us quite a while to figure this out properly, is that inbred strains, I'm talking about classic C57 black 6 animals, they do not show organized dominance and subordinate behaviors. So not only, I'm just showing up on top, so CD1 animals are outbred animals on the left, in the middle is black 6 animals. Very few of them show this split, but if you actually go and you look at correlations between the behaviors, for the black 6 animals, you actually see correlations, strong positive correlations between flights and attacking, for example. So they do show attacks, and they show flight behavior, but it lacks organization. It seems to be random. The animals don't fall into a kind of a persistent state, dominance or subordinate state. And luckily, because CD1 animals are difficult to use in combination with all the molecular genetic tools, we have found if you use F1 hybrids, where you can use these tools in heterozygosity, you get really nice, robust behaviors, look like outbred animals, and so we're actually working with these F1 hybrid animals. So just a lesson to the community a little bit, I would just be aware, some animals, obviously inbred animals, have lots of homozygous knockout. I think they have about 110 different genes that are homozygously knocked out because of stop codons in black 6, just to be aware of that. You guys work with knockout animals, which is 1 mutation, black 6 has 100 of them. Behavior is very different in these animals when you get to these more, particularly when you get to these more ethologically demanding environments. So the other direction we've been taking is to try to understand why social defeat seems to be required for the emergence of these contextual encoding. So I'm just showing you the behavior, the data again about the recording on the right hand, and just saying our idea here would be that, as I told you before, right, this contextual information would be projected by the hippocampus and septal system onto the medial hypothalamus. Medial hypothalamus would be a slave to that information. Plasticity over time would happen in the hippocampus, and the change in the encoding in the medial hypothalamus would depend on the hippocampus. Alternatively, you could actually have plasticity occurring in the VMH. Regardless of whether there's contextual information coming from the hippocampus, the VMH might not be receptive to that information, so it might change and only acquire that information or have access to it or use it after the animal has had a socially important salient experience, whose information presumably would come through the amygdala. So we decided to test that. So one way to test that is to actually test whether the VMH by itself can change its function before and after a social defeat. So we actually took animals. In this case, we're actually optogenetically stimulating them in the social threat part of the medial hypothalamus. And if you do that before the animals have had any sort of aversive social experience, so on the left side, so these are animals that are actually engaging in social interaction with another animal in a novel cage. When you stimulate in this region, you don't get any dramatic changes in behavior. If you do that 24 hours after the animals have experienced social defeat, those animals, when stimulated, will show a dramatic avoidance response. And similar data have actually been seen by other laboratories done in a somewhat different manner, but I do think this is a very robust response, and it really suggests that the VMH itself, it functions differently before and after social defeat, and plasticity then should be either in the VMH or potentially downstream or in its connections. So that suggests that the medial hypothalamus actually undergoes experience-dependent plasticity, and that really fascinated us. I think this is not the way, again, we've been thinking about the hypothalamus in the context of innate responses, which are typically supposed to be or thought to be or seen to be not particularly responsive to the environment. So where is that plasticity? What type of plasticity is occurring, right? We're not in cortical structures, we're not in the hippocampus, so it may be AMPA NMDA receptor dependent plasticity. That could be one type, could be another type of synaptic plasticity. It could be changes in intrinsic properties of the cells. It could be structural plasticity. And so we decided to try to take a kind of more unbiased, holistic approach. And we went back to a literature which argues that transcriptional activation of immediate early genes is actually critical for many forms of synaptic plasticity. And these are studies that have been done, primarily done in hippocampus and primarily done in vitro. Actually, there's very few studies that have actually tried to block immediate early genes and look at their contribution to plasticity in vivo. One of the most compelling ones actually used a knockout of SRF, serum response factor. It's actually been shown to be a master control gene for immediate early genes, and I won't show you this, but we confirmed this is also the case in the hypothalamus, so subcortically. And so we used a knockout of SRF, a conditional knockout, to block the ability of the hypothalamus now to activate immediate early genes and ask whether we could block this plasticity response. So as before, the behavior is the same. We're optogenetically activating the region to try to elicit these avoidance behaviors, and then we do this before and after social defeat. And we're doing this now in animals in which we have selectively knocked out SRF using a Cre-expressing virus just in the medial hypothalamus, nowhere else in the brain. And the data is at the bottom. It's just, I want to walk you through it because it's not, so you understand, because I'm now showing you the differential response. So it's the response to the light. So if you focus on the left side here, if you look at wild-type animals, so essentially there's no behavioral response to the light in a wild-type animal before the defeat. If you defeat the animals now, so after the defeat, that animal will show this reduction in social investigation. That's what I showed you before. So that's a defeat-dependent effect. But if you do this same experiment in an SRF knockout locally, you have no effect of the social defeat. And if you look at another measure, if you look at this avoidance, so cornering behavior, where there's a dramatic increase in avoidance behavior, again, in the SRF knockouts, you don't see this. And so I think this kind of suggests we're at the initial stages, but maybe a tool where we might be able to now kind of go around, potentially in a cell-type specific manner, and sort of see, well, where is the plasticity? And I do think this is something important in the neuroscience community. You know, we have lots of tools for looking at neural activity, GCamp is one of our favorite ones, where you see the acute kind of real-time changes in activity, but we have very few tools for watching or for manipulating plasticity. And I think we need more of those. And these experiments also kind of pushed us to think about other ways to actually visualize various types of plasticity. So this here on the right is a tool we've developed, been working on it for a number of years actually, in which we aim to try to quantify AMPA receptors, so classic AMPA receptor-dependent excitatory plasticity, by quantifying surface AMPA receptors. I think tomorrow when you hear the talk by Daniel, you'll get the full version of this, exactly what these AMPA receptors are doing. This is based on the fusion of the GluR1 subunit of AMPA receptors to the SNAP protein. You might have heard of that. It's an artificial protein and enzyme that's been engineered so that it covalently binds to a small molecule. It's called BG, benzoguanine, under physiological conditions. So it's an irreversible reaction, and you can hook up whatever you want to the BG. In this case, we put a fluorophore. And so if you perfuse the brain, we put this drug into the ventricles, actually. It rapidly perfuses in the extracellular space. This molecule is not cell permeable, and it binds only to surface AMPA receptors that have your SNAP ligand on it. And at the bottom, I'm just showing you what it looks like when you then crack open the brain. If you do this, in this case, these are on single neurons, you see this beautiful cytoplasmic staining, and at the tip, the spines, basically, you see this fluorophore binding, which presumably is proportional to the number of surface AMPA receptors. So we've been playing around with this. We actually made a mouse now, so this allows us to look kind of across the entire brain. The hope is to do sort of more unbiased measurements of AMPA receptor strength across the brain. Again, we're delivering this in the ventricles, and then we allow a certain period of time. I think right now we're working with sort of 2 hours. And this is, of course, done days after the behavioral experience, right? So in our case, you were looking at social defeat. The animals experience social defeat. You wait a couple days, then you perfuse the ligand to actually see whether there might be an increase or change or decrease in AMPA receptor in individual synapses, individual spots in various brain regions. Just to show you that, some validation here, this might work. We've actually done this in response to social defeat in the medial hypothalamus. I'm showing that on the left. And what we see is a significant reduction, actually, in the amount of surface AMPA receptors on average. And if you go and you do the kind of classic ex vivo slice electrophysiology recording, this was done by Ingrid Reverta in the Ragozzino group, you also see a reduction in classic AMPA-NMDA ratio, so potentially a kind of a correlation, a direct correlation between this imaging-based approach to look at AMPA receptors and a classic electrophysiological measurement. We're also interested in following plasticity in these brain regions in a more real-time way, right? So in the last method I showed you, of course, it's terminal. You're killing the animal. You get one time point. Can we look at synaptic plasticity? Can we follow it real time in behaving animals during these social experiences? And so we kind of go back. I mean, the classic way to do this was evoked LFP recordings. People have done this for decades, where you stimulate one part of the brain, you record another part of the brain, typically far away, and you look at the short latency responses, and people have used that. You can directly read out excitatory synaptic strength. You can follow learning in the hippocampus, for example, in that way. Really wonderful work by the Delgado Garcia lab many years ago. And so we wanted to try to do this also in a manner that would allow us to do this in specific cell types. And so we went to calcium imaging. And this is now miniscope recording, and this is now evoked by channel by Crimson, so by an optogenetics tool. This is now expression in the same soma in which we're recording, and as you can expect, if you shine the light to activate Crimson, you get a GCamp response in those cells. It's very robust, and you can measure the slopes of those as some sort of measure of the strength of that response over time. So can we use that to actually look at synaptic, at the strength of synaptic connectivity? So these are some initial results. Valeria actually had a poster just recently, so maybe you stopped by to see that. And what Valeria has shown that, for example, in VMH, if you look at inhibitory inputs or if you look at excitatory inputs, between the shell and the core, these two parts of the VMH, she can show these evoked responses, and she can actually find the first data that showing that actually the slopes of these responses changes over multiple days of social defeat and this doesn't happen in control animals that don't have this social experience. So the idea of trying to follow this in real time in animals that are freely behaving in these apparatuses and try to understand how this plasticity kind of evolves on a much more rapid time frame and how this occurs in different components of the circuit. Okay, my last slide. I hope I haven't gone too much over. But I just wanted to come back to the beginning where I was advocating for us to think more intensely or more deeply about these subcortical structures, ancient instinctive structures that are able to produce intense, important emotional states that we care about. Of course, they're coordinated with all of those wonderful forebrain and cortical areas that we also care about so much. And if you look at the anatomy, right, I mean, one of the major outputs of the basal ganglia cortical system, the forebrain system, is actually part of the hypothalamus. It's the substantia nigra. And that information is passed on and then merges at some point, actually at various points, but primarily in the medulla, in the MLR actually, with instinctive information that's coming through the PAG, for example, or superior colliculus, into the medulla. And so there must be some sort of integration, right? Our behavior is presumably some sort of sophisticated combination of what we would like to believe is willful cognitive behaviors or assessment, and then this drive underneath, this need, this initially subconscious need actually to act on our stimuli. And so I think that's where, I think there's a lot of excitement there. I'd like to understand how that works. And I do think that this has major implications for how we understand human behavior and pathologies. And just to kind of try to make it real, if you think about, as I wrote on the bottom here, if you think about speech or utterances, you can think of this kind of transition where you have vocalizations, so we can cry, we can whine, we can express our emotions through these very fundamental, also by the way, PAG-controlled behaviors, vocal behaviors, and that can transfer into something like song, which is presumably a mixture of cognitive and non-cognitive. You know, the musicians always tell you shouldn't be thinking. If you use your cortex too much while you're playing music, it's going to go. It's not going to jive. And but there must be that sweet spot of that kind of combination of speech. You are singing, after all. They are words. There is something cognitive, and the crying. The emotional crying. And at the other end, you have speech, which is intensely cognitive. So maybe that's a model for you to think about a little bit how other types of emotions and behaviors are integrated. Thank you very much, and I'd be happy to follow up on some of that.
Thank you very much, Cornelius, for this fantastic talk. There are no questions at plenary, but I'm sure that if people want to ask questions here in the corridors, Cornelius will be ready to ask. Thank you again.
Grazie, tutto bene. Tutto a posto. Grazie veramente, lo so che ogni tanto ci sono i disastri. No, non di colpa nostra.